Dear all, we are going live now.
College is often viewed as a mere institution, a labyrinth of classrooms, textbooks, and endless exams, where our future is shaped. Yet, when we step foot on campus, we realize that we are not alone on this journey. University transcends academia, transforming into a vibrant community where individuals unite. To learn from one another, to grow and to forge unforgettable bonds. It's not always about having a precise plan. It's not always about figuring out. It's not always about reaching the finish line first. It's about discovering what we are truly passionate about. The true essence of college, however, lies beyond the confines of academic pursuits. What truly makes college so special is the life that's lived within its walls. It's about seeing things from different perspectives, exploring unique possibilities, taking on new challenges, finding joy in friendship, embracing individuality, expanding our knowledge and skills, and immersing ourselves in extraordinary experiences. It's a path that teaches us about resilience, courage, and determination. A quest that allows us to pursue our goals with purpose and clarity. A quest that helps us find balance, happiness, and fulfillment. A transformation that inspires us to live with intention, curiosity, and joy. A trail that leads us to a deeper understanding of who we are and what truly matters. It's a journey of self-discovery, where every experience shapes us into the best version of ourselves. We attend college for the education, but what we live with is a treasure trove of experiences that shape us forever. Experience a whole new world of academic excellence in a global environment. Daya Sagar University, inspiring future trailblazers today.
Good evening, students. Welcome to Daya. Dear students, today you are going to have biology class of Professor Pavan Kumar. Topic is going to be evolution and strategies for enhancement in the food production. Dear student, I welcome you wholeheartedly. I request you to be very attentive and interactive. As usual, I am really appreciating students. You are really putting your efforts. I appreciate for that. I could able to see your questions in Q&A. I welcome you and I'll hand over a session to Professor Pavan Kumar. Sir.
Yes, sir.
These students will post their questions in Q&A box.
Okay.
Can keep please checking Q&A box and can respond either by audio or in Q&A, sir.
Sure.
Thank you so much and over to you, sir.
Thank you. Yeah. Good afternoon, students. So let us start with the chapter called evolution. As sir told, there will be two chapters today. One is evolution and the other one is strategies for enhancement in food production. Before I proceed further, I just need a quick reply from any one of you. Is it audible? Am I audible? And can you see the screen with the heading evolution?
You are audible and screen is clear, sir.
Yeah, thank you. So what you have studied, students, in this chapter is basically these 5 concepts. That is, one is theory of origin of life. Next, you have seen about what are the evidences for evolution as it was proposed by Darwin. What are the theories of evolution? Brief account of evolution, which starts from the beginning of the time, or rather beginning of Earth, and right now, what are the different stages of evolution that we can see. And finally, we will look into human evolution. So the first part that you have studied in this is about origin of life. When you are studying about origin of life, first thing is important to understand about origin of Earth itself. Earth ಮೊದಲು ಅಂದ್ರೆ ಭೂಮಿ ಯಾವ ತರ ಫಾರ್ಮ್ ಆಯ್ತು ಅಂತ. So for formation of Earth or anything else in this universe, it all started with something called as Big Bang. And what does Big Bang theory states is, everything started from a singular large explosion. When you say everything, you mean even time, atoms, elements, electrons, everything started from this singular large explosion, which is unimaginable in physical terms, which liberated large amount of energy, and that gradually led to formation of different elements, molecules, etc. As it went on, gradually universe started expanding, and the temperature went on reducing. It started cooling down. Within few fractions of second after Big Bang, what it was formed was hydrogen and helium was formed. Gradually, gases condensed under gravitation, and they started forming different galaxies that you see today in the universe. And one such galaxy that was formed was Milky Way. Within that Milky Way, what was formed was solar system. And within that solar system, what was formed was Earth. And this Earth was formed around 4.5 billion years back. ಸುಮಾರು 4.5 ಬಿಲಿಯನ್ ವರ್ಷದ ಹಿಂದೆ ಅರ್ತ್ ಅನ್ನೋದು ಮೊದಲು ಹುಟ್ಕೊಳ್ತು. How was the early Earth's atmosphere? When the Earth was formed in the beginning, how was the Earth's atmosphere? There was a lot of water vapor, which obviously is because the temperature of Earth was very hot compared to today. ಇವತ್ತಿಗೆ ಕಂಪೇರ್ ಮಾಡಿಕೊಂಡ್ರೆ ಟೆಂಪರೇಚರ್ ಆಫ್ ಅರ್ತ್ ತುಂಬಾ ಜಾಸ್ತಿ ಇತ್ತು. Most importantly, Earth gas had a lot of methane. And why methane plays an important role is this one. Methane is CH4. Methane is CH4. And for evolutionists, why methane is considered as an important component is, if there are certain substitution, like hydrogen, if it is substituted by carboxylic group, and another hydrogen is substituted by amino group. Now this leads to formation of simple amino acids. And formation of amino acid means it is beginning to form simple organic molecules. Now, depending upon the different types of organic acids, different types of functional group, it started forming different amino acids. But why methane plays in such an important role is because methane might have led to formation of different organic molecules that we see today. It had amino acid, and these gases were released in the from molten mass, which was covered the surface. UV rays from sun broke up water. Please understand this. There was no free oxygen when the Earth was formed. ಭೂಮಿ ಮೊದಲು ಫಾರ್ಮ್ ಆದಾಗ ಫ್ರೀ ಆಕ್ಸಿಜನ್ ಇರಲಿಲ್ಲ. ಆಕ್ಸಿಜನ್ ಇತ್ತು, ಬಟ್ ಇಟ್ ವಾಸ್ ಕಂಬೈನ್ಡ್ ಇನ್ ಅದರ್ ಫಾರ್ಮ್ ಲೈಕ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್, or it was in water or something like that. If oxygen was not present, obviously we can assume there was no ozone also. If ozone was not present, it means UV rays easily entered into the Earth's atmosphere. I hope you know that UV rays are powerful radiations. or it will be having energy more than the visible light, and that light energy or energy in UV was used to break up hydrogen and oxygen, and gradually hydrogen started escaping. Oxygen, meanwhile, combined with ammonia and methane to form water, carbon dioxide, and others. As it cooled, the water vapor fell as rain to fill up the depression in the form of oceans. Gradually, oceans started forming, and then life appeared around 500 million years after the formation of Earth, which means Earth was formed 4.5 billion years back. 0.5 is nothing but 500 million years, and minus 0.5, it will be 4 billion years back. 4 billion years ago, the first life form originated. Now, once if the Earth was formed, how was the life originated? For that, there are different theories. Out of that, the first theory that you have studied is about spontaneous generation, or theory of spontaneous generation. What does theory of spontaneous generation says is, life originated from abiotic components, or life originated from inanimate substances, or you can say life originated from non-living things such as soil, water, dead body of an organism. So that is what it is called as theory of spontaneous generation. Second one is cosmic theory, which is also called as theory of panspermia, which states that life on planet Earth came from some extraterrestrial objects in the form of spores, not in the form of humans or plants or anything. Yaudo microorganism tara, yaudo Akasha Kayada Rupadali, it has entered into the earth and then it started evolving. That is what the cosmic theory of panspermia states. Then this theory of special creation, theory of special creation, you can say that every life on planet Earth, including Earth, was formed by some supernatural power, which we call it as God. That is theory of special creation. And then you have studied about theory of chemical evolution. And the theory of chemical evolution, which is the most accepted one, it says that various types of inorganic molecules which were present on the Earth's atmosphere, they gradually started reacting each other and then led to formation of organic molecules. Once when the organic molecules were there formed, organic molecules started interacting with each other and they started forming life-like organism or living-like organism, life-like organism or living-like organism, and from there different complex organisms started evolving. That is what the most acceptable theory says, that is theory of chemical evolution. Now, what does theory of spontaneous generation says? As I told you, it originates from inanimate objects. Like on olden days, people were observing that some living organisms used to come out of non-living things. For example, right now it is summer. You can see that ponds are all dried up, and the moment when rain appears or moment when it starts raining, you can see frog. So what earlier scientists or earlier philosophers thought that from soil, frog is arising, in the sense frog is formed from soil. If they have stored some grains, rat used to come out of those grains. So it was assumed on those days that these non-living things will give rise to living organisms. And it was Louis Pasteur who demonstrated that life comes only from the pre-existing life. And he disproved this theory of spontaneous generation. He said, no, it is not possible for non-living things to give rise to any living organisms. It should come from already pre-existing life, but not from non-living things. And this he showed it by pre-sterilizing the flask. When you say sterilization, it means he killed all the microorganisms. And then he left it he added medium inside it, which was sterilized. And then he saw that life did not come until and unless the flask was open. Life originated only after flask was open, which means some of the living organism from outside, they went into the medium, and then living organism came. So the point is, theory of spontaneous generation was disproved by Louis Pasteur. See, I saw somebody raising the hand. If you have any doubts, please just message it in Q&A box. As you know, you cannot ask question through audio. Please type your questions if you have any. Don't raise the hands. Now, the second theory is theory of panspermia. I told you theory of panspermia says that life originated or life came into Earth rather than originated. Life came into Earth in the form of seeds. When you say these seeds, it was in the form of certain spores. And these spores came in the form of some extraterrestrial objects like meteoroids or asteroids. They brought in some spore-like living organisms, which gradually evolved into all the organisms that we see today. The third theory is theory of special creation, which states that living and non-living things, both entire Earth and all the living organisms that we see today, it was created by some supernatural power, which you can consider it as God. Theory of special creation also has certain assumptions like the Earth as well as all the living organisms were formed around 4,000 years back. But we know that in India alone, we have written history, or at least we have some kind of history of 5,000 years back from Harappa and Mohenjo-daro. So 4,000 years back, entire thing was created. That's what the theory of special creation states. And it also says that all the living organisms that we see today on the planet Earth, it has remained same. It has not changed, and it will not change in further also. ಹಿಂದೆ ಹೆಂಗಿತ್ತೋ ಹಂಗೆ ಇರುತ್ತೆ, ಮುಂದೆನೂ ಹಾಗೆ ಇರುತ್ತೆ ಅಂತ. ಬರೀ 4,000 ವರ್ಷದಿಂದ ಎಲ್ಲಾ ವಸ್ತುಗಳು, ಲಿವಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ಗಳಾಗ್ಲಿ, ನಿರ್ಜೀವಿಗಳ ವಸ್ತುಗಳಾಗ್ಲಿ, ಅದೆಲ್ಲದನ್ನು ಬಂದಿದ್ದು ಬರೀ 4,000 ವರ್ಷದ ಹಿಂದೆ ಅನ್ನೋದು ಥಿಯರಿ ಆಫ್ ಸ್ಪೆಷಲ್ ಕ್ರಿಯೇಷನ್ ವಾದ. But the most accepted theory is theory of chemical evolution. As I told you, what do you mean by theory of chemical evolution is that this was first, keep this in mind, that this was first proposed by two scientists called as Oparin and Aldin. Now, what did Oparin and Aldin said was Oparin and the other one was Aldin. What was the proposal from these two was that Earth's atmosphere was reducing, but it was not oxidizing. Why it was not oxidizing but reducing was, as I told you earlier, oxygen was absent in the free form. Please do not confuse. I'm not telling oxygen was completely absent. Oxygen was present, but not in the free form, but it was present along with some other molecules. By the interaction of elements and compounds, various organic compounds like aldehydes, sugars, fatty acids, lactic acid, ketones, acetates, and amino acids were gradually started forming. And one of the reasons for this was gradual appearance of gradual appearance of all these molecules was due to reduction in the temperature. Chandru S. Chakre, first photosynthetic organisms to appear on the planet Earth were. See, if you look at the stages of evolution, the first evolutionary products were prokaryotic organism, especially the archaebacteria. The first one to come out were archaebacteria. Now, as prokaryotes were formed, you know, the most primitive ones are prokaryotes because they do not have nucleus. Gradually, what started evolving was eukaryotic organisms, which had true nucleus in them. Now, the first photosynthetic organisms that evolved were basically cyanobacteria, uh, which were prokaryotic organisms. But there are some theories which also states that there were some non or anoxygenic photosynthetic organisms, which means they were not producing oxygen. So the organisms which were able to perform photosynthesis at the same time, they were not able to release oxygen were bacteria. But the first photosynthetic organisms to evolve on planet Earth which were able to release oxygen were cyanobacteria. Cyanobacteria's evolution was important because that was the one which led to release of oxygen or release of free oxygen in the atmosphere. I hope Chandru, yes, Chakre, you got this. Now, moving forward, what we were looking was that gradually these different types of organic acids started forming, or organic molecules started forming. Now, for all this reaction, the most important requirement was energy. And energy for all this reaction came in the form of heat. And that heat probably came by volcanic eruption. Also, the electric discharge, which was in the form of lightning. ತುಂಬಾ ಮಳೆ ಆಗ್ತಾ ಇತ್ತು, ವಾಲ್ಕ್ಯಾನಿಕ್ ಎರಪ್ಷನ್ ಇರ್ತಿತ್ತು, ಮಿಂಚುಗಳು ಜಾಸ್ತಿ ಬರ್ತಾ ಇತ್ತು. ಜೊತೆಗೆ along with that, we also know that Earth's early atmosphere did not add ozone. Since there was no ozone, UV radiations were also easily entering, which also contributed for energy on the planet Earth for all this chemical reaction. After this, remember I told you last slide that theory of organic or theory of chemical evolution was given by Oparin and Aldin. It was given by Oparin and Aldin. So that Oparin's theory was experimentally proved by Stanley Miller. In fact, there is one more name associated with that, that is Urey and Miller. Urey and Miller together, they started experiments which were meant for proving the Oparin's theory. So, you know, this experiment is also famously called as spark discharge experiment. What they basically did was they tried to recreate the early Earth's atmosphere. Like we saw early Earth's atmosphere had gases such as methane, ammonia, water, hydrogen. At the same time, it was high temperature for which they used boiling water. It was reducing atmosphere, and hence they removed all the gas. In the sense, they removed all oxygen, but basically they removed all the gas, and then they filled up with these gas, that is ammonia, methane, and hydrogen. And then they kept these two electrodes to stimulate lightning. ಈಗ ತಾನೇ ಹೇಳಿದ್ದೆ ನಿಮ್ಗೆ, ಲೈಟ್ನಿಂಗ್ ಇರೋದ್ರಿಂದ ಒಂದು ಎನರ್ಜಿ ಸೋರ್ಸ್ ಇತ್ತು, ಹೀಟ್ ಇರೋದ್ರ ಟೆಂಪರೇಚರ್ ಜಾಸ್ತಿ ಇದ್ದಿದ್ರಿಂದ ಹೀಟ್ ಇಂದು ಕೂಡ ಎನರ್ಜಿ ಸೋರ್ಸ್ ಇತ್ತು ಅಂತ. ಸೊ ಅದೆರಡನ್ನು ದೇ ರೀಕ್ರಿಯೇಟೆಡ್ ಇಟ್, ಅಂಡ್ ದೇ ಆಲ್ಸೋ filled up with various kinds of gases inside it. Now this, there was at boiling of water vapor came into this, reacted with different types of gases, and then it condensed here because they were putting in cold water and removing it, condensed, and water containing organic compounds were formed within this. Within few weeks, ಕೆಲವೇ ವಾರಗಳಲ್ಲಿ they were able to see some organic molecule. ಮತ್ತೊಂದ್ಸರಿ ಹೇಳ್ತಾ ಇದ್ದೀನಿ, ಕೆಲವೇ ವಾರಗಳಲ್ಲಿ organic molecules ಗಳನ್ನ ಅವ್ರು ನೋಡ್ತಾರೆ. And the organic molecules that they found inside this liquid water in trap, it contained urea, lactic acid, sugar, acetic acid, amino acids such as glycine, glutamate, alanine, aspartate. ಈ ತರದ ಸಿಂಪಲ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಗಳನ್ನು ಅದರೊಳಗಡೆ ನೋಡ್ತಾರೆ. I just want to again draw your attention to this, that this was just kept for few weeks. ಕೆಲವೇ ತಿಂಗಳಲ್ಲಿ ಈ ತರ ನೋಡೋದಾದ್ರೆ, according to one of the earlier slide that we saw, life took around 500 million years to originate. ಅದರ ಅರ್ಥ 500 ಮಿಲಿಯನ್ ವರ್ಷಗಳಲ್ಲಿ ಏನೇನ್ ಆಗಿರ್ಬೋದು ಅನ್ನೋದನ್ನ you can imagine this. So this was one of the strong evidence to accept that life originated first time on the planet Earth in the form of or through the uh, organic molecules which are formed from inorganic molecules. So this was the one which is widely accepted. Then you have studied about evolution of life form, which were the different theory. One of such early theories was proposed by Charles Darwin. Charles Darwin went around the earth for about 5 years in a ship called as HMS Beagle. HMS Beagle ಅನ್ನೋ ಹಡಗಲ್ಲಿ ಸುಮಾರು 5 ವರ್ಷ ಪ್ರಪಂಚ ಪರ್ಯಟನೆ ಮಾಡ್ತಾರೆ. And he collected various types of specimen. He studied various type of specimen, and then he came back to England. Once after coming back to England, he spent his entire life studying about this evolution. So he gave what is called as, or he wrote a book called as Origin of Species. I hope we remember this book, which is considered as one of the greatest book, which is Origin of Species. And Darwin, in that book, talks about what is called as fitness, or also called as survival of fittest. Please understand that fitness, according to Darwin, is not about physical fitness. Gatti muttagirodalla, jasti muscles ಬೆಳೆಸ್ಕೊಳ್ಳೋದಲ್ಲ. Fitness, according to Darwin, is about the fitness of an organism to survive in that environment and ultimately leave more progenies. I'm sure you people understand what you mean by progenies. Fitness ಅಂತ ಹೇಳಿದ್ರೆ ಸರ್ವೈವ್ ಆಗಿ ಎಷ್ಟು ಸಾಧ್ಯನೋ ಅಷ್ಟು ಹೊಸ ಹೊಸ ಆರ್ಗಾನಿಸಮ್ಸ್ ಗಳನ್ನ ಬಿಡೋದು. ಅಂದ್ರೆ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಫಿಟ್ನೆಸ್ ಅನ್ನ ಡಾರ್ವಿನ್ ಅವರು ಫಿಟ್ನೆಸ್ ಅಂತ ಹೇಳ್ತಾರೆ, ನಾಟ್ ದ ಸ್ಟ್ರಾಂಗೆಸ್ಟ್. So its ability of an organism to survive in that environment and produce more number of offspring. That is what fitness is, according to Darwin. So hence, those who are better fit in an environment leave more progeny than others. When you produce more progeny, if there is any population which is producing more progeny, that is what it is called as natural selection, and implied it as a mechanism of evolution. So according to Darwin, there were two important things that he observed or he proposed. One is natural selection, and the other one is called as branching with descent. These two were the strong propositions that were put forward by Darwin. So along with Darwin, almost equal contribution should go to one more scientist called as Alfred Wallace, who was a naturalist who worked in, in and around Philippines. There are hundreds of islands. All those hundreds of islands in and around Philippines to Australia or New Zealand, it is called as Malay Archipelago. He also came to similar conclusion at around the same time. So both of them together put forward this theory of fitness. Now, this is what you mean by natural selection. Now, if you carefully observe this diagram, there is some, what can you say? You can say dark gray color rock. On this dark gray color rock, there are two population of mice. One is again gray, one is gray, and the other one is somewhat pink. There is some selection pressure here in the form of eagle. Now, what can happen is eagle can easily spot these pink color mice than that of the gray color mice. Now, observe what is happening here. The number of gray color mice is 3 here, whereas number of pink color mice is around 6 in the beginning. But some mice are eaten by birds. So chances of this bird eating mice is more for pink rather than gray, which means gradually what happens is the population of pink mice reduces, and this will be considered as fit now, no matter how strong is it, no matter whether it can run faster, no matter whether it is going to eat less or more, less muscle or more muscle. But the fit among these two mice is gray because the environment So for this environment, the color of this mice is suitable and that will survive and produces more progeny. Whereas these progenies, which are pink color progeny, it keeps on reducing. It is not fit for natural selection. So ultimately, from 6 pink color mice, after a few months or years, you can see only 2 are there, but the number of gray color mice that has increased to 6. So this is what you mean by natural selection according to Darwin. See, I don't want to keep on telling this again. I can see some of you people raising hands. By raising hand, it will be of no use. So please make sure that you type your questions in your Q&A box. So if Darwin and Wallace said that life or different species evolve because of natural selection and branching with descent, what are the evidences that we have for this? How can you agree that this is true? ಇದಕ್ಕೆ ಆಧಾರ ಏನು ಅಂತ ಹುಡುಕಿದಾಗ so some of the evidences for all this evolution comes in the following types. One, it is evidence of evolution through paleontological evidence. You know what do you mean by paleontology? Paleontology is study of fossils. Fossils ಅಂತ ಹೇಳಿದ್ರೆ ಪಳೆಯುಳಿಕೆಗಳು. ಆ ಪಳೆಯುಳಿಕೆಗಳನ್ನ ಅಧ್ಯಯನ ಮಾಡೋದನ್ನ it is called as paleontology ಅಂತ ಹೇಳ್ತಾರೆ. Now, why paleontology is so important? Paleontology gives us an idea about how or what is the age of the fossil. Age of the fossil ಅರ್ಥ ಆಗೋದ್ರಿಂದ ಏನು ಅರ್ಥ ಆಗುತ್ತೆ ನಿಮಗೆ? We can understand how many years ago this animal was surviving. ಈಗಿಲ್ಲ ಡೈನೋಸರಸ್, ಬಟ್ ಡೈನೋಸರಸ್ಗಳದ ಫಾಸಿಲ್ ಸಿಗುತ್ತೆ ನಮಗೆ. There are techniques such as carbon dating, which will help us to identify what is the approximate age of this fossil, which means we can understand by continuously subjecting all the fossils of dinosaurs to carbon dating, we can know what is the age of this dinosaurs period is. ಯಾವ ವರ್ಷದ ಹಿಂದೆ, ಎಷ್ಟು ಮಿಲಿಯನ್ ವರ್ಷದ ಹಿಂದೆ ಈ ಡೈನೋಸರಸ್ಗಳು ಇದ್ವು ಅನ್ನೋದು ಗೊತ್ತಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ there is only one evidence for us, that is in the form of fossils. It is not only it gives us approximate age of these fossils or how many years back these organisms were surviving. ಈ ಫಾಸಿಲ್ಸ್ ಗಳನ್ನ ಅಧ್ಯಯನ ಮಾಡೋದ್ರಿಂದ ನಮ್ಗೆ ಇನ್ನೊಂದು ಏನ್ ಸಿಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಬೇರೆ ಬೇರೆ ಫಾಸಿಲ್ಸ್ ಗಳನ್ನ ಕಂಪೇರ್ ಮಾಡೋದ್ರಿಂದ, ಇಫ್ ಯು ಕೀಪ್ ಆನ್ ಕಂಪೇರಿಂಗ್ ಡಿಫರೆಂಟ್ ಫಾಸಿಲ್ಸ್, ಯು ಕ್ಯಾನ್ ಹ್ಯಾವ್ ಅನ್ ಐಡಿಯಾ ಲೈಕ್ ವಿಚ್ ಆರ್ಗ್ಯಾನಿಸಮ್ evolved from what. For example, you say Homo sapiens, that is we evolved from Homo erectus anta heltare, but we don't have Homo erectus today. And it is said that Homo erectus evolved from Homo habilis. So what is the evidence now? Evidence ಹೆಂಗ್ ಸಿಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ by comparing the fossil structure of Homo habilis with fossil structure of Homo erectus, we can know what are the changes that has taken place and how many years back these fossils were present. ಅದರಿಂದ ಒಂದು ಸ್ಟ್ರಾಂಗ್ ಎವಿಡೆನ್ಸ್ ನಮ್ಗೆ ಯಾವ್ದು ಸಿಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಎವಲ್ಯೂಷನ್ಗೆ, it will be in the form of fossils. This is what I was telling. Fossils ಗಳನ್ನ ಅಧ್ಯಯನ ಮಾಡ್ತಾ ಹೋದ್ರೆ ಈ ಕುದುರೆ, it has evolved from its ancestor called as Eohippus ಅಂತ ಕರೀತಾರೆ. Now, how it has evolved, how the shape of its forelimb or hind limb has changed over the years. ಇಷ್ಟು ವರ್ಷದಿಂದ ಹೆಂಗ್ ಚೇಂಜ್ ಆಯ್ತು ಅಂತ ಗೊತ್ತಾಗ್ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ it comes from fossils. This is again one more importance of fossil. We say that birds evolved from reptiles ಅಂತ. ಬರ್ಡ್ಸ್ evolve ಯಾವ್ದ್ರಿಂದ ಆಗಿದ್ದು ಅಂತ ಹೇಳಿದ್ರೆ ರೆಪ್ಟೈಲ್ಸ್ ಇಂದ. ಅದಕ್ಕೆ ಎವಿಡೆನ್ಸ್ ಏನಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ it is the fossils of Archaeopteryx, wherein you can say ಫೆದರ್ ತರ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಇತ್ತು, ಅದರ ಬಾಯಲ್ಲಿ ಬೀಕ್ ಗಳಿದ್ವು, ರೆಕ್ಕೆಗಳಿದ್ವು. ಇದೆಲ್ಲದನ್ನು ಯು ಫೌಂಡ್ ಇಟ್ ಇನ್ ಡೈನೋಸರಸ್ ವಿಚ್ ಆರ್ ಆಕ್ಚುಲಿ ಇನ್ ರೆಪ್ಟೈಲ್. ಬಟ್ ಈ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಗಳೇನಿದೆಯೋ ಅದೆಲ್ಲ ಕ್ಯಾರೆಕ್ಟರ್ಗಳು ಬರ್ಡ್ಸ್ ಇನ್. ಸೊ ಏನ್ ಹೇಳ್ಬೋದು ಇವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ ವಿ ಕ್ಯಾನ್ ಕಮ್ ಟು ಅನ್ ಕನ್ಕ್ಲೂಷನ್ ದಟ್ ಬರ್ಡ್ಸ್ ಎವಾಲ್ವ್ಡ್ ಫ್ರಮ್ ರೆಪ್ಟೈಲ್ಸ್ ಅಂತ. ಸೊ ಅಗೈನ್ ನಿಮ್ ಬುಕ್ ಅಲ್ಲಿ ಡಯಾಗ್ರಾಮ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಆಲ್ ದಿಸ್ ಕನ್ಕ್ಲೂಷನ್ ಯು ಕ್ಯಾನ್ ಮೇಕ್ ಇಟ್ ಫ್ರಮ್ ಸ್ಟಡಿಂಗ್ ದ ಫಾಸಲ್ಸ್. So that is first evidence, that is paleontological evidence. Second evidence is morphological and anatomical evidence. I hope you know that this is going to be important. Morphological and anatomical evidence nalli ನೀವು ನೋಡೋದು comparative anatomy of morphology, comparison of anatomy as well as morphology. ಅದನ್ನು ಓದುವಾಗ ನೀವು ಎರಡು ತರದ ಆರ್ಗನ್ ಓದಿದ್ದೀರಾ. You have studied two types of organs in them. One is homologous organs and the other one is analogous organs. Now, what do you mean by homologous organs? These are the organs which are having fundamental similarities in structure and origin, but they perform different function. To be put in even more simple sentence, those organs which are structurally similar but performs different function. Structure onde tara irutte, function bere tara idre, then you call it as homologous organ, and its phenomenon is called as homology. Examples is human ant. ಇಲ್ಲಿ ನೋಡಿ, ಹ್ಯೂಮನ್ ಆ್ಯಂಡ್ ಅಲ್ಲೂ ಹ್ಯೂಮರಸ್ ಇರುತ್ತೆ, ರೇಡಿಯಸ್ ಮತ್ತೆ ಅಲ್ನಾ ಇರುತ್ತೆ, ಕಾರ್ಪಲ್ಸ್ ಗಳಿರುತ್ತೆ, ಮೆಟಾಕಾರ್ಪಲ್ಸ್ ಗಳಿರುತ್ತೆ, ಫೆಲಾಂಜಸ್ ಗಳಿರುತ್ತೆ. ಸೇಮ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಯು ಕ್ಯಾನ್ ಸಿ ಇನ್ ಕೇಸ್ ಆಫ್ ಕ್ಯಾಟ್ ಆಲ್ಸೋ, ಯು ಕ್ಯಾನ್ ಸಿ ಇಟ್ ಇನ್ ವೇಲ್ ಆಲ್ಸೋ, ಯು ಕ್ಯಾನ್ ಸಿ ಇಟ್ ಇನ್ ಬ್ಯಾಟ್ ಆಲ್ಸೋ. ರೇಡಿಯಸ್ ಅಂಡ್ ಅಲ್ನಾ, ರೇಡಿಯಸ್ ಅಂಡ್ ಅಲ್ನಾ, ರೇಡಿಯಸ್ ಅಂಡ್ ಅಲ್ನಾ, ಕಾರ್ಪಲ್ಸ್, 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 and metacarpals, 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 which means we can see what is called as structural similarities. But we humans, we use our ant for certain work, cat use it for running, whale use it for swimming, bat use it for flying, which means structurally they are all similar, but functionally they are different. Then you call it as homologous organs. One more example for homologous organs is this one, the diagram which is also given in your book. One is thorns of bougainvillea and tendrils of cucurbita. And I'm sure most of you know this. If there is a stem like this, and if you find a leaf over here, at the base of the leaf, you can see something called as axillary bud. ಜ್ಞಾಪಕ ಇರ್ಬೋದು, ಇದನ್ನ axillary bud ಅಂತ ಕರೀತಾರೆ. Axillary bud indane thorn barodu. I hope you know from morphology of flowering plant allu odirtira. Or axillary bud modifies into thorn, axillary bud modifies into tendrils also, which means they are all, both of them are similar in origin. I'm repeating, both of them are similar in origin because both have originated from axillary bud, but they perform different functions. Thorns are going to meant for protection, whereas tendril in cucurbita it is meant for supporting. So this is again important. Homologous organs andre ಏನು ಅಂತ ಅರ್ಥ ಮಾಡ್ಕೊಂಡಿರ್ತೀರಾ, that is organs which are similar in structure or origin but perform different function. But what does homologous organ explains? Homologous organs explains what is called as divergent evolution. What do you mean by divergent evolution? From a common ancestor, if different populations or different species, ಒಂದು ಕಾಮನ್ ಆಗಿರೋ ಪೂರ್ವಜರಿಂದ ಬೇರೆ ಬೇರೆ ತರದ ಸ್ಪೀಸೀಸ್ ಗಳೇ ಎವಾಲ್ವ್ ಆದ್ರೆ ಅದನ್ನ ಡೈವರ್ಜೆಂಟ್ ಎವಲ್ಯೂಷನ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಅದನ್ನ ಡೈವರ್ಜೆಂಟ್ ಎವಲ್ಯೂಷನ್ ಅಂತ ಕರೀತಾರೆ. ಡೈವರ್ಜೆಂಟ್ ಎವಲ್ಯೂಷನ್ ಅಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, ವಾಟ್ ಎವರ್ ದ ಕೈಂಡ್ ಆಫ್ anatomical structure is found in its ancestor, whatever anatomical structures is found in the ancestor, same anatomical structure it will modify in certain way which will be suitable for their environment, and thereby they undergo natural selection. So which means the similar structure will be present, but functionally they will be different, and hence you can see there is a divergent evolution. Physics allu odirtira, ಒಂದು ಕಾಮನ್ ಪಾಯಿಂಟ್ ಇಂದ ರೇ ಎಲ್ಲಾ ಕಡೆಗೂ ಎಮರ್ಜ್ ಆಗ್ತಾ ಇದ್ರೆ ಯು ಕಾಲ್ ಇಟ್ ಆಸ್ ಡೈವರ್ಜೆಂಟ್ ರೇ ಅಂತ. ಅದೇ ತರ ಒಂದು ಕಾಮನ್ ancestor in the ಬೇರೆ species ಗಳು evolve ಆಗೋದನ್ನ it is called as divergent evolution. Please keep this in mind that homologous organs will give us the explanation or give us the evidence for divergent evolution. Next type of morphological and anatomical evidence that we have seen is analogous organs. What do you mean by analogous organs? These organs have similar function, but structurally they are different. Homologous structure it will be similar, but function it will be different. But it will be having similar function, but structurally it will be different. For example, wings of birds, it will be having humerus, radius, ulna, carpals, metacarpals, ella irutte. Whereas the wings of a butterfly, it is just a flap. It is just an extension of exoskeleton. Nothing else will be there. So, which means structurally they are both different, but they'll be performing same functions. This kind of analogy, this kind of evidence gives us what is called as convergent evolution. For example, carefully understand this. Shark is a fish. Penguin, you know, it is a bird. Dolphin is a mammal, which means they're all different organisms. Muru bere classiಗೆ ಸೇರುತ್ತೆ. Fishes belongs to pieces, birds belongs to aves, mammals belongs to, that is dolphin belongs to mammals. ಮೂರು ಬೇರೆ ತರ. But ಅವುಗಳ ಸ್ಟ್ರಕ್ಚರ್ ಏನಾಗಿರುತ್ತೆ ಅಂದ್ರೆ, if you look at their modifications, they will be all having similar structures. Sorry, they will be all having similar function. ಯಾಕೆ ಅಂತ ಹೇಳಿದ್ರೆ ಶಾರ್ಕು ನೀರಲ್ಲಿರುತ್ತೆ, ಪೆಂಗ್ವಿನ್ನು ನೀರಲ್ಲಿರುತ್ತೆ, ಡಾಲ್ಫಿನ್ನು ನೀರಲ್ಲಿರುತ್ತೆ, ವೇಲು ನೀರಲ್ಲಿರುತ್ತೆ. ಅದರ ಅರ್ಥ ಅದೆಲ್ಲದಕ್ಕೂ ಕಾಮನ್ ಡಿಮ್ಯಾಂಡ್ ಇರುತ್ತೆ. All of them will be having common demand, and hence they will all develop into similar structure. Sorry, they will all develop into similar function, but structurally they may be different. This kind of evolution is called as divergent. Sorry, this kind of evolution is called as convergent evolution anta karitare. So I'm repeating this once again. There are two types of organs. One is called as homologous organs, and the other one is called as analogous organs. From homologous organs, what is the evidence that we get is, from homologous organs we will get what is called as evidence for divergent evolution, but from analogous organs we'll get what is called as convergent evolution. I hope you people have understood what is homologous, analogous, and what is divergent evolution and convergent evolution. Similarly, here again, if you look at the structure of shark's fin and flippers of dolphin, they both perform same function, but structurally they are different. Dolphin in a fin nalli, sorry, shark in a fin nalli, nimge humerus, radius, ulna, idenu iralla shark alli. But dolphin in a flippers galalli adirutte, which means structurally they are different. But both of them are meant for performing same function, that is for swimming. ಏನಕ್ಕೆ ಆ ತರ ಬರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, they both are adapted to similar environment. And which is that similar environment in this case is, ಈ ಕೇಸಲ್ಲಿ similar environment ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ವಾಟರ್ ನಲ್ಲಿ ಬದುಕೋದು. That is about morphological and anatomical evidence. Let us look into adaptive radiation. What do you mean by adaptive radiation? It is a process of evolution of different species in a given geographical area, starting from a point and literally radiating to other areas of the geography. It is called as adaptive radiation. Of course, process takes place through natural selection. Examples are, you can understand adaptive radiation by looking into examples of adaptive radiation, such as Darwin's finches, Australian marsupials, and also the placental mammals of Australia. Now look at this. There was one common ancestor which was present in the Galapagos Island. But Galapagos Island nalli This common ancestor was eating seed anta ankoಣ. But as the population of this common ancestor increased, there was a lot of competition. ಯಾವ competition ಜಾಸ್ತಿ ಆಯ್ತು, there were some changes in the structure of the beak of this. Observe the structure of beak of these Darwin's finches. Seed eating ಗಳ finches ಬೇರೆ ತರ ಇದ್ರೆ, insect eaters ಗಳ finches ಬೇರೆ ಬೇರೆ ತರ ಇರುತ್ತೆ. ಆ ಇನ್ಸೆಕ್ಟ್ ಅನ್ನು ಎಲ್ಲಿ ತಿಂತಾ ಇದೆ ಅನ್ನೋದ್ರ ಮೇಲೂ beaks ಗಳು ಚೇಂಜ್ ಆಗುತ್ತೆ. Is it picking up those insects from earth or from the soil, or is it picking it from the leaf, or is it picking up from the wood, hard bark ಅನ್ನ ಕೊರೆದು ಕಿತ್ಕೊಂಡು ತಿಂತಾ ಇದ್ರೆ ಬೀಕ್ ಬೇರೆ ತರ ಇರುತ್ತೆ. ಬೇರೆ ಬೇರೆ ಸೀಡ್ ಗಳನ್ನ ತಿನ್ನುವಾಗ ಬೇರೆ ಬೇರೆ ತರದ ಸೀಡ್ ಈಟರ್ಸ್ ಗಳು ಎವಾಲ್ವ್ ಆಯ್ತು. ಕ್ಯಾಕ್ಟಸ್ ಸೀಡ್ಸ್ ಅನ್ನ ತಿನ್ನೋ ಫಿಂಚಸ್ ಗಳು ಬೀಕೆ ಚೇಂಜ್ ಆಯ್ತು. ಬಡ್ಸ್ ಮತ್ತು ಫ್ರೂಟ್ಸ್ ಗಳನ್ನ ತಿನ್ನುವ ಫಿಂಚಸ್ ಗಳ ಬೀಕು ಮತ್ತೆ ಚೇಂಜ್ ಆಯ್ತು. Now, if you go back to the definition that we saw, what does the definition says? It says evolution of different species, like different finches that we saw, in a same given geographical area, starting from a point, which was a starting point from here for Darwin's finches. Darwin's finches ಗೆ starting point ಯಾವ್ದಿತ್ತು ಅಂತ ಹೇಳಿದ್ರೆ seed eaters, and literally radiating ಅಂದ್ರೆ ಯಾವ್ದಾಯ್ತು sorry, maybe insect eaters, fruit eaters, ಬೇರೆ ಬೇರೆ ತರದ seed eaters. ಸೊ ಒಂದು ಕಾಮನ್ ಆನ್ಸೆಸ್ಟರ್ ಇಂದ ಒಂದೇ ಜಿಯೋಗ್ರಫಿಕಲ್ ಲೊಕೇಶನ್ ನಲ್ಲಿ ಬೇರೆ ಬೇರೆ ತರದ ಸ್ಪೀಸೀಸ್ ಗಳು ಎವಾಲ್ವ್ ಆಗ್ತಾ ಹೋಗೋದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್. ಅದೇ ತರ ಒನ್ ಮೋರ್ ಎಕ್ಸಾಂಪಲ್ ನೀವು ನೋಡಿರೋದು ಮಾರ್ಸುಪಿಯಲ್ ರೇಡಿಯೇಷನ್. ಯು ನೋ ಮಾರ್ಸುಪಿಯಲ್ಸ್ ಆರ್ ದೋಸ್ ಅನಿಮಲ್ಸ್ ವಿಚ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕೀಪ್ ದೇರ್ ಯಂಗ್ ಒನ್ಸ್ ಇನ್ ದೇರ್ ಪೌಚ್. ಸೊ ಆಸ್ಟ್ರೇಲಿಯಾದಲ್ಲಿ ಮಾರ್ಸುಪಿಯಲಿನ ಯಾವ್ದಾದ್ರು ಒಂದು ಕಾಮನ್ ಆನ್ಸೆಸ್ಟರ್ ಇಂದ, ಡಿಪೆಂಡಿಂಗ್ ಅಪಾನ್ ದ ಜಿಯೋಗ್ರಫಿಕಲ್ ಲೊಕೇಶನ್, some became hunters like this, some became anteaters, some became feeding on the soil, some became kangaroo. ಈ ತರ ಬೇರೆ ಬೇರೆ ಸ್ಪೀಸೀಸ್ ಗಳು ಎವಾಲ್ವ್ ಆಗೋದನ್ನ it is called as adaptive radiation. Again, this is one more important point, so please concentrate and understand this. When there are two adaptive radiation occurring in the same geographical area, it leads to what is called as convergent evolution. We have already discussed what do you mean by convergent evolution. Evolution of similar functioned organs, since they are living in the same area, it is called as convergent evolution. Placental mammals ಗಳು ಮತ್ತೆ ಆಸ್ಟ್ರೇಲಿಯನ್ mammals ಗಳು ಅಥವಾ ಮಾರ್ಸುಪಿಯಲ್ಸ್ ಗಳು ಎರಡು ಒಂದೇ ಜಾಗದಲ್ಲಿ ಬದುಕಿದಾಗ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಮಣ್ಣಿನ ಒಳಗಡೆ ಬಾರೋರ್ಸ್ ಮಣ್ಣನ್ನ ತೋಡ್ಕೊಂಡು ಬಿಲ ಮಾಡ್ಕೊಂಡು ಹೋಗೋದು, ಇದರ ಸ್ಟ್ರಕ್ಚರ್ ನ ನೋಡಿದ್ರೆ ಮಾರ್ಸುಪಿಯಲ್ ಮೋಲ್ಗಳ ಸ್ಟ್ರಕ್ಚರ್ ನ ನೋಡಿದ್ರೆ ಎರಡು ಬೇರೆ ಬೇರೆ ತರ ಇರುತ್ತೆ. ಬಟ್ ದೇರ್ ಆರ್ಗನ್ಸ್ ಆರ್ ಮಾಡಿಫೈಡ್ ಟು ಪರ್ಫಾರ್ಮ್ ಸೇಮ್ ಫಂಕ್ಷನ್ ಬಿಕಾಸ್ ದೇ ಬೋತ್ ಆರ್ ಅಡಾಪ್ಟೆಡ್ ಆಸ್ ಬಾರೋರ್ಸ್. ಅದೇ ತರ ಫ್ಲೈಯಿಂಗ್ ಸ್ಕ್ವಿರಲ್ಸ್ ದಟ್ ಯು ಫೈಂಡ್ ಇನ್ ಆಸ್ಟ್ರೇಲಿಯಾ ಅಂಡ್ ಫ್ಲೈಯಿಂಗ್ ಫೆಲಾಂಜಸ್ ದಟ್ ಯು ಫೈಂಡ್ ಇನ್ ಆಸ್ಟ್ರೇಲಿಯಾ, ದೇ ಶೋ analogous organs because you can see what is called as convergent evolution. When does the convergent evolution takes place? When two adaptive radiation taking place in the same geographical location, then it leads to what is called as adaptive radiation. What is the embryological evidence states? You may remember that embryological evidence was given by a scientist called as Ernst Haeckel. It was given by a scientist called as Ernst Haeckel. According to him, it says some of the embryonic states are common to all vertebrates, which means all these vertebrates have been evolved from a common ancestor. But however, this was disproved by a scientist called as Carl Ernst von Baer. If you observe this anatomical structure of them, ನಿಮ್ಗೆ ಇಲ್ಲಿ ಫಸ್ಟ್ ಮೂರ್ embryological structure ಏನ್ ಕಾಣಿಸ್ತಾ ಇದೆ, it is of fishes. Now if you compare the fishes early embryo with salamander, which is amphibian, with that of tortoise, which is a reptile, which that of chick, which is a bird, and all these mammals. Almost one day you can see very, very little changes, but not much significant changes. But in the next stage, you can see there is again, these two are closely related. Still, they resemble some amount of resemblance, which means fishes and reptiles, they resemble each other. Sorry, fishes and amphibians have some resemblance, and there is some amount of resemblance between the amphibians and reptiles. So, based on this observation, Ernst Haeckel gave this what is called as embryological evidence for evolution. And then we have evidence for evolution by natural selection. Natural selection, again, you example of notirodu industrial melanism anta odidira. You know, industrial melanism took place in England, and it is found in case of a moth called as biston or Byston Betularia. There were two populations of this biston. One was white-winged and the other one was dark-winged. Now, before industrial revolution or before industrialization in England, the population of white-winged were more, either jasti. Population of dark-winged were less. Before industrial revolution, the trees were covered with lichens. You know, lichens are pollution indicators. Yavaga pollution jasti aguto, lichens will vanish from that particular location, or the population of lichens is going to decrease. Industrialization agok modulu lichens gal jasti iddidrinda, so white-winged moths survived better than the dark color. ಏನಕ್ಕೆ ಅಂತ ಹೇಳಿದ್ರೆ ಮರನು ವೈಟ್ ಕಲರ್ ಇರುತ್ತೆ ಬಿಕಾಸ್ ಆಫ್ ಲೈಕನ್ಸ್. ಅದರ ಮೇಲೆ ಆನ್ ದೋಸ್ ವೈಟ್ ಕಲರ್ಡ್ trees, because of lichens, you can see white-winged moths before industrialization. But once after industrialization, what happened is gradually this lichens, population of lichens decreased. Lichens decreased. This is, I hope you are getting, this is before industrial revolution, this is after industrialization. Now, gradually what happened, the lichens population went on reducing. See, I don't know if it is visible on your screen. There is a white-winged moth here, which cannot be seen on the background because of this lichens, which means white-winged moth will jasti. Whereas after industrial development, again, I don't know if the resolution and clarity is there clearly on your this one. There is a black color moth here. The population of black-winged moth went on increasing after industrial revolution or industrialization. Yeah. So again, so if any students have any doubt, please feel free to ask at any time or during any time during this sessions. Don't hesitate to ask questions.
Yeah, student, if you have any doubts, you can put in Q&A box.
Yeah. So the last evidence for evolution came from artificial selection, which was based on humans as in selection pressure. So there were some ancestors which were grown by our early farmers. And depending upon certain characters which were useful for us, our ancestors went on selecting only those characters and neglecting other characters. And thereby different characters went on evolving from a common ancestor. Like for example, from an Brassica oleracea, which is a primitive or ancestor of all these vegetables. Some of our ancestors started selecting only the terminal bud, which went to evolve into what is called as cabbage. Some of them went on evolving, or some of them only selected flower, so that flower can be edible, and that became cauliflower. So this is what we call it as artificial selection. And in last part of this, we have studied about theory of biological evolution. One of the earliest theory came from a scientist called as Lamarck. It is also called as Lamarckism. And this theory is also called as theory of inheritance of acquired character. Now, the biggest problem for Lamarckism is this part. Acquired characters are not passed on from generation to generation. For example, if somebody has pierced their nose or ears, which is an acquired character, it will be not passed on to the next generation. Kivi ಮೂಗು ಚುಚ್ಚಿಸ್ಕೊಂಡಿದ್ರೆ ಅದು acquired character. ಅದು ಮುಂದಿನ ಜನರೇಷನ್ಗೆ ಹೋಗಲ್ಲ. Doberman, where you can see most of the generations they are going to cut the tail. Acquired character, absence of tail. But still, the newborn Doberman, it will be having tail in it. So, biggest drawback of Lamarckism is that he said some of the organs will be used more, sometimes some of them may be used less. Depending upon that, some of the organs will become strong, some of the organs will become weak. And thereby the strong organs keeps on evolving as in strong and more prominent organs, whereas the other one will disappear. So, but the problem again, as I told you, he said it is acquired character, so it was not easily accepted. Then came the important contribution that was from Darwin, which is called as Darwinism. So I told you already, Darwinism depends on two main theories or two main concepts. That is, one is branching descent, that all organisms are modified descents of the previous life form. If there is an ancestor, it will undergo certain change, and that change, it will be passed on to the next generation. And if there is any other change in the next generation, that will be passed on to the next generation. So this is what we mean by natural descent. Whereas in one more, natural selection, I told you, branching with descent, and natural selection, I told you that the organisms which is best suitable for the environment, they will survive. Otherwise, the population will be wiped out. The best suitable will survive and produce more progeny, and then that population is going to keep on increasing. Now, what were the main facts about Darwinism? He said heritable minor variations occurs, and these variations are passed on from generation to generation, which was not told by Lamarck. Lamarck said it is acquired character, whereas Darwin said it is inheritable character. All organisms overproduce, but the resources are always limited. These two ideas, Darwin got it from an economist called as Malthus. Malthus' essay on population greatly influenced Darwin to put forward this. ನಮ್ಮ ಪ್ರಪಂಚದಲ್ಲಿ ಎಲ್ಲೂ ಕೂಡ ರಿಸೋರ್ಸಸ್ಗಳು ಅನ್ಲಿಮಿಟೆಡ್ ಆಗಿರಲ್ಲ. ಅದು ಗಾಳಿ ಇರ್ಲಿ, ಬೆಳಕಿರ್ಲಿ, ಸಾಯಿಲ್ ಇರ್ಲಿ, ಲ್ಯಾಂಡ್ ಇರ್ಲಿ, ದುಡ್ಡಿರ್ಲಿ, ಚಿನ್ನ ಇರ್ಲಿ, ಕೆಲಸ ಇರ್ಲಿ, ಕಾಲೇಜ್ ಸೀಟ್ಗಳಿರ್ಲಿ, ನಥಿಂಗ್ ಇಸ್ unlimited. But organisms are all evolved in such a way that they are going to produce more offspring. So obviously, since the resources are limited and organisms are continuously reproducing, it will lead to what is called as struggle for existence. And during this struggle for existence, if these minor variations produces any favorable characters, then that will survive, that will be fit, and it will be producing more offsprings, and that leads to natural selection. This is what you have studied according to Darwin's idea. And then you have studied about mechanism of evolution. Now again, understand carefully that one of the biggest disadvantage of Darwin's is, Darwin said about variation, but the problem is Darwin did not explain how variation takes place. Darwin ignored about origin of variation, and also it ignored and it did not explain about mechanism of speciation. One of the tragedy of biological science is that both Darwin and Mendel, they were contemporaries. ಒಂದೇ ಸಮಯದಲ್ಲಿ ಇದ್ದವರು, ಇಬ್ರು ಯುರೋಪಿಯನ್ ಕಾಂಟಿನೆಂಟ್ ಅಲ್ಲೇ ಇದ್ದವರು. ಒಂದ್ ಕಡೆ ಮೆಂಡಲ್, who is father of genetics, was explaining about how characters are passed on. On the other hand, Darwin was explaining about origin of species. And Darwin was talking about variation and how they are passed on from generation to generation. But unfortunately, as I told you, it is a tragedy of biological science that these two did not came together and they did not explain, they did not exchange their idea. And then Darwin ignored it somehow. But later on, it was mutation theory which shed light on the origin of variation. Variation ಬರೋದಕ್ಕೆ ಕಾರಣ ನಮಗೆ ಬಂದಿದ್ದು ಗೊತ್ತಾಗಿದ್ದು ಯಾವಾಗಿಂದ ಅಂತ ಹೇಳಿದ್ರೆ mutation theory ಉಟ್ಕೊಂಡಾಗಿಲ್ಲ. Mutation theory was proposed by a scientist called as Hugo de Veris. I hope you remember this name. He is also one of the scientists who rediscovered the Mendelian genetics. And Darwin ಸಿದ್ಧಾಂತಕ್ಕೂ Hugo de Veris ಅವರ ಸಿದ್ಧಾಂತಕ್ಕೂ there are certain changes, wherein Darwin says that variations are very, very slow. ತುಂಬಾ ಸ್ಲೋ ಆಗಿ ವೇರಿಯೇಷನ್ ಆಗುತ್ತೆ ಅಂತ ಡಾರ್ವಿನ್ ಹೇಳಿದ್ರೆ, whereas Hugo de Veris said no, variations are not slow, but it happens in single large step variations. And those single large step mutations, he called it as saltation. Important term. I hope you will remember that saltation is a single large mutation which will lead to formation of different species according to de Veris. So this is the key differences between Darwin's theory and mutations theory. Evolution occurs because of some small changes, whereas mutation theory says it is because of large changes caused by mutation. Darwin's variations are small, slow, and directional, whereas Hugo de Veris mutations theory, it says mutations are random, sudden, and directionless. Evolution is gradual according to Darwin, but according to de Veris, it is saltation, which is a single step large mutation. So this is again important concept. I hope you people are going to concentrate for another 5-10 minutes. Why it is important is because questions come from this both in genetics as well as evolution. So Hardy Weinberg's principle was explained by Hardy. G.H. Hardy was a mathematician and W. Weinberg was a physician. So they used algebraic equation to explain the genetic equilibrium within a population. And they said, I'm sure you know what is allele by now. Alternative forms of a gene is called as alleles. Alleles in a population, it says the allele frequency in a population are stable and constant from generation to generation. So which means in a gene pool, that is the total genes and their allele in a population will remain constant. If it do not remain constant ಆಗಿ ಇಲ್ಲ ಅಂತ ಹೇಳಿದ್ರೆ ಅರ್ಥ, it means it is undergoing some kind of evolution ಅಂತ. For example, you have studied about dominant allele and recessive alleles. So for this dominant alleles and recessive alleles, Hardy and Weinberg use the letters P and Q, where P represents the dominant allele and Q represents the recessive allele. Then P square, it will be like capital T, which means P squared means it is homozygous dominant. I hope you people can understand. If dominant allele, either capital T or capital R, capital Y, whichever the allele is, the dominant allele is represented by P, and P squared represents the homozygous dominant condition, which is capital T, or capital R, or W plus. Whereas Q square on the other hand, it represents the homozygous recessive condition, which means this allele can be represented as small t, homozygous recessive. Then can anyone quickly tell me what does PQ represents? ಯಾಕಂದ್ರೆ ಮೊನೋಟನಸ್ ಆಗಿ ಬರೀ ನಾನೇ ಹೇಳ್ತಾ ಇದ್ದೀನಿ. ಪ್ಲೀಸ್ ಟೆಲ್ ಮಿ ದಿಸ್. What does PQ means? If P square means homozygous dominant and Q square means homozygous recessive, what does PQ means? Yeah, very good, Sneha. It is not. You don't have to tell it as heterozygous dominant. Very heterozygous andre saku. It means it is having one dominant allele. So PQ, it represents what is called as heterozygous condition. So this is what they used for explaining the gene pool in a population. So hagadre sum total of allelic frequencies is equal to 1 anta karitare. Adartha P plus Q will be equal to 1. Adartha number of homozygous or frequency of distribution of dominant allele plus frequency of distribution of recessive allele will be equal to 1. So the frequency of homozygous dominant, that is nothing but P squared, and frequency of homozygous recessive, that is nothing but Q squared, and frequency of heterozygous, that is 2PQ, which means if you express it in the binomial expansion of P plus Q whole square, it will be P square plus Q square plus 2PQ, where P square represents homozygous dominant, Q square represents homozygous recessive, and 2PQ it will represents the heterozygous condition in the whole population. Now, based on this, they may ask a question like this. So, question na itara eston sari kelidare. So, gamna itti keliskoli. So, they may give you frequency of a dominant allele, frequency of recessive allele, and heterozygous condition. Homozygous dominant tu kotirtare ankolona, heterozygous tu kotirtare ankolona, homozygous recessive tu kotirtare ankolona. Illi capital A frequency ಎಷ್ಟಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ 0.45 ಇದೆ. According to Mendel's first law, alleles which are present together, they are going to separate from each other during the gamete formation. ಅದರ ಅರ್ಥ ಒಂದು ಡಾಮಿನೆಂಟ್ allele ಒಂದು ಗ್ಯಾಮೆಟಿಗೆ ಬಂದ್ರೆ ಇನ್ನೊಂದು ಡಾಮಿನೆಂಟ್ allele ಇನ್ನೊಂದಕ್ಕೆ ಹೋಗುತ್ತೆ. ಬಟ್ ಈ ಬಟರ್ಫ್ಲೈ ಇಂದ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವ ಎಲ್ಲಾ ಅಲೀಲ್ಗಳು it will be having, all of them will be having capital A in them. Then the frequency of this allele that occurs in a population, it will be 0.45. ಯಾಕಂದ್ರೆ ಇದರಲ್ಲಿ ಏನಿದೆಯೋ ಎಲ್ಲಾದು ಕ್ಯಾಪಿಟಲ್ ಏನೇ ಬರುತ್ತೆ. That is dominant allele. Whereas heterozygous condition ನೋಡಿ, if you say 0.20 and dominant allele and recessive allele separates during the formation of gamete according to the law of segregation, it means 0.1 frequency of this, it will be dominant allele, and 0.1 frequency of this, it will be recessive allele. ಈ dominant allele ಇಲ್ಲಿ ಎಷ್ಟಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ 0.5, ಇದರಿಂದ ಬಂದಿರೋದು 0.5, ಇದರಿಂದ dominant allele ಮಾತ್ರ ತಗೊಂಡ್ರೆ it will be 0.1, recessive allele ಇಲ್ಲಿ ಬಂದಿದೆ, which means ಒಂದು ಪಾಪ್ಯುಲೇಷನ್ ನಲ್ಲಿ ಡಾಮಿನೆಂಟ್ allele ಎಷ್ಟಾಯ್ತು ಹಾಗಾದ್ರೆ ಅಂತ ಹೇಳಿದ್ರೆ it will be 0.55. So A, it will be 0.55, which also means that P will be 0.55 because dominant allele ಅನ್ನ P ಅಂತ ಕರೀತೀವಿ. ಇನ್ನೊಂದು ಇಲ್ಲಿರೋದು small a, which means all the alleles, all the gametes, it will be having 0.35 in them. Plus idond 10, idu recessive allele, eradu ಸೇರ್ಕೊಂಡ್ರೆ it will be 0.45, which means recessive allele it will be 0.45, which means Q will be 0.45. Now just put it into P plus Q whole square, which means 0.55 plus 0.45 whole square. I hope you people are understanding why. P is nothing but dominant allele. P is 0.55 ಇದೆ frequency appearance, whereas recessive allele is 0.45. So ಹಾಗಾದ್ರೆ P square plus 2PQ plus Q square will be P square ಎಷ್ಟಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ it will be 0.55 square 0.55 square plus 2PQ it will be nothing but 0.2475 plus Q square will be 0.45 square, which means this will come to 0.3025 plus 0.2475 plus this will come to 0.2025. So if you add all of them, it will come to 1, which means the distribution of these frequency in a population will be always equal to 1. This is also important. ನಿಮಗೆ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ ಈ ತರ ಕ್ವಶನ್ಸ್ ಕೇಳ್ತಾರೆ. They may give that in a population the P was found to be 0.4. What will be the frequency of homozygous recessive ಅಂತ ಕರೀತಾರೆ. P 0.4 ಇತ್ತು ಅಂತ ಹೇಳಿದ್ರೆ Q should be 0.6. Q ಆಗಾದ್ರೆ ರಿಸೆಸಿವ್. Homozygous recessive ಎಷ್ಟಾಗುತ್ತೆ ಹಾಗಾದ್ರೆ? Homozygous recessive will be Q square, which means 0.6 square, which means it will be 0.36. So frequency of homozygous recessive to occur in that population will be 0.36. ಅದೇ ತರ ಇದನ್ನೇ ಕೊಟ್ಟಿರ್ತಾರೆ ಅನ್ಕೊಳ್ಳಿ, like they may give you the P dominant allele frequency 0.4 ಇದೆ. What is the frequency of homozygous dominant allele ಅಂತ ಹೇಳಿದ್ರೆ just homozygous dominant ಅಂದ್ರೆ P square ಆಗುತ್ತೆ, which means 0.04, which will be 0.16. So I hope you people are understanding. ಯಾವ್ದಾದ್ರೂ ಒಂದು ಕಂಡೀಷನ್ ಅನ್ನ ಕೊಟ್ಟು ಇನ್ನೊಂದು ಕಂಡೀಷನ್ ಯಾವ ತರ ಕಂಡುಹಿಡಿಯೋದು ಅಂತ ಕೇಳ್ತಾರೆ. ಬಟ್ ಈ ಎಲ್ಲಾ ಅಸಂಪ್ಷನ್ಸ್ ಗಳು ನಿಜ ಆಗ್ಬೇಕು ಅಂದ್ರೆ P plus Q whole square 1 ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ if it has to be 1, which means if the constant or if the population it has to be stable, ಹಾಗಾದ್ರೆ ಈ ಕಂಡೀಷನ್ ಇರಬೇಕು. ಏನ್ ಕಂಡೀಷನ್ ಅಂತ ಹೇಳಿದ್ರೆ there should be no gene migration. ಜೀನ್ ಮೈಗ್ರೇಷನ್ ಆಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ಹೊಸ ಜೀನ್ ಬರುತ್ತೆ ಪಾಪ್ಯುಲೇಷನ್ಗೆ, ಹಾಗಾದ್ರೆ ಎಕ್ಸಿಸ್ಟಿಂಗ್ ಜೀನ್ ಏನಿದೆಯೋ ಅದು ಡಿಸ್ಟರ್ಬ್ ಆಗುತ್ತೆ, ಅವಾಗ ಈ ವ್ಯಾಲ್ಯೂ ಚೇಂಜ್ ಆಗುತ್ತೆ. ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಅಂತ ಹೇಳಿದ್ರೆ ಸಡನ್ ಚೇಂಜ್ ಆಗೋದು, ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಪಾಪ್ಯುಲೇಷನ್ ಅಲ್ಲಿ ಸಡನ್ ಚೇಂಜ್ ಆಗೋದನ್ನ ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಆದ್ರೂ ಇಟ್ ವಿಲ್ ಲೀಡ್ ಟು ಚೇಂಜ್ ಇನ್ ದ ಫ್ರೀಕ್ವೆನ್ಸಿ, ಹಾಗಾದ್ರೆ ಒನ್ ವ್ಯಾಲ್ಯೂ ಕಳ್ಕೊಳ್ಳುತ್ತೆ, ಅದರ ಅರ್ಥ ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಆಗಬಾರ್ದು. ಮ್ಯೂಟೇಶನ್ ಅಂತ ಗೊತ್ತಿದೆ ನಿಮ್ಗೆ, ಇಟ್ ಇಸ್ ಅ ಚೇಂಜ್ ಇನ್ ದ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್. ಎಕ್ಸಿಸ್ಟಿಂಗ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಚೇಂಜ್ ಆಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿನೂ ಚೇಂಜ್ ಆಗುತ್ತೆ, ಹಾಗಾದ್ರೆ ಪಾಪ್ಯುಲೇಷನ್ ಅಲ್ಲಿ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಸ್ಟೇಬಲ್ ಆಗಿರ್ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಮ್ಯೂಟೇಶನ್ ಆಗಬಾರ್ದು. ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್ ಆದ್ರೂ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಚೇಂಜ್ ಆಗುತ್ತೆ, ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್ ಆಗಬಾರ್ದು. ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಆದ್ರೂ ನಿಮಗೆ ಜ್ಞಾಪಕ ಇರ್ಬೋದು, ಬ್ರೌನ್ ಕಲರ್ ಇಲಿಗಳು ಮತ್ತೆ ಪಿಂಕ್ ಕಲರ್ ಇಲಿಗಳು ನಾವು ಮೊದಲು ನೋಡಿದಾಗ, ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಆದಾಗ ಬ್ರೌನ್ ಕಲರ್ ಇಲಿಗಳು ಜಾಸ್ತಿ ಆಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ಅವುಗಳ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಪಿಂಕ್ ಗಳ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಕಡಿಮೆ ಆಗುತ್ತೆ. ಅದರ ಅರ್ಥ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಆದ್ರೂ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಹಾಳಾಗುತ್ತೆ. ಸೊ ಪ್ಲೀಸ್ ಕೀಪ್ ಆಲ್ ದೀಸ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಇನ್ ಮೈಂಡ್. Gene migration, genetic drift, mutation, genetic recombination, natural selection. These are the factors which affects the Hardy-Weinberg's equilibrium. You have studied the last thing about the basic of evolution like this. The graph in textbook allu ide, but slightly different type. ಬರ್ದಿರೋದು ಬೇರೆ ತರ ಬರ್ದಿದ್ದಾರೆ ಅಷ್ಟೇ. ಇದು ನಮ್ದು ಒರಿಜಿನಲ್ ಪಾಪ್ಯುಲೇಷನ್ ಈ ತರ ಇದೆ. ಈ ತರ ಅಂದ್ರೆ ಮೇಲ್ಗಡೆ 3 ಗ್ರಾಫ್ ಏನಿದೆಯೋ, ದೀಸ್ 3 graphs represents the original distribution of the gene frequency. ಇಲ್ಲಿ ನೋಡಿ, ಬಾಡಿ ಸೈಜ್ 25, 50, 75, 100, 125, which means more number of organisms are in the body size between around 55 to 65 ಅಂತ ಅನ್ಕೋಣ, ಸಾರಿ 55 to 85 or 90 ಅಂತ ಅನ್ಕೋಣ. The selection of for small and large individuals, ಅದರ ಅರ್ಥ Because of some environmental change, ಯಾವ ಬಾಡಿ ಸೆಲೆಕ್ಷನ್ ಆಗಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ either small body is favored or large body is favored, but medium size body ಇದೆಯಲ್ಲ ಅದು ಬೇಕಾಗಿಲ್ಲ ಆ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ಗೆ. ಅವಾಗ ಸೆಲೆಕ್ಷನ್ ಆದಾಗ ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿಕಮ್ ಲೈಕ್ ದಿಸ್, ಸ್ಮಾಲ್ ಬಾಡಿ ಇಲ್ಲಿರೋದು, ಇನ್ನೊಂದು ಲಾರ್ಜ್ ಬಾಡಿಡ್ ಇಂಡಿವಿಜುವಲ್ ಸೆಲೆಕ್ಟ್ ಆಗುತ್ತೆ, ಬಟ್ ಮೀಡಿಯಂ ಸೈಜ್ ಬಾಡಿಗಳಿರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ಗಳು ಅಲ್ಲಿಂದ ಎಕ್ಸ್ಟಿಂಕ್ಷನ್ ಆಗುತ್ತೆ. This kind of selection, I hope you remember, this is called as disruptive selection anta kottidani book alli. In the sense, according to your book, the peripheral characters are selected, whereas the median character is not selected. Median character ಗಳು select ಆಗಲ್ಲ, but peripheral character ಗಳು select ಆಗುತ್ತೆ. Then it is called as disruptive selection. But because of some environmental change, natural selection might have selected only the large individuals. ಹಾಗಾದ್ರೆ ಬಾಡಿ ಸೈಜ್ ಜಾಸ್ತಿ ಇರೋದನ್ನ ಮಾತ್ರ ಸೆಲೆಕ್ಟ್ ಆಗಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿಕಮ್ ಒಂದೇ ಒಂದು ಪೀಕ್ ಶಿಫ್ಟ್ ಆಗುತ್ತೆ ಒಂದ್ ಕಡೆಗೆ. ನೋಡೋಣ ಹೇಳಿ ಏನಂತ ಕರಿತೀರಾ ಇದನ್ನ? ವಾಟ್ ಯು ಕಾಲ್ ದಿಸ್ ಟೈಪ್ ಆಫ್ ಸೆಲೆಕ್ಷನ್ ಆಸ್? ಆನ್ಸರ್ ವೆಯ್ಟ್ ಮಾಡ್ತಿರ್ತೀನಿ. ಮೀನ್ ವೈಲ್ ಇಲ್ಲಿ ಮೀಡಿಯಂ ಸೈಜ್ ಅಂದ್ರೆ ಮಿಡ್ ಸೈಜ್ ಇಂಡಿವಿಜುವಲ್ಸ್ ಗಳು ಜಾಸ್ತಿ ಸೆಲೆಕ್ಟ್ ಆಗಿದೆ, ಪೆರಿಫೆರಲ್ ಇರೋದು ಸೆಲೆಕ್ಟ್ ಆಗಿಲ್ಲ. ವಾಟ್ ಯು ಕಾಲ್ ದ ಸೆಕೆಂಡ್ ಒನ್ ಆಸ್ ಅಂಡ್ ವಾಟ್ ಯು ಕಾಲ್ ಥರ್ಡ್ ಒನ್ ಆಸ್? ಕ್ವಿಕ್ ಆನ್ಸರ್ಸ್. These graphs are given in the textbook in the last part. The peak shifts either to left side or right side, what do you call it as? Or if the peak gets narrower, what do you call it as? Okay, I got one answer, 3. What do you mean by 3? Okay, ಇನ್ನೊಂದು ಆನ್ಸರ್ ಬಂದಿದೆ. Let us check that and then proceed. Very good, very good, ಸ್ನೇಹ. ಎರಡನೇದಲ್ಲಿರೋದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಡೈರೆಕ್ಷನಲ್ ಸೆಲೆಕ್ಷನ್ ಅಂತಾರೆ, ಇನ್ನೊಂದನ್ನ ಸ್ಟೆಬಿಲೈಸಿಂಗ್ ಸೆಲೆಕ್ಷನ್ ಅಂತ ಕರೀತಾರೆ. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಡೈರೆಕ್ಷನಲ್ ಸೆಲೆಕ್ಷನ್. ಇಟ್ ಮೇ ಶಿಫ್ಟ್ ಟುವರ್ಡ್ಸ್ ರೈಟ್ ಆರ್ ಇಟ್ ಮೇ ಶಿಫ್ಟ್ ಟುವರ್ಡ್ಸ್ ಲೆಫ್ಟ್. ಪೀಕ್ ಗೆಟ್ಸ್ ನ್ಯಾರೋರ್ ಏನಿದೆಯೋ ಅದನ್ನ ಸ್ಟೆಬಿಲೈಸಿಂಗ್ ಸೆಲೆಕ್ಷನ್ ಅಂತ ಕರೀತಾರೆ. ವೆರಿ ಗುಡ್. ನೆಕ್ಸ್ಟ್ ನೀವು ಓದಿರೋದು ಲಾಸ್ಟ್ ಪಾರ್ಟ್, ದಟ್ ಇಸ್ ಅಕೌಂಟ್ ಆಫ್ ಎವಲ್ಯೂಷನ್ ಅಂತ. ಇಟ್ ಜಸ್ಟ್ ಶೋಸ್ ಯಾವ್ಯಾವ ಎರಾದಲ್ಲಿ ಏನೇನ್ ಚೇಂಜಸ್ ಆಯ್ತು ಅಂತ. Just try to remember all this fact that 500 million years ago invertebrates evolved. Around 400 to 600 million years ago, first land organisms that are plants evolved. 400 million years ago, arthropods invaded the land. 550 million years ago, jawless fishes, which had a stout and strong fins, they were able to crawl out of the water and come to land and go back. These jawless fishes were formed around 350 million years ago. 320 million years ago, seaweeds and few plants evolved. Then came amphibians and reptiles. In the next 200 million years ago, the whole earth was dominated by reptiles. That is where dinosaurs were ruling the whole earth. And then came giant ferns such as pteridophytes, and all of them died for some mass extinction, and they started converting it into fossil fuels. See, ಈ ಗ್ರಾಫ್ ಅನ್ನ ಯಾವ ತರ ಅರ್ಥ ಮಾಡ್ಕೊಬೇಕು ಅಂತ ಹೇಳ್ತೀನಿ. I hope you can memorize all this. See, you know, these are called as different eras, and these are different periods. Now, there was a chlorophytic ancestor in the Carboniferous period. In the Carboniferous period, gradually bryophytes started evolving. Same ಅದೇ ತರ evolve ಆಗ್ತಾ ಬಂದಿದೆ. ಅದರ ಅರ್ಥ bryophytes ಗಳು ಫಸ್ಟ್ evolve ಆಗಿದ್ದು ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ in the Carboniferous period or in Paleozoic era. ಅದಾದ್ಮೇಲೆ ಟ್ರೇಕಿಯೋಫೈಟ್ಸ್ ಗಳ ಆನ್ಸಿಸ್ಟರ್ ಇಂದ ಇನ್ನೊಂದು ಹೊಸ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಗಳು ಎವಾಲ್ವ್ ಆಗಕ್ಕೆ ಶುರು ಆದವು. ಅದರಲ್ಲಿ arborescent lycopods evolved somewhere during Devonian. ಆ ಪೀರಿಯಡ್ ನಲ್ಲಿ ಎವಾಲ್ವ್ ಆದ್ರು. ಬಟ್ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ನೀವು ಅರ್ಥ ಮಾಡ್ಕೊಬೇಕಾಗಿರೋದು, they got extinct by the end of Permian period, and rest of them evolved into lycopods. And then Coelopian ancestors in the Devonian period ನಿಂದ ಹಿಡಿದು Permian period ತನಕ horse-tails ಗಳು evolve ಆಗಿದ್ದು, and then their species diversity went on reducing from Triassic period. Triassic period ಇಂದ ಅದು ಕಡಿಮೆ ಆಯ್ತು. So I hope you can understand rest of them, which means ginkgo started evolving somewhere during Permian period. Cycads, which belongs to gymnosperms, they started evolving the same period, that is Permian period. But angiosperms that we see today, they only evolved in the Cretaceous period. So itara ಈ ಗ್ರಾಫ್ ಅನ್ನ ಅರ್ಥ ಮಾಡ್ಕೊಂಡು ಜ್ಞಾಪಕ ಇಟ್ಕೊಬೇಕು. ಇಟ್ ಇಸ್ ಜಸ್ಟ್ ಅಬೌಟ್ ಮೆಮೊರೈಸಿಂಗ್. This is about evolution of different vertebrates. So dinosaurs got extinct somewhere between Cretaceous and Tertiary period. Idella again, just you have to read the graph and memorize it. Pterapsids, they got extinct during Triassic period. Thecodons, they got extinct during Permian period. Phyloceras, they got extinct, or rather, they got extinct in Carboniferous period. ಈ ತರ graph ಅನ್ನ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. And the last concept that you have studied is about human evolution. You know, human evolution can be traced back to this ancestors of ours, which is called as Dryopithecus. Just try to remember these things, that it was there on the planet Earth about 10,000 million years ago. And then from that, what evolved was Ramapithecus. I hope you know Ramapithecus fossils are found in India. And that evolved around 15 million years ago. Then Australopithecus evolved. Please remember, Australopithecus fossils are not found in Australia, but they are found in Africa. And this evolved around 2 million years ago. And what is important here is to remember the brain capacity. Brain capacity of Australopithecus was about 450 cubic centimeter. And it was full erect posture while walking. Mainly it was herbaceous. It used stone weapons and lived in the trees, had canine teeth as well as incisors. Then came the first human-like organism that was called as Homo habilis. And Homo habilis evolved around 1.5 million years ago. Brain capacity was 735 cubic centimeter, fully erect posture, carnivorous. Then canine were there. And these were the first organism or the first species to started evolving tools. Then came Homo erectus 1.5 million years ago. It had a brain capacity of 800 to 1,100 cubic centimeter. Even this also had fully erect posture. Food was omnivorous. They also started using stones and bones for hunting. Then evolved Homo neanderthals as well as Homo sapiens. Homo erectus indane Homo neanderthals ಮತ್ತು Homo sapiens ಗಳು evolve ಆಗಿತ್ತು. It is assumed that they evolved around 40,000 years to 1 lakh years ago. Brain capacity is 1,300 to 1,600 cubic centimeter. It had a fully posture body. It had an omnivorous food. Cave dwellers, they used to hide their bodies using animals' leathers. They buried their deads. And then came us. We are called as Homo sapiens, 25,000 million years ago. Sorry, not 25,000 million years ago. It was 25,000 years ago, not million years ago. Brain capacity is 1,200 to 1,600 centimeters square. And you know about us. Now let us start looking into multiple choice questions for another at least more than 30 minutes. First question. The universe originated how many years ago? Remember, it is not Earth. Question is not about Earth. Can I get an answer quick? Answer ಹೇಳ್ಬೇಕು ಇಲ್ಲ ಅಂದ್ರೆ it is of no use. Just listening to synopsis, it will be of no use. Please try to be interactive, at least in multiple choice questions. Good. So universe originated around 20 billion years ago. That was when the Big Bang took place. But Earth was formed somewhere around 4.5 billion years ago. So answer should be third one. ಮತ್ತೊಂದು ಸರಿ ಹೇಳ್ತಾ ಇದ್ದೀನಿ, I request all of you to actively participate in this question and answers. ನೀವು ಆನ್ಸರ್ ಮಾಡಲಿಲ್ಲ ಅಂದ್ರೆ ನಿಮ್ಗೆ ಅಕ್ಯುರಸಿ ಸ್ಪೀಡ್ ಎರಡು ಬೆಳೆಯಲ್ಲ. ಅಕ್ಯುರಸಿ ಸ್ಪೀಡ್ ಎರಡು ಬೆಳಿಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ you should answer it. So keep trying to answer it, either right or wrong, answer it. Next question. The process of evolution of different species in geographical areas, starting from a point and literally radiating to other areas of geography or habitat, is called as. Very good. I can see 6 quick answers and all of them are telling B or C. Correct. ಇದನ್ನ adaptive radiation ಅಂತ ಕರೀತಾರೆ. What do you mean by convergent evolution is when two different organisms start evolving into a similar structure because they are sharing this common habit. It is called as convergent evolution. So answer for this, it should be third one. Wonderful. Very good. Next one. Good. Asha and Sneha, ibru matra answer helidare, ಉಳಿದವರು. Pavitra says it is C. Akash A. Yashwant C. Placental wolf and Tasmanian wolf. Illide illi. This is placental wolf. And this is Tasmanian wolf. ಎರಡು ಒಂದೇ ಕಡೆ evolve ಆಗಿದೆ. ಅದರಿಂದ ಒಂದೇ ತರದ ಬಾಡಿ ಫಂಕ್ಷನ್ಸ್ ಗಳಿರುತ್ತೆ. ಅದರ ಅರ್ಥ you can see what is called as analogy or analogous organs ಗಳು ಕಾಣುತ್ತೆ ಹೊರತು homologous organs ಗಳು ಕಾಣಲ್ಲ. You do not see homologous organs. Homologous organs ಕಾಣಿಸೋದು ಯಾವಾಗ? ಒಂದು ಕಾಮನ್ ಆನ್ಸಿಸ್ಟರ್ ಇದ್ದು ಅದರಿಂದ ಬೇರೆ ಬೇರೆ ಆರ್ಗಾನಿಸಮ್ಸ್ ಗಳು ಎವಾಲ್ವ್ ಆದ್ರೆ then you call it as homologous, and that shows what is called as divergent evolution. ಬಟ್ ಇಲ್ಲಿ ಆಗ್ತಾ ಇರೋದಕ್ಕೆ ನಿಮ್ಗೆ ಎರಡು ಆನ್ಸರ್ ಕೊಡ್ಬೋದು. ನೀವು ಇದನ್ನ ಎರಡು ಆನ್ಸರ್ ಕೊಡ್ಬೋದು. ಒಂದು ಕನ್ವರ್ಜೆಂಟ್ ಎವಲ್ಯೂಷನ್ ಆದ್ರೂ ಕೊಡ್ಬೋದು, ಅಥವಾ ಯು ಕ್ಯಾನ್ ಸೇ ದಟ್ ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಶೋ ಅನಲಾಗಸ್ ಆರ್ಗನ್ಸ್ ತೋರಿಸುತ್ತೆ ಅಂತನಾದ್ರೂ ಕೊಡ್ಬೋದು. ಅದು ಎರಡೂ ಬೇಟೆ ಆಡುತ್ತೆ, ಎರಡಕ್ಕೂ ಬಾಲ ಉದ್ದ ಇರುತ್ತೆ, ಎರಡರಿಗೂ ಸ್ಟ್ರಾಂಗ್ ಆಗಿರೋ ಕಾಲಿರುತ್ತೆ. They both chase their prey for a long period or for long distance and then hunt it down. So ಏನ್ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡುತ್ತೆ ಇದು ಅಂತ ಹೇಳಿದ್ರೆ it explains what is called as convergent evolution. Okay, ಯಾರು ಸಿ ಅಂತ ಆನ್ಸರ್ ಕೊಟ್ಟಿದ್ರು, ಐ ಹೋಪ್ ಯು ವಿಲ್ ಅಗ್ರಿ ದಟ್ ಇದರಲ್ಲಿ ಹೋಮೋಲಾಗಸ್ ತೋರ್ಸಲ್ಲ, ಇದ್ರಲ್ಲಿ ತೋರಿಸೋದು ಅನಲಾಗಸ್ ಅಥವಾ ಕನ್ವರ್ಜೆಂಟ್ ಎವಲ್ಯೂಷನ್. Next. Oh, this is varied answer. ಕೆಲವೊಬ್ರು ಸಿ ಹೇಳ್ತೀರಾ, ಇನ್ನೊಬ್ರು ಎ ಹೇಳ್ತಾ ಇದ್ದೀರಾ, ಇನ್ನೊಬ್ರು 2 ಹೇಳ್ತಿದ್ದೀರಾ, which means ಎ ಬಿ ಸಿ ಡಿ ಎಲ್ಲ ಬಂತು. ನೋಡೋಣ. What is the main essence of Darwinian theory? Darwinian theory ಹೇಳಿದ್ದು ಎರಡು ಏನೇನು, ಒಂದು ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್, ಇನ್ನೊಂದು ಬ್ರಾಂಚಿಂಗ್ ವಿತ್ ಡಿಸೆಂಟ್. ಜೆನೆಟಿಕ್ ವೇರಿಯೇಷನ್ ಅನ್ನ ಡಾರ್ವಿನ್ ಅವರು ಹೇಳಿಲ್ಲ. ಈ ಕುಡ್ ನಾಟ್ ಎಕ್ಸ್ಪ್ಲೈನ್ ಜೆನೆಟಿಕ್ ವೇರಿಯೇಷನ್, ಈ ಸೆಟ್ ಜಸ್ಟ್ ವೇರಿಯೇಷನ್. ಆ ವೇರಿಯೇಷನ್ ಇಂದಾನೆ ಅವ್ರು ಏನ್ ಹೇಳ್ತಾರೆ ಅಂದ್ರೆ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಬರುತ್ತೆ. ಐಸೋಲೇಷನ್ ವಾಸ್ ನಾಟ್ ದ ಮೇನ್ ಎಸೆನ್ಸ್ ಆಫ್ ಡಾರ್ವಿನ್ಸ್ ಐಡಿಯಾ. ಸ್ಟ್ರಗಲ್ ಫಾರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್ ಇತ್ತು. ಸ್ಟ್ರಗಲ್ ಫಾರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್ ಇದ್ದಾಗ ವೇರಿಯೇಷನ್ ಆಗಿ ಅದು ಕೂಡ ಇಟ್ ವಿಲ್ ಲೀಡ್ ಟು ವಾಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಅಸ್, ದಟ್ ಆಲ್ಸೋ ಲೀಡ್ಸ್ ಟು ವಾಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಅಸ್ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. Yeah, very good. I can see almost 21 of you answering. Wonderful. Answer for this question, it should be should be fourth one. ಅಕಸ್ಮಾತ್ ಇನ್ನೊಂದು ಆಪ್ಷನ್ ಅಲ್ಲಿ ಎಲ್ಲಾದ್ರೂ ಬ್ರಾಂಚಿಂಗ್ ವಿತ್ ಡಿಸೆಂಟ್ ಅಂತ ಕೊಟ್ಟಿದ್ರೆ then the answer would have been both of them. Next one. According to Hugo de Veris is also considered as father of mutation. Please remember, Hugo de Veris, along with two other scientists, rediscovered the Mendelian genetics. ಅವ್ರ ಮಿಕ್ಕಿದ ಹೆಸರು ಜ್ಞಾಪಕ ಇರ್ಬೋದು ನಿಮ್ಗೆ, ಟೆಶ್ಮಾರ್ಕ್, ಯುಗೋ ಡಿ ವೆರಿಸ್ ಮತ್ತೆ ಕ್ಯಾರೆನ್ಸ್ ಅಂತ ಮೂರು ಜನ rediscovered it. According to Hugo de Veris, it is single step large mutation which is called as saltation. It will be directionless. Sorry, it will be directionless. It will be random, and it will be large step. But according to Darwin, it is not going to be single step. It is going to be very slow variations. It will be having certain direction, and it random iralla. Just 1 minute. Okay, next question. The first cellular form of life appeared on Earth before how many years? The same thing is given in your textbook also. I hope you can easily answer this. Answer ಹೇಳ್ತೀರಾ ಇದಕ್ಕೆ? ಭೂಮಿ ಮೇಲೆ ಫಸ್ಟ್ ಸೆಲ್ಯುಲಾರ್ ಫಾರ್ಮ್ ಆಫ್ ಲೈಫ್ ಬಂದಿದ್ದು ಎಷ್ಟು ವರ್ಷದ ಹಿಂದೆ? ವೆರಿ ಗುಡ್. ಹರ್ಷ 2000 years ಅಂತ ಹೇಳ್ತಾರೆ. ಗುಡ್. ಹರ್ಷ, ಲಾವಣ್ಯ, ಆಕಾಶ್, ಕರೆಕ್ಟ್. ಸೊ ಫಸ್ಟ್ ಸೆಲ್ಯುಲಾರ್ ಲೈಫ್ ಅಪಿಯರ್ ಆಗಿದ್ದು ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ 2000 ಮಿಲಿಯನ್ ವರ್ಷದ ಹಿಂದೆ. ದಿಸ್ ಇಸ್ ಸಮವಾಟ್ ಕನ್ಫ್ಯೂಸಿಂಗ್ ದಟ್ ಇಸ್ ಗಿವನ್ ಇನ್ ಯುವರ್ ಟೆಕ್ಸ್ಟ್ ಬುಕ್. ಯಾಕೆ ಅಂತ ಹೇಳಿದ್ರೆ ನಿಮ್ಗೆ ಸ್ಟಾರ್ಟಿಂಗ್ ನಲ್ಲಿ ನಾನು ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡ್ಬೇಕಾದ್ರೆ ನಿಮ್ಗೆ ಜ್ಞಾಪಕ ಇದ್ರೆ ಲೈಫ್ ಆನ್ ಪ್ಲಾನೆಟ್ ಅರ್ತ್ ಒರಿಜಿನೇಟೆಡ್ 500 million years after the formation of Earth ಅಂತ. ಅದರರ್ಥ 4000 million years back. ಯಾಕೆಂದ್ರೆ ಭೂಮಿ ಸೃಷ್ಟಿ ಆಗಿದ್ದು 4500 ವರ್ಷದ ಮಿಲಿಯನ್ ಇಯರ್ಸ್ ಹಿಂದೆ. 500 ವರ್ಷ ಆದ್ಮೇಲೆ ಲೈಫ್ ಫಾರ್ಮ್ ಬಂತು ಅಂತ ಹೇಳಿದ್ರೆ ಅರ್ಥ 4000 ಮಿಲಿಯನ್ ವರ್ಷದ ಹಿಂದೆ ಬರಬೇಕು. ಲೈಫ್ ಫಾರ್ಮ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಬಟ್ ಇದೇ ನಿಮ್ಮ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ನಲ್ಲೇನೆ ಫಸ್ಟ್ ಸೆಲ್ಯುಲಾರ್ ಫಾರ್ಮ್ ಆಫ್ ಲೈಫ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. So the key difference is cellular form of life and the other one is life. Cellular form of life ಬಂದಿದ್ದು ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರೆ it was 2 million years ago. Good. Next question. According to chemical evolution, the condition on primitive earth were ಯಾವ ಕಂಡೀಷನ್ ಇದ್ದಿದ್ರಿಂದ ಭೂಮಿ ಮೇಲೆ ಲೈಫ್ ಒರಿಜಿನೇಟ್ ಆಗಕ್ಕೆ ಈಸಿ ಆಯ್ತು. I have inserted the slide which I used for explaining this condition. ಏನೇನಿತ್ತು ಅಂತ ಹೇಳಿದ್ರೆ, the temperature was very high. There was high temperature. Volcanic storms ಗಳಿದ್ವು. Reducing atmosphere ಇತ್ತು. And it contained methane, ammonia, etc. Low temperature ಇರಲಿಲ್ಲ. A, B, C ಇತ್ತು. Low temperature ಇರಲಿಲ್ಲ. Oxidizing atmosphere ಇರಲಿಲ್ಲ. ಏನಕ್ಕೆ ಅಂತ ಹೇಳಿದ್ರೆ, I hope you can understand why there was no oxidizing atmosphere, because there was no free oxygen ಇರಲಿಲ್ಲ. ಬಟ್ ಇವಾಗ ನಮ್ಮ ಭೂಮಿ ಮೇಲೆ ಫ್ರೀ ಆಕ್ಸಿಜನ್ ತುಂಬಾ ಇದೆ. ಅದಕ್ಕೋಸ್ಕರ ಸೈಂಟಿಸ್ಟ್ ಅಸ್ಯೂಮ್ ಈ ಆಕ್ಸಿಡೈಸಿಂಗ್ ಅಟ್ಮಾಸ್ಫಿಯರ್ ಈಗಿದೆ ನಮ್ಮ ಅರ್ತ್ ನಲ್ಲಿ. ಬಟ್ ಕ್ವಶನ್ ಇರೋದು ಇಟ್ ಇಸ್ ಅಬೌಟ್ ಪ್ರಿಮಿಟಿವ್ ಅರ್ತ್. ಈ ಆಕ್ಸಿಡೈಸಿಂಗ್ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ನಮ್ಮಲ್ಲಿ ಈಗ ಇರೋದ್ರಿಂದನೇ ಹೊಸ ಮಾಲಿಕ್ಯೂಲ್ ಆಗಿ ಅಥವಾ ಹೊಸ ಲೈಫ್ ಆಗಿ ಒರಿಜಿನೇಟ್ ಆಗಕ್ ಸಾಧ್ಯ ಇಲ್ಲ ಅಂತ ಹೇಳ್ತಾರೆ ಸೈಂಟಿಸ್ಟ್ ಅವರು. ಬಟ್ ಇದಕ್ಕೆ ಆನ್ಸರ್ ಏನಾಗ್ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, ಇಟ್ ಶುಡ್ ಬಿ ಫೋರ್ತ್ ಒನ್. ಗುಡ್. ನಂಬರ್ ಆಫ್ ರೆಸ್ಪಾನ್ಸಸ್ ಆರ್ ಗೆಟಿಂಗ್ ರೆಡ್ಯೂಸ್ಡ್. ಸ್ವಲ್ಪ ಹೇಳ್ಬೇಕು, ಇಲ್ಲ ಅಂದ್ರೆ ನನ್ಗೂ ಇಂಟ್ರೆಸ್ಟ್ ಇರಲ್ಲ, ನಿಮ್ಗೂ ಇಂಟ್ರೆಸ್ಟ್ ಹೊರಟೋಗುತ್ತೆ. ಮೋರ್ ಇಂಪಾರ್ಟೆಂಟ್ಲಿ ಫಾರ್ ಯುವರ್ ಓನ್ ಗುಡ್, ಯು ಹ್ಯಾವ್ ಟು ಆನ್ಸರ್ ದ ಕ್ವಶನ್ಸ್. How many statements are true for the given figure? These are placental mammals found in Australia. It is correct or wrong? Idu placental mammals alla. These are marsupial organisms, not placental mammals. ನಾನು ಈಗಾಗಲೇ ಹೇಳಿದ್ದೀನಿ marsupial ಅಂದ್ರೆ ಏನು ಅಂತ. They are going to keep their young ones in their pouch. But they show divergent evolution. It is correct. They show homology also correct. Divergent evolution ಇದೆ ಅಂತ ಹೇಳಿದ್ಮೇಲೆ homology ಇರಲೇಬೇಕು. These animals are found only in Australian continent is also correct. ನೀವು ಇಲ್ಲಿ ಏನೇನ್ ಕಾಣ್ತಾ ಇದ್ದೀರೋ, all these organisms are found only in Australia and nowhere else. ಆಸ್ಟ್ರೇಲಿಯಾ ಬಿಟ್ಟು ಬೇರೆ ಯಾವ ಜಾಗದಲ್ಲೂ ಮಾರ್ಸುಪಿಯಲ್ ಅನಿಮಲ್ಸ್ ಗಳಿಲ್ಲ. ವೆರಿ ಗುಡ್. ಅಗೈನ್ ಹರ್ಷ, ಸಂಧ್ಯಾ 2 ಅಂತ ಕೊಟ್ಟಿದ್ದೀರಾ, ಬಟ್ ದ ಆನ್ಸರ್ ಶುಡ್ ಬಿ, ಇಫ್ ಯುವರ್ ಆಪ್ಷನ್ ಇಸ್ ಟ್ರೂ ದೆನ್ ಕರೆಕ್ಟ್. ದೀಸ್ 3 ಆರ್ ಕರೆಕ್ಟ್, ಸೊ ದ ಆನ್ಸರ್ ಶುಡ್ ಬಿ ಆಪ್ಷನ್ 2. ವೆರಿ ಗುಡ್. ಆನ್ಸರ್ ಮಾಡಿದ್ರೆ ತುಂಬಾ ಒಳ್ಳೇದು. ಸ್ವೀಟ್ ಪೊಟ್ಯಾಟೊ ಅಂಡ್ ಪೊಟ್ಯಾಟೊ ಅಂದ್ರೆ ಗೆಣಸು ಮತ್ತು ಗೆಡ್ಡೆ, ಏನ್ ಹೇಳ್ತೀರಾ ಅದಕ್ಕೆ? ಗೆಣಸು ಮತ್ತೆ ಆಲೂಗೆಡ್ಡೆ. These two are the examples of what? I hope you know sweet potato helps in storage of food. Food anna store ಮಾಡುತ್ತೆ. Potato also helps in storage of food. ಎರಡು ಒಂದೇ ಕೆಲಸ. But sweet potato ಏನಿದೆಯೋ, on one hand, sweet potato, sorry, just a minute. Yeah. I'm sorry. Yeah. So food store ಮಾಡೋ sweet potato ಏನಿದೆಯೋ, you know, it is a modification of root, whereas potato which also stores food, it is a modifier of shoot. ಅದರ ಅರ್ಥ structurally they are dissimilar. They are structurally dissimilar, whereas the function that they perform is similar. ಕೆಲಸ ಒಂದೇ ತರ ಮಾಡುತ್ತೆ, ಸ್ಟ್ರಕ್ಚರ್ ಬೇರೆ ತರ ಇದೆ. ಹಾಗಾದ್ರೆ ಏನಂತ ಕರಿತೀರಾ ಅದನ್ನ? Can you call it as homologous organs or heterologous organs? Structurally dissimilar ಇದ್ದು ಕೆಲಸ ಒಂದೇ ಮಾಡ್ತಾ ಇದೆ ಅನ್ನೋದಾದ್ರೆ you will call it as analogous. ಇಲ್ಲಿ ಅನಾಲೋಗಸ್ ಇಲ್ಲ, ಹೋಮೋಲಜಿ ಇದೆ. ಅನಾಲೋಗಸ್ ಇಂದ ಏನ್ ಬರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, you will find convergent evolution. So second and third tappagutte. Answer should be first one. Agreed? Very good. Wonderful. Next. Recent ancestor of modern man is, when I say modern man or modern human, it is Homo sapiens. Homo sapiens ಗಳ most nearest ಯಾರು ಅಂತ ಹೇಳಿದ್ರೆ ಕ್ರೋಮ್ಯಾಗ್ನಾನ್ ಆಗ್ಬೇಕು. ಕ್ರೋಮ್ಯಾಗ್ನಾನ್ ಆಸ್ ವೆಲ್ ಆಸ್ ನಿಯಾಂಡ್ರಿಥಾಲ್, ಇವರಿಬ್ರು ನಮ್ಮ ನಿಯರೆಸ್ಟ್ ಆನ್ಸಿಸ್ಟರ್ಸ್. ಇನ್ ಫ್ಯಾಕ್ಟ್ ಕ್ರೋಮ್ಯಾಗ್ನಾನ್ ಇಸ್ ಆಲ್ಸೋ ಕನ್ಸಿಡರ್ಡ್ ಆಸ್ ಹೋಮೋ ಸೇಪಿಯನ್ಸ್. ನಮ್ಗೆ ಇನ್ನೊಂದು ಹೆಸರನ್ನೇ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಕ್ರೋಮ್ಯಾಗ್ನಾನ್ ಅಂತ ಕರೀತಾರೆ. ಕ್ರೋಮ್ಯಾಗ್ನಾನ್ ಅನ್ನ ಇಟ್ ಇಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ಫಾಸಿಲ್ ಹ್ಯೂಮನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಬಿಕಾಸ್ this particular species or it is found only in the form of fossils. ಅದರಿಂದ ಆನ್ಸರ್ ಏನಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ it should be Cro-Magnon and Neanderthals. Doubt? Any doubt? See, peaking man anta kottidara ಕೆಲವೊಬ್ರು ಹೆಸರು ಆನ್ಸರ್ ನ. I hope you know peaking man belongs to Homo erectus. Java ape agli, peaking man agli, they belong to Homo erectus. I hope it is not confusing. Yeah, D should be answered. Next. Which of the following describes correctly the homologous structures? Organs with anatomical similarities but performing different or same function. Organs with anatomical dissimilarities but performs the same function. This second option ಏನಿದೆಯೋ ಅದನ್ನ analogous organs ಅಂತ ಕರಿಬಹುದು. Organs that have no function now but had an important function in their ancestry ಅನ್ನ it is called as vestigial organs ಅಂತ ಕರೀತಾರೆ. For example, ನಮ್ಮ ಕಿವಿ ಹಿಂದೆ ಮಸಲ್ಸ್ ಇದೆ. ನಮ್ಮ ancestors ಗಳಲ್ಲಿ ಕಿವಿ ಅಲ್ಲಾಡಿಸ್ಲಿಕ್ಕೆ it was playing an important role, but in our body it do not play an important role. ಅದನ್ನ vestigial organs ಅಂತ ಕರೀತಾರೆ. Organs appearing only in the embryonic stage and disappearing later in the adult, no. ಹಾಗಾದ್ರೆ ಆನ್ಸರ್ ಏನಾಗ್ಬೇಕು? I'm sorry. Just be online. ಒಂದೇ ಒಂದು ನಿಮಿಷ. Yeah. Here we go. So answer ಹಾಗಾದ್ರೆ ಏನಾಗ್ಬೇಕು? I can see some of you have answered as second one. Second ಆಗುತ್ತೆ answer. Yeah, Sneha, you are absolutely right. Vermiform appendix is an example for vestigial organ. ನಮ್ಮ ಕಿವಿ ಮೇಲೆ ಒಂದು there is a peak called as Darwin's peak ಅಂತ ಕರೀತಾರೆ. ಕಿವಿ ಹಿಂದೆ ಒಂದಿದೆ, ವರ್ಮಿಫಾರ್ಮ್ ಅಪೆಂಡಿಕ್ಸ್ ಇರ್ಬೋದು. ಆಮೇಲೆ ಕಾಕ್ಸಿಜಿಯಲ್ ಬೋನ್ ಅಂತ ಇರುತ್ತೆ ನೋಡಿ, ನಮ್ಮ ವರ್ಟಿಬ್ರಲ್ ಕಾಲಮ್ ನಲ್ಲಿ ಯು ಹ್ಯಾವ್ ಸೀನ್ ಅಬೌಟ್ ಸರ್ವೈಕಲ್ ಬೋನ್, ಥೊರಾಟಿಕ್ ಬೋನ್. sacral, lumbar, sacral, ಮತ್ತೆ ಕೊನೆಯಲ್ಲಿ coccygeal bones ಇರುತ್ತಲ್ಲ, those are also examples of vestigial organs. Okay. Next question. Which of the following are required to maintenance of Hardy-Weinberg's equilibrium? Hardy-Weinberg's equilibrium ಗೆ maintain ಅಂದ್ರೆ P plus Q whole square 1 ಆಗೇನೆ maintain ಮಾಡಿ ಇಟ್ಕೊಬೇಕು ಅಂದ್ರೆ ಏನೇನ್ ಇರಬೇಕು ಅಂತ. What are required for maintenance of Hardy-Weinberg's equilibrium? Presence of natural selection, no, ಇರಬಾರ್ದು. Presence of mutation, ಇರಬಾರ್ದು. Absence of genetic drift, and there should be random mating. There should be random mating. ಇಲ್ಲಿ ಏನಂತ ಕೊಟ್ಟಿದ್ದಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ, they have given it as absence of genetic drift as well as random mating is not correct. Answer should be fourth one. That is absence of genetic drift as well as there should be presence of random mating. Random mating ಇಲ್ಲ ಅಂತ ಹೇಳಿದ್ರೆ ಎಲ್ಲಾದು ಒಂದೊಂದೇ ಕಡೆ ಇರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಗಳು ಒಂದೊಂದೇ ತರ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಹೋದ್ರೆ ಅಲ್ಲೇನೆ ಜೀನ್ಸ್ ಗಳು ಬೇರೆ ಬೇರೆ ತರ ಫ್ರೀಕ್ವೆನ್ಸಿ ಬರ್ತಾ ಹೋಗುತ್ತೆ. ಅದರಿಂದ ಆನ್ಸರ್ ಶುಡ್ ಬಿ ಫೋರ್ತ್ ಒನ್. See, I will give you 1 or 2 minutes. Can you just try to answer this? Okay, ಜೊತೆಲೆ ಸಾಲ್ವ್ ಮಾಡೋಣ. ಇಲ್ಲ ನೀವು ಸಾಲ್ವ್ ಮಾಡ್ತಾ ಇದ್ರೆ ಮಾಡ್ತಾ ಇರಿ, because we just have around 12 more minutes. In a population, an autosomal recessive disorder is present in 60 individuals. Autosomal recessive disorder ಅಂತ ಹೇಳಿದ್ರೆ ಹಿಮೋಫಿಲಿಯಾ ಅಂತ ಅನ್ಕೊಳ್ಳಿ ಉದಾಹರಣೆಗೆ. Okay, hemophilia ಬೇಡ, sickle cell anemia ಅನ್ಕೊಳ್ಳಿ, ಅಥವಾ any other recessive triad. Let us call this as small p ಅಂತ ಕರೆಯೋಣ, ಬೇಡ. Let us call it as small h ಅಂತ. ಹಾಗಾದ್ರೆ it is present in 60 individuals ಅನ್ನೋದಾದ್ರೆ frequency of appearance it will be 0.6. Okay, ಇಲ್ಲಿ 6000 ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, which means 60 divided by 6000 ಎಷ್ಟಾಗುತ್ತೆ? It will be 0.06. What will be the number of homozygous normal, that is capital A, capital A? Okay, small a ನೇ ತಗೋಣ. What will be the number of homozygous normal, that is dominant offspring in the population? Now you can consider it as capital A and capital A, right? I hope you people are trying it. Try ಮಾಡಿ ಹೇಳ್ತೀರಾ answer?
Sir, it's a break time now.
Okay.
Break till 6:05. 15 minutes break.
Okay.
Students, please join back exactly at 6:05. You should not miss any important questions. You have still questions, sir, of this chapter?
Yes, sir. I will continue for another 5-10 minutes after the break for the same chapter, and then I'll start the next chapter.
Okay. Students, go on break, get refreshed for 15 minutes and log in back 1 minute before. Don't delay it. As if you are really serious, you should be coming back. Thank you. Are you there, Mr. Ravi? Dear students, what you are watching on the screen is the Anand Sagar admission test DSAT. Anyone who wish to join engineering, pharmacy, B pharmacy, or B design, write the Anand Sagar admission test. I am sharing the link in chat box. As earlier also I shared, with the same link, you can apply for the Anand Sagar admission test, which is going to be on 29th and 30th. Still time is there. You can choose for tomorrow's date or day after tomorrow, 30th. Probably you would be choosing 30th only because it's already late. If you're not applied today, we'll try to get you the slot for 30th. The DSAT exam syllabus would be of CET syllabus. We'll be having 100 marks question paper, 2 hours, 30 marks each, physics, chemistry, maths, aptitude 10 marks. There will not be any negative marking. If you reach to our threshold score, you are eligible for the admission for engineering with scholarship. The fees for direct admission is different. The fees with the student who has written DSAT and joining is, he'll be getting the scholarship fees. A good opportunity for you, dear students. You can go ahead and apply. I will put a link in the chat box. Any query regarding the Anand Sagar admission test, you can just call me or WhatsApp me. I am here to clarify your doubts. Ask your friends to join back. Dear students, under 3 to 4 minutes are there. Very important chapters in the NEET perspective. The student who are preparing for NEET should always be attending all the chapters whichever are going on for biology. If you prepare biology, 50% of your work is done. Most of the students who succeed in NEET, they do exceptionally good in biology. Dear students, try to attend. If you have any doubt, put your queries. Pawan sir, another 3-4 minutes are there. As I showed my video, Dayanand Sagar Universities, College of Nursing, College of Physiotherapy, College of Allied Health Science, Harohalli, Kanakpura Road, spread across 140 acres. It's a beautiful campus with wonderful ambience. All of you have seen the video. That is a real one. Anyone wish to visit, you can visit the campus and have wonderful, lavish the campus visit. Any queries regarding campus, Harohalli campus, you can call me or put your question in WhatsApp. I am available. It's such a beautiful campus. If you enter 140 acres campus with the international level of stadium, you will be amazed to see the campus. You are seeing the photo of the campus in the screen. That is not an artwork, that is a real one. Dear students, I have posted the link for DSAT exam in chat box. You can definitely apply for. And then just 2 minutes are there. I request students, ask your friends to join back. Each and every minute is important. My dear students, till you write the exam, whatever time you are having now, which may not come back, whatever efforts you put now, that is going to yield you the fruits. I'm not talking any philosophy, that is a reality. Work hard. I wish all of you should, anyone who is aspiring for NEET, all of you should score better marks and get your admission to MBBS in government colleges. I wish for that. But you need to put your efforts. The question, each and every question is important. As the important topics are going on, do not miss it. The next topic, which is going to be strategies for enhancement in food production. The next topic. Evolution chapters. Some 3-4 more questions will be solved later on. Sir will be taking the next topic. Pawan sir, over to you. You can start your session.
Yeah, thank you, sir.
Thank you, sir. You can start your session, sir.
Yeah. So,
But
before break,
your voice. Hello, Pawan sir.
Yes, sir.
Bit voice disturbance is there. Kindly check
Okay,
your mic.
Am I audible now, sir?
You are audible and screen is also visible.
Okay.
Thank you.
So before break, we were looking into this problem where in a population, autosomal recessive disorder is present in 60 individuals. And the size of the population is 6,000, which means what will be the percentage of this autosomal recessive is 60 divided by 6,000 into 100, it will be around 0.01. So both of them are homozygous condition. So this will be Q square. This will be Q square, which means the Q square value, it will be 0.01. So what will be the value of Q is, value of Q, it will be 0.1. So if the value of Q is 0.1, according to R.D. Weinberg's equation, we have seen P plus Q is equal to 1. So P minus 0.1, or we can see P plus 0.1 is equal to 1. So which means the value of P, it will be 1 minus 0.1, which means it will be 0.9. So once if you know what is the value of P and Q, you know value of Q is 0.1 and value of P is 0.9, which means value of recessive allele is 0.1, frequency of dominant allele is 0.9. Now we know that P square is 0.1, leave that, plus 2 into 0.1. This 0.1 is P value and Q value is 0.09 plus 0.09. Now, we know the value of this one. We know the value of this one will become how much? We have calculated. It will come to 1080. Come to 1080 less. We know already 0.09. This is not 0. This is 0.09. So value of this, it will be already given. That is 60 of them. 60 individuals are recessive disorders. Recessive value is nothing but Q square, which means 60. So heterozygous and these two is together 1080 plus 60, which will be equal to 4 1140. I hope you're understanding what are this. These are heterozygote individuals, and 60 numbers what you are seeing here, this is of already given autosomal recessive disorder, which means it is homozygous recessive. What is the question is about? Question is about finding the number of homozygous normal, that is homozygous dominant. So which means out of 6,000 individual, if 1,140 represents heterozygous and homozygous recessive, ಇದೆರಡು ಸೇರ್ಕೊಂಡು ಇಷ್ಟಾದ್ರೆ ಉಳಿದಿದ್ದು ಎಷ್ಟಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ it will be 4,860. So the answer will be first one. Yeah. So with this, let us stop the questions on evolution, and let us start with the questions on strategies for enhancement in food production. So let us look into the synopsis for next 1 hour at least, and then we can start solving the questions. Yeah, here we go. So, under this chapter called strategies for enhancement in food production, you have basically studied about what are the methods that are there to enhance the food production. For that, you have seen basic introduction that human population is continuously increasing, but the resource, including the land that we have, is very limited, and hence there is a demand for higher production of food. So for higher production of food, we will look into two things. One is animal husbandry and the other one is plant breeding technique. So any biological principles such as animal husbandry as well as plant breeding have a major role to increase the food production, which is important for feeding ever increasing population. So there are several techniques like embryo transfer technology and tissue culture technique that plays an important role in enhancing the food production. So out of that, the first one that you have studied is animal husbandry. Under animal husbandry, it usually includes animals such as cattle, pig, sheep, poultry, which can be any form of domesticated bird. It can be chicken, duck, turkey, geese. All these will be considered as poultry as well as fish, which are useful for humans in various ways, either as a food or as in working animals, in the form of milk or in the form of meat, in the form of egg. So all these together, it is referred as livestock. And maintaining of this all livestock is what is called as animal husbandry. These animals are managed by production of commercially important products such as milk, which we get from cattle, sometimes from sheep also, meat, which we get from sheep, poultry, as well as fish, wool, which we get it from sheep, egg from poultry, honey, silk, etc. Now, the third point says 70% of the world livestock population is in just India and China. But India and China put together, their contribution to the world farming is just 25%. ಅಂದ್ರೆ ನಮ್ಮಲ್ಲಿ ರಿಸೋರ್ಸಸ್ಗಳು ಜಾಸ್ತಿ ಇದೆ, ಅಂದ್ರೆ ಲೈಫ್ ಸ್ಟಾಕ್ ಜಾಸ್ತಿ ಇದೆ ಇಂಡಿಯಾ ಮತ್ತೆ ಚೈನಾ ಎರಡನ್ನು ಸೇರ್ಕೊಂಡ್ರೆ, ಬಟ್ ಔಟ್ಪುಟ್ ತುಂಬಾ ಕಡಿಮೆ ಇದೆ. ಸೊ ಅದರಿಂದನೇ we need to have new technologies as well as new strategies which needs to be applied to improve the quality as well as productivity from 25% to much more, because we are having large amount of livestock. So under that, first thing that you have studied is about dairy farm management. What do you mean by dairy farm management? As any other management, either business management or hotel management or industrial management, it basically involves the right processes and the systems which are meant for increasing the quality as well as quantity of milk production. ಸರಿಯಾದ ಪ್ರೋಸೆಸ್ ಮತ್ತು ಸಿಸ್ಟಮ್ಸ್ಗಳನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಮಿಲ್ಕ್ ಪ್ರೊಡಕ್ಷನ್ ಇನ ಕ್ವಾಲಿಟಿ ಮತ್ತು ಕ್ವಾಂಟಿಟಿನ ಜಾಸ್ತಿ ಮಾಡೋದು ಡೈರಿ ಫಾರ್ಮ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ನ ಮುಖ್ಯ ಉದ್ದೇಶ. Milk production is primarily dependent on choosing improved cattle breeds, ಒಳ್ಳೆ breed ಇರಬೇಕು, giving a proper type of feed for cattle, which we together called as fodder anta, maintenance and proper shelter facilities and regular cleaning of cattle. In other words, maintaining an hygienic condition is also important one. Now, what are the systems and process of dairy farm management? The right systems and process of dairy farm management includes choosing an improved cattle breeds. Now, cattle breed can be of two types. One is called as milch variety, which is meant for production of milk alone, whereas the other one is called as draught variety, which is meant for working in farms. And the third one is called as mixed variety. Milk ಬಂದ್ರೆ ಡ್ರಾಟ್ ಅಲ್ಲಿ ಕೆಲಸ ಮಾಡೋದಾದ್ರೆ ಮಿಕ್ಸ್ಡ್ ವೆರೈಟಿನಲ್ಲಿ ಎರಡು ಕೆಲಸಕ್ಕೆ ಬರುತ್ತೆ. ಕೆಲಸಕ್ಕೂ ಉಪಯೋಗಿಸ್ಕೊಂತಾರೆ, ಮಿಲ್ಕ್ ಅನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾರೆ. Cattle should also be given healthy, nutritious food, which will be contain right type of rugged fiber concentrates as well as high levels of proteins, because this high levels of protein is the one which is going to decide the milk yield. ಇದೆಲ್ಲದನ್ನು ಕೆಲವೊಮ್ಮೆ ಫೋಡರ್ ಅಂತ ಕೂಡ ಕರೀತಾರೆ, ಮೇವು ಅಂತ ಹೇಳ್ತೀವಿ ಕನ್ನಡದಲ್ಲಿ. Is also referred as fodder sometimes. Cattle should also be housed in a proper cattle house, also referred as shed sometimes. Should be kept in well-ventilated roofs to keep them from harsh weather conditions such as heat, cold, or rain. Other systems and process includes regular bath, which is meant for keeping in proper hygienic condition. And also time to time checkup by a veterinary doctor, who should keep on visiting the dairy farms to see whether they are well vaccinated, if there are any diseases, etc. Stringent cleanliness and hygienic condition, because milk, which is the product of dairy farm management, or any other milk products such as butter, ghee, cheese, buttermilk, etc., they are highly perishable items, which means they can easily get spoiled. So if it has to be properly maintained, then the hygienic condition should be very, very important, both of cattle as well as the person who is handling the cattle. The main thing is about milking, storage, as well as transport of milk. And for all this, there should be also regular inspection and also record keeping, so that you can understand what kind of changes are happening and what are the reasons for it. So idistunu ಜ್ಞಾಪಕ ಇಟ್ಕೊಂಡಿರಿ. These are the systems and processes of dairy farm management. Choosing an improved cattle breed, it should be provided right type of food. It should be kept in well-ventilated hygiene shed. There should be regular bathing of cattle. There should be regular visit by veterinary doctors, and there should be hygienic condition while milking, storing, and transporting. And also there should be a record keeping. These are the important systems and processes of dairy farm management. Second one is poultry farm management, and I hope you can guess, poultries includes any domesticated birds. ಯಾವುದೇ ಪಕ್ಷಿಯನ್ನ ನಾವು ಸಾಕೊಳ್ಳೋ ರೀತಿನಲ್ಲಿ ಇದ್ರೆ ಅದನ್ನ ಪೌಲ್ಟ್ರಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಅದರಲ್ಲಿ ಕೋಳಿ ಇರ್ಬೋದು, ಬಾತು ಕೋಳಿ ಇರ್ಬೋದು, ಟರ್ಕಿ, which is very much in demand during Christmas, ಅಥವಾ geese, which is very closely resembles to that of duck. So what are the components of poultry farm management? Almost similar to dairy farm management. ಏನೇನಿರುತ್ತೆ? Selection of disease free and suitable breeds. For example, ನಿಮ್ಗೆ ಲೇಯರ್ಸ್ ಅಂಡ್ ಬ್ರಾಯ್ಲರ್ಸ್ ಅಂತ ಎರಡು ಪೌಲ್ಟ್ರಿನಲ್ಲಿ ಮೇನ್ ಆಗಿ ಸಾಕ್ತಾರೆ. Where layers is meant for production of egg, whereas broilers are meant for production of meat, or it may be exotic birds. So depending upon your demand, there may be different types of breeds. There should be proper and safe farm conditions. I'm repeating, there should be proper and safe farm condition. Especially it should be having safety from predators. ಯಾವುದಾದ್ರೂ ಬೇರೆ ಪ್ರಾಣಿಗಳು ಬಂದು ಅದನ್ನು ತಿನ್ನಬಾರ್ದು. Next one is proper feed and water. I'm sure you will understand protein rich feed kottostunu koli mamsa bega jasti ಆಗ್ತಾ ಹೋಗುತ್ತೆ. And at last, hygienic and health condition. ನಮ್ಗೆ ಜವಾರಿ ಕೋಳಿ ಅಥವಾ ನಾಟಿ ಕೋಳಿ ಅಂತ ಹೇಳ್ತಾರೆ. For those, hygienic condition is not really so important. ತುಂಬಾ important ಅಲ್ಲ. Yes, important, but not so important. Whereas those breed of chicken which are housed ಅಥವಾ ಯಾವುದನ್ನ ಪೌಲ್ಟ್ರಿ ಫಾರ್ಮ್ ಅಂತ ಮಾಡಿ ಬೆಳೆಸ್ತಾರೋ ಅದು it will be having lot of sensitivity ಜಾಸ್ತಿ ಇರುತ್ತೆ ಅದಕ್ಕೆ. ಅದಕ್ಕೋಸ್ಕರ ಎಸ್ಪೆಷಲಿ ಅದರಲ್ಲಿ ಪೌಲ್ಟ್ರಿ ಫಾರ್ಮ್ ನಲ್ಲಿ ದೇ ಶುಡ್ ಬಿ ವೆರಿ ಹೈಜಿನಿಕ್ ಕಂಡೀಷನ್. ಯಾಕಂದ್ರೆ ಒಂದ್ಸರಿ ರೋಗ ಬಂತು ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಮೇ ಕಿಲ್ ಲಾಟ್ ಆಫ್ ಬರ್ಡ್ಸ್. ಒಂದೇ ಸರಿ ಕಡಿಮೆ ಆಗೋಗುತ್ತೆ. ಸಾರಿ, ಒಂದೇ ಸರಿ ಅದರದ್ದು ಈಲ್ಡ್ ಕಡಿಮೆ ಆಗೋಗುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಸ್ ನೀವು ಓದಿರೋದು ಅನಿಮಲ್ ಬ್ರೀಡಿಂಗ್. ಕ್ವಶನ್ಸ್ ಗಳು ಜಾಸ್ತಿ ಬರೋದು, ಮೋಸ್ಟ್ ಆಫ್ ದ ಕ್ವಶನ್ಸ್ ಫಾರ್ ದಿಸ್ ಚಾಪ್ಟರ್ ಕಮ್ಸ್ ಫ್ರಮ್ ಅನಿಮಲ್ ಬ್ರೀಡಿಂಗ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್ಸ್. So try to understand what are the different breeding techniques that are present. What do you mean by breed first? Breed is a group of animals which are related by descent and similar general and have similar general appearance, features, size, etc. And what do you mean by breeding? Breeding is the modification of genotype of an organism to make organism more useful for human beings. So that is what we mean by breeding. Now, what are the aims of animal breeding? To increase the yield of animals, either in the form of meat, or in the form of egg, or in the form of wool. ಇವುಗಳನ್ನ ಜಾಸ್ತಿ ಮಾಡೋದು ಅದರ ಉದ್ದೇಶ. To improve the desired quality of animals produce, to produce disease-resistant varieties of animals. These are some of the aims of animal breeding. This is important. Ilinda mundak barodu important. So please concentrate and understand this. Just 1 minute. Let me check if you have any questions. As of now, I do not see any questions from anyone. Okay. Yeah. So there are three major types of animal breeding. One is called as inbreeding. Second one is outbreeding. There are three types of outbreeding techniques. One is outcrossing, crossbreeding, and interspecific hybridization. And third one is controlled breeding experiment. In that there are two types: artificial insemination and multiple ovulation embryo transfer technology. So first, if you start with inbreeding, please understand this carefully. It is a mating of more closely related individuals within the same breed for about 4 to 6 generations. Then it is called as inbreeding. For example, let's say this is a superior male. On what basis you will consider it as superior male? Somebody which is something which is having an more production of more amount of or a strong bull, disease resistant bull, will be considered as superior male. Whereas superior female is someone which will produce more amount of milk. So if you cross them, you may get 3 different offsprings, out of which if you cross between the same breed, which are genetically related, they have got same ancestors. If you breed them together, this kind of breeding, it will be called as inbreeding. I hope you are understanding. And this may continue for another 5 to 6 generations, which means from breeding this and this, if you get one more cow, you may breed between these two. So ಈ ತರ 4-5 ಜನರೇಷನ್ ಅಥವಾ 6 ಜನರೇಷನ್ ವರೆಗೂ ಕಂಟಿನ್ಯೂ ಆಗ್ತಾ ಹೋದ್ರೆ it is called as inbreeding. If you observe this diagram carefully, you can see this is having a dominant allele and this is having an homozygous dominant allele, whereas this is heterozygous condition. So according to this diagram, A allele ಏನಿದೆಯೋ, this is recessive dangerous allele. When you do inbreeding, what happens is, because of continuous inbreeding, it leads to condition which is called as homozygous condition. When homozygous recessive condition comes together, ಅವಾಗ ನಿಮ್ಗೆ ಆ ಕಾಯಿಲೆನಲ್ಲಿ ಅಥವಾ ಆ ಪಾಪ್ಯುಲೇಷನ್ ಆಫ್ ಕೌ ನಲ್ಲಿ ಏನಾದ್ರೂ ಡೇಂಜರಸ್ ಡಿಸೀಸ್ ಇದ್ರೆ ನಮ್ಗೆ ಗೊತ್ತಾಗುತ್ತೆ. ಇಲ್ಲ ಅಂದ್ರೆ ಗೊತ್ತಾಗಲ್ಲ. ಈಗ ಉದಾಹರಣೆಗೆ ದಿಸ್ ಇಸ್ ಕ್ಯಾರಿಯರ್. We don't know whether this is affected or not. But if you subject this for continuous inbreeding, in some or the other generation, you can see that homozygous recessive genes will express. ಯಾವಾಗ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗುತ್ತೋ ನೋಡೋಣ, ಹೇಳಿ. ಯು ಶುಡ್ ಆನ್ಸರ್ ದಿಸ್. ಈಸ್ ಇಟ್ ರೈಟ್ ಆರ್ ಈಸ್ ಇಟ್ ರಾಂಗ್ ಟು ಕಂಟಿನ್ಯೂ ದಿಸ್ ಕೌ ಫಾರ್ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮ್? ಇದನ್ನ ಮತ್ತೆ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮ್ ನಲ್ಲಿ ಇನ್ವಾಲ್ವ್ ಮಾಡ್ಕೊಳ್ಳೋದು ಸರಿನಾ ತಪ್ಪಾ? ವೆರಿ ಗುಡ್. ಐ ಕ್ಯಾನ್ ಸಿ ಕ್ವಿಕ್ ಆನ್ಸರ್. ಯಾರೊಬ್ರು ಆನ್ಸರ್ ಮಾಡಿ ರಾಂಗ್. ಇಟ್ ಇಸ್ ನಾಟ್ ಗುಡ್ ಟು use that offspring as a source of breeding program. ಏನಕ್ಕೆ ಅಂತ ಹೇಳಿದ್ರೆ, we know that this is carrying a dangerous gene. ಅದರಿಂದ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ, this one, it will be eliminated from breeding program. Now if you go back to what we were seeing here, it is mating of more closely related individuals within the same breed. ಒಂದೇ breed ಇರಬೇಕು, closely related individual ಆಗಿರಬೇಕು. ಎಷ್ಟು ಜನರೇಷನ್ ವರ್ಗು ಅಂತ ಹೇಳಿದ್ರೆ 4ರಿಂದ 6 ಜನರೇಷನ್ ವರ್ಗು. Then you call it as inbreeding. Now, superior male and superior females of the same breed are identified and mated in a pairs. Progeny obtained are evaluated and superior males and females among them are identified and further mated. ಅಂದ್ರೆ ಅದರ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಗಳನ್ನೇ ಮತ್ತೆ ಅವುಗಳ ನಡುವೆನೇ ಬ್ರೀಡಿಂಗ್ ಮಾಡ್ತಾ ಹೋಗುದು. So what do you mean by superior female in case of cattle is something which produces more milk. What do you mean by superior male is something which is going to give rise to a more strong and superior progeny. What are the advantages of inbreeding? Just now we saw inbreeding leads to increase in homozygosity and thence it will help in developing of pure line plant. ಎಲ್ಲ ಒಂದೇ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಬರ್ತಾ ಹೋಗುತ್ತೆ. ಎಲ್ಲ ಹೆಟ್ರೋಸೈಗಸ್ ಆಗ್ಬೋದು ಅಥವಾ ಎಲ್ಲ ಹೋಮೋಸೈಗಸ್ ಆಗ್ಬೋದು. ದಟ್ ಇಸ್ ವಾಟ್ ವಿ ಮೀನ್ ಬೈ ಪ್ಯೂರ್ ಲೈನ್ ಅನಿಮಲ್ಸ್. ಇಟ್ ಎಕ್ಸ್ಪೋಸಸ್ ಹಾರ್ಮ್ಫುಲ್ ರಿಸೆಸಿವ್ ಜೀನ್ಸ್ ದಟ್ ಆರ್ ಎಲಿಮಿನೇಟೆಡ್ ಬೈ ಸೆಲೆಕ್ಷನ್. ಇದನ್ನೇ ನಾನು ಲಾಸ್ಟ್ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ನೋಡಿದ್ದೆ. ಈ ತರ ಹಾರ್ಮ್ಫುಲ್ ರಿಸೆಸಿವ್ ಜೀನ್ಸ್ ಗಳಿದ್ರೆ ಇನ್ಬ್ರೀಡಿಂಗ್ ಮಾಡ್ತಾ ಎಲ್ಲೋ ಒಂದ್ ಕಡೆ ಅದು ಎಕ್ಸ್ಪೋಸ್ ಆಗುತ್ತೆ. ಅದರಿಂದ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಎಲಿಮಿನೇಟ್ ದಿಸ್ ಫ್ರಮ್ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮ್. ಸಾಯಿಸೋದು ಅಂತಲ್ಲ, ಮತ್ತೆ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮಿಗೆ ಕಂಟಿನ್ಯೂ ಮಾಡ್ಕೊಂಡು ಹೋಗೋಣ. ಅದು ಇನ್ನೊಂದು ಅಡ್ವಾಂಟೇಜ್. ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಅಕ್ಯುಮುಲೇಷನ್ ಆಫ್ ಸುಪೀರಿಯರ್ ಜೀನ್ಸ್ ಅಂಡ್ ಎಲಿಮಿನೇಷನ್ ಆಫ್ ಲೆಸ್ ಡಿಸೈರಬಲ್ ಜೀನ್. ಪ್ರತಿ ಜನರೇಷನ್ ನಲ್ಲೂ ಸುಪೀರಿಯರ್ ಮೇಲ್ ಮತ್ತು ಸುಪೀರಿಯರ್ ಫಿಮೇಲ್ಸ್ ಗಳನ್ನೇ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಳ್ತಾ ಹೋಗೋದ್ರಿಂದ we will eliminate less desirable gene, but it keeps on accumulating superior genes together. This approach increases the productivity of inbred population. But what are the disadvantages of it? What is the main disadvantage of inbreeding is it leads to what is called as inbreeding depression. I'm sure you people have heard about what you mean by inbreeding depression is. Inbreeding depression is reduction in fertility as well as productivity due to continuous inbreeding. It is called as inbreeding depression. Continued inbreeding, especially close inbreeding, may reduce the fertility and productivity. This is what we call it as inbreeding depression anta karikare. So how to solve this problem? It is best suitable to continue inbreeding only for about 4 to 6 generations. After 4th to 6th generation, it leads to what is called as inbreeding depression. ಹಾಗಾದ್ರೆ 6th generation ಆದ್ಮೇಲೆ to solve this problem, selected animals should be mated with unrelated superior animals, which will lead to formation of what is called as heterozygous condition. Homozygous condition ಬಂದಿರುತ್ತೆ, ಅದರಿಂದ inbreeding depression ಆಗ್ತಾ ಇರುತ್ತೆ. ಸಡನ್ ಆಗಿ ಯಾವ್ದಾದ್ರು ಅನ್ರಿಲೇಟೆಡ್ ಸುಪೀರಿಯರ್ ಅನಿಮಲ್ಸ್ ಜೊತೆ ನೀವು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಇಟ್ ವಿಲ್ ಲೀಡ್ ಟು heterozygous condition and thereby it is going to prevent or it is going to reduce the inbreeding depression. Now, that is what we studied about. That is about inbreeding. Now, what we are going to see now is called as outbreeding. It is the breeding of unrelated animals, which may be between individuals of same breed or between different breeds, or it may be between different species also. If it is between the same breed, you call it as outcrossing. If it is between different breeds, you call it as crossbreeding. If it is between different species, is called as interspecific hybridization. Now let us try to understand further. What do you mean by outbreeding? First of all, I hope you are going to keep understanding this in reference to inbreeding. Inbreeding anta helidre it is a cross between a superior male and a superior female, which belongs to the same breed, and it should be having common ancestor. It should be having common ancestor for about 4 to 6 generations. Outcrossing is mating between same breed. Inbreeding tarane outcrossing allu same breed alle agutte. But they will be not having a common ancestor. Inbreeding alli enagutte anta helidre they will be having common ancestor, but outcrossing nalli, not outbreeding, outcrossing nalli, there will be no common ancestor on either side of the pedigree for about 4 to 6 generations. Then you call it as outcrossing. The offsprings of such mating is called as outcross. This is the best method for increasing the milk productivity. More importantly, this is the best method to overcome the inbreeding depression. Inbreeding depression is because of inbreeding, where the fertility and productivity is going to decrease. Inbreeding ಮಾಡ್ತಾ fertility ಮತ್ತು productivity ಕಡಿಮೆ ಆಯ್ತು ಅಂತ ಗೊತ್ತಾದಾಗ inbreeding depression ಅನ್ನ ಹೋಗ್ಲಾಡಿಸಲಿಕ್ಕೆ, to overcome that inbreeding depression, they are going to go for outcrossing, which means they are going to cross between the same breed, but they will be having no common ancestor for about 4 to 6 generations. Then you call it as outcrossing. For example, this is one ancestor, this is another ancestor. You breed them, you are going to get these 3. Now again you breed this and this, you will get this one. Now idanna you call it as inbreeding anta karitare. This you can continue for about 4 to 6 generations. But then you will go for outcrossing, unrelated female or unrelated male. When you cross it with an individual which is the product of inbreeding, then it is called as outcrossing. Next is crossbreeding. I already told you what I mean by crossbreeding. In this method, male of one breed is mated with the superior female of the another breed. Eradu bere bere breed. Whereas now ಹಿಂದೆ ನೋಡಿದ್ದು outcrossing ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ it is the same breed, but having no common ancestor for about 4 to 6 generations. Whereas in this case, it is a cross made between the same breed. Sorry, it will be between the different breed. Superior male of one breed is mated with the superior female of another breed. This allows desirable quantities of 2 different breeds to combine to form progeny hybrid animal that may be used for commercial production of different products. Example important. E point ಜ್ಞಾಪಕ ಇಟ್ಕೊಂಡಿರಿ. Examples of this is that crossed and Bikaner Eve. Bikaner Eve was crossed with Merino ram. Bikaner ಅನ್ನೋದು ರಾಜಸ್ಥಾನಲ್ಲಿ ಇರೋ ಜಾಗ, ಅದರ ಈಗ ನಮ್ಮಲ್ಲೆಲ್ಲ ಹೇಳ್ತಾರೆ, ಈಗ ಮುದ್ದೋಳ್ ನಾಯಿ ಅಥವಾ ಇನ್ನೊಂದು ಯಾವುದೋ ಕುರಿ ಜಾತಿ ಇನ್ನೊಂದು ಹೇಳ್ತಾರೆ, ಡೆಕ್ಕನ್ ಅಂತ ಒಂದು ಕುರಿ ಬ್ರೀಡ್ ಇದೆ, ಅಥವಾ ಪೊಮೇರಿಯನ್ ಬ್ರೀಡ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಆ ತರ ಬಿಕನೇರ್ ಈವ್ ಇಸ್ ಒನ್ ಬ್ರೀಡ್ ಆಫ್ ಶೀಪ್. ಮೆರಿನೋ ರ್ಯಾಮ್ ಇಸ್ ಆಲ್ಸೋ ಅಂಡ್ ಶೀಪ್ ವಿಚ್ ವಾಸ್ ಇಂಪೋರ್ಟೆಡ್ ಫ್ರಮ್ ಯುರೋಪಿಯನ್ ಕಂಟ್ರೀಸ್. ಇದೆರಡನ್ನು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಬಂದಿದ್ದು ಸುಪೀರಿಯರ್ ವೆರೈಟಿ ಆಫ್ ಬ್ರೀಡ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಇಸಾರ್ಡೇಲ್. ಇಸಾರ್ ಅನ್ನೋದು ಪಂಜಾಬ್ ಹತ್ರ ಇರೋ ಒಂದು ಊರು. ಆ ಊರಲ್ಲಿ ರಿಸರ್ಚ್ ಸ್ಟೇಷನ್ ಅಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡಿದ್ರಿಂದ ಆ ಕುರಿನ ಇಸಾರ್ಡೇಲ್ ಅಂತ ಕರೀತಾರೆ. ಇದು ಬಿಕನೇರ್ ಈವ್. ಬಿಕನೇರ್ ಈವ್, ಇದು ಮೆರಿನೋ ರ್ಯಾಮ್. ಎರಡು ಬೇರೆ ಬೇರೆ ಬ್ರೀಡ್. ಇದು ಎರಡನ್ನು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಬಂದಿದ್ದು ಇಸಾರ್ಡೇಲ್. ಇಸಾರ್ಡೇಲ್ ಇಸ್ ಅನ್ hybrid got from cross between Bikaner Eve. This is female. This is female and this is male. So what do you mean by outbreeding? It is the mating of male and female animals of two different species itself. ಎರಡು ಬೇರೆ ಬೇರೆ ಪ್ರಭೇದ. Progeny formed may combine desirable features of both the parent and it may be of considerable economic value. For example, horse is very strong, but not easy to control. But donkey is not strong as horse, but it is easy to control. Now, if you cross donkey, which is a different species, equus asinus, with horse, which is a different breed, which is equus caballus, the resulting one, it will be mule, which is an interspecific hybrid. And I hope you know the product of interspecific hybrids will be sterile, or you can say it will be infertile. So animal breeding nalli agale ಎರಡು ತರ. I hope you have understood. Inbreeding is breed between the same breed. Chaitra, raising hands will be of no use. Please type your questions in question and Q&A box. Inbreeding is breed between the same breed and it should be having a common ancestor for about 4 to 6 generations, same breed. Outbreeding, it is having outcrossing. It will be between the same breed. Will be between, this is not correct. It will be between the same breed, but having no common ancestor for about 4 to 6 generations. Whereas crossbreeding is a breed between two separate breed. I hope you remember the example. ಯಾವುದು ನಾವೀಗ ನೋಡಿದ್ದು ಅಂತ ಹೇಳಿದ್ರೆ ಬಿಕನೇರ್ ಈವ್ ಅಂಡ್ ಮೆರಿನೋ ರ್ಯಾಮ್. Interspecific hybridization ಅಂತ ಹೇಳಿದ್ರೆ it will be cross between two different species itself, such as cross between horse and donkey. Next one is artificial insemination. See, imagine this situation. ನಿಮ್ಗೆ ಈ ಸಿಚುವೇಶನ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. Let's say in Karnataka there is a place called as Raichur, and in somewhere in Maharashtra there is a small village. In Raichur there is an extremely superior bull, and far away in some village, let's say there is an extremely bull andre artha agirbodu, it means we are talking about male. In yavado sun village ali, let's say there is an extremely superior female ide anta ankoಣ. Now distance between them is several hundreds of kilometers, so it is not practically possible that you are going to take the superior bull to mate it with superior female, which is hundreds of kilometers away. ಅವಾಗ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ, they are going to collect sperm from this male and introduce into the genital tract of superior female. That method is called as artificial insemination. This process Semen is collected from male parent and injected into the reproductive tract of selected female by breeders. Lord, please don't forget that we are talking about transporting it for a long distance. ಅಷ್ಟೊಂದು ದೂರ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಮಾಡುವಾಗ ಏನಾಗಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಸತ್ತೋಗ್ಬೋದು. ಅದರಿಂದ ಸ್ಪರ್ಮ್ಸ್ ಆರ್ ಸೀಮನ್ ಆರ್ ಫ್ರೋಸನ್ ಯೂಸಿಂಗ್ ಸಾಲಿಡ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಆರ್ ಕ್ರಯೋ ಪ್ರಿಸರ್ವೇಶನ್ ಮೆಥಡ್ ಅಲ್ಲಿ ಅದನ್ನ ಫ್ರೀಸ್ ಮಾಡಿ ಸೇಫ್ ಆಗಿ ತರ್ತಾರೆ. The success rate of crossing mature male and female animals is low, even though if artificial insemination is carried out. This is how artificial insemination is done. If you observe this, they will be collecting and sperm in this, and they are going to introduce that sperm into a reproductive tract of female. Sometimes it may be even introduction of embryos also. So what do you mean by multiple ovulation embryo transfer? Please remember, this is meant for increasing the herd size. ಹಸುಗಳು ಎಷ್ಟಿರುತ್ತೋ ಹಸುಗಳ ಸಂಖ್ಯೆಯನ್ನ ಜಾಸ್ತಿ ಮಾಡಲಿಕ್ಕೆ, to increase the number of cattle or herd, it is going to be useful. In this process, cow is administered with hormones like FSH. You know, FSH stands for follicle stimulating hormone, which induces the development of follicles, and hence, instead of producing one egg, it is going to produce 6 to 8 eggs. Production of 6 to 8 egg due to injection of FSH, it is called as super ovulation anta karitare. So first step in embryo transfer technology or MOET is subjecting a superior female to super ovulation. Super ovulation heng ಮಾಡಿಸೋದು? By injecting an hormone called as follicle stimulating hormone. Then that female who has been subjected for super ovulation is either mated with an elite bull, or it may be subjected for artificial insemination. In either way, what happens is sperms are released into female reproductive tract. Inside female reproductive tract, just now we saw, because of super ovulation, around 6 to 8 eggs will be present, and those 6 to 8 eggs will undergo fertilization, and each of them, pratiyondu, ಇನ್ನೊಂದ್ಸರಿ ಹೇಳ್ತಾ ಇದ್ದೀನಿ, ಪ್ರತಿಯೊಂದು, each of these egg will develop into 8 to 32 cell stage of embryo. Now that embryos are recovered, which means theoretically speaking, you may get around 6 to 8 egg ಏನಿರುತ್ತೋ, all 6 to 8 eggs have been converted into 6 to 8 embryos, which means you are going to get 6 to 8 embryos which are at 8 to 32 cells, ಅಥವಾ 8 to 62 celled stage. ಪ್ರತಿಯೊಂದು ಎಂಬ್ರಿಯೋನಲ್ಲೂ 8 ರಿಂದ 32 ಸೆಲ್ಗಳಿರೋ ಸ್ಟೇಜ್ಗಳ ಎಂಬ್ರಿಯೋನ ಆಚೆಗೆ ತೆಗಿತಾರೆ, and those are transferred into surrogate mothers. Now, high milk yielding breed of female and high quality that lean meat with less lipid meat yielding bulls have been bred successfully to increase the herd size. ಇದನ್ನ ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡ್ತೀನಿ ನೋಡಿ. What do they do? First ಕೆಲಸ ಮಾಡೋದು, they are going to subject it for super ovulation, which means after 4 to 8 days after super ovulation, it is going to produce, ಎಷ್ಟು egg produce ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ 6 to 8 eggs are produced. Now after that 6 to 8 eggs are produced, you can subject it for mating. Mating ಆದ್ಮೇಲೆ it is going to produce 6 to 8 embryos. Now observe this sentence. It says isolation of those embryos and transfer those embryos into surrogate mothers. After 9 months, you are going to get 6 to 8 calves. 6 ರಿಂದ 8 ಮರಿ ಬರುತ್ತೆ, because it has been subjected for super ovulation. Suppose if it was not done, ಇದು ಮಾಡಿರ್ಲಿಲ್ಲ ಅಂದಿದ್ರೆ, then this female would have produced only one calf, ಒಂದೇ ಒಂದು ಕರು ಆಗ್ತಾ ಇತ್ತು. But illénagide anta helidre 6 to 8 calves are produced, and this superior female has not undergone gestation, which means she is now again free to undergo superovulation, which means in next month she will again produce 6 to 8 embryos and the whole cycle continues. ಈ ತರ ಸೈಜ್ ಜಾಸ್ತಿ ಮಾಡೋದನ್ನ ಅಂದ್ರೆ ನಂಬರ್ ಆಫ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಗಳನ್ನ ಜಾಸ್ತಿ ಮಾಡೋ ಟೆಕ್ನಿಕ್ ಅನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಅಸ್ multiple ovulation. I hope you can understand the name itself. Multiple ovulation. It is because of superovulation. And then there will be embryo transfer. So, multiple ovulation, embryo transfer increases the number of herds or number of offsprings. It is to animal breeding na lodindira. After this, you have studied about beekeeping, which is also called as apiculture. Apiculture maduvaga, you should be having following ideas. And person who is doing apiculture or beekeeping should have following knowledge. Number one, knowledge and of the nature and habits of the bee. Bee colony nalli yav yavudirutte? Drone, queen, worker. What is the function of drone? What is the function of worker? How queens behave? Yavaga jane tuppa jasti agutte? Yav tara jane tegiyodu? Selection of suitable location for keeping beehive, which should be behind some windbreaks. There should be no direct sunlight. It should be having good water source. There should be no water water ನಿಂತ್ಕೊಂಬಾರ್ದು, ನಿಂತಿದ್ದ ನೀರೆಲ್ಲ ನಿಂತ್ಕೊಂಬಾರ್ದು. There should be good drainage. So that knowledge should be having a person who is doing this. Catching and hiving of swarms. After production of honey, how to carefully take out that honey without killing the bees. That knowledge you should be having. Management of beehive during different seasons. You know, India being partly temperate region, we will be not having rain always. Adrinda hoova yavaglu produce agala. If flowers are not produced throughout the year, then it will be difficult for honeybees to survive. So during those off season, how to maintain honeybees? Spring ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಅರ್ಲಿ ಸಮ್ಮರ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಲೇಟ್ ಸಮ್ಮರ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಅದನ್ನೆಲ್ಲ ಗೊತ್ತಿರಬೇಕು. ಯಾವ್ಯಾವ ಸೀಸನ್ ಅಲ್ಲಿ ಯಾವ್ಯಾವ ತರ ಅದನ್ನ ಮೇಂಟೈನ್ ಮಾಡಬೇಕು ಅನ್ನೋದು ಗೊತ್ತಿರಬೇಕು. ಹ್ಯಾಂಡ್ಲಿಂಗ್ ಅಂಡ್ ಕನೆಕ್ಷನ್ ಆಫ್ ಹನಿ ಅಂಡ್ ಬಿ ವ್ಯಾಕ್ಸ್. ಒಂದ್ಸರಿ ಹನಿ ಪ್ರೊಡಕ್ಷನ್ ಆದ್ಮೇಲೆ ಹನಿ ತೆಗಿಯೋದು ಹೇಗೆ, ಅದರಿಂದ ಬರೋ ಬಿ ವ್ಯಾಕ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡಬೇಕು. All these are the knowledge that a person should be having. Just 1 minute, just give me a minute. Yeah. So next thing that you have studied is about fisheries. Fisheries ಬಗ್ಗೆ ನಿಮ್ಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿರೋದು, which are the examples of freshwater fishes and which are the examples of marine water fishes. So examples of freshwater fishes includes these things: katla, which is called as katla scientifically; rohu, which is called as Libia rohita; and common carp. These are the common freshwater fishes which are either reared, catched, sold, or it can be processed and sold. Remember the names. And these are the marine water fishes, sea water, samudradalirodu. One is hilsa, which I think you know, very famous fish in Kolkata. And the other one is sardine. ಕನ್ನಡದಲ್ಲಿ ಇದನ್ನ ಮಂಗಳೂರು ಸೈಡ್ ಭೂತಾಯಿ ಮೀನು ಅಂತ ಹೇಳ್ತಾರೆ. ಮಾರ್ಕೆಲ್, ಬಾಂಗ್ಡೆ ಮೀನು ಅಂತ ಹೇಳ್ತಾರೆ. ಪಾಂಫ್ರೆಟ್. These are the some examples of marine water fishes. Marine water fishes ಮತ್ತು fresh water fishes ಗಳ ಹೆಸರು ಜ್ಞಾಪಕ ಇಟ್ಕೊಂಡಿದ್ದೀವಿ. Now there are two main techniques in fisheries. One is called as aquaculture and the other one is pisciculture. First look at the pisciculture. In pisciculture, what happens is breeding and rearing of only fishes in water, bodies for commercial purpose is called as pisciculture. Whereas in case of aquaculture, it is not just about fish. Bari meen matra alla, culturing of all aquatic organisms, including fish or prawns, crabs, shells, in both brackish and marine environment, including some of the seaweed. Seaweedಗಳನ್ನು ವೆಸ್ಟರ್ನ್ ಕಂಟ್ರೀಸ್ ಅಲ್ಲಿ ತಿನ್ನಿಸ್ತಾರೆ, ಅದನ್ನು ತಿಂತಾರೆ, ಅದನ್ನು ಬೆಳೆಯೋದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಆಕ್ವಾಕಲ್ಚರ್. Whereas pisciculture is only for fish, not any other aquatic organisms. ಇಲ್ಲಿ ನಿಮ್ಗೆ ಡಿಫ್ರೆನ್ಸ್ ಅರ್ಥ ಆಗ್ಬೋದು. ಇಲ್ಲಿ ನಡೀತಾ ಇರೋದು ಫಿಶ್ ಫಾರ್ಮಿಂಗು ನಡೀತಾ ಇದೆ, ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಮೈಕ್ರೋ ಆಲ್ಗೆನೂ ನಡೀತಾ ಇದೆ, ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಮಸಲ್ಸು ಆಗ್ತಾ ಇದೆ. ಎಲ್ಲಾದು ಆಗ್ತಾ ಇರೋದ್ರಿಂದ ದಿಸ್ ಇಟ್ ವಿಲ್ ಬಿ ಕಾಲ್ಡ್ ಆಸ್ ಆಕ್ವಾಕಲ್ಚರ್. ಬರೀ ಮೀನ್ ಮಾತ್ರ ಆದ್ರೆ, ಕಮರ್ಷಿಯಲ್ ಆಗಿ ಈ ತರ ಮೀನ್ ಸಾಕಿದ್ರೆ ಮೀನನ್ನು ಮಾತ್ರ ಸಾಕಿದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿ ಕಾಲ್ಡ್ ಆಸ್ pisciculture. ಸೊ ಇಲ್ಲಿವರೆಗೂ ನೋಡಿರೋದನ್ನ ಯು ಸ್ಟಡಿಡ್ ಇಟ್ ಆಸ್ ವಾಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅನಿಮಲ್ ಹಸ್ಬೆಂಡ್ರಿ, ಪ್ಲಸ್ ವಿ ಹ್ಯಾವ್ ಸೀನ್ ಅಬೌಟ್ ಎಪಿಕಲ್ಚರ್ ಅಂಡ್ ಫಿಶರೀಸ್. Now let us look into the plant breeding technique. What do you mean by plant breeding? It is a purposeful manipulation. What do you mean by manipulation? Change. Purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation and give better yield and are disease resistant. That is what we call it as plant breeding. I'm sure you heard about green revolution. It was basically the point where the production of agriculture products such as wheat, rice, maize increased. And green revolution ಆಗಿದ್ದ ಕಾರಣ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಬ್ರೀಡಿಂಗ್ ಮಾಡಿ, ಪ್ಲಾಂಟ್ ಅನ್ನ ಬೇರೆ ಬೇರೆ ತರದ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್ ಮುಖಾಂತರ ಇಂಪ್ರೂವ್ ಮಾಡಿ. ಸೊ ವಾಟ್ ಆರ್ ದ ಸ್ಟೆಪ್ಸ್ ದೆನ್ ಇನ್ ದ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್? ಒನ್, collection of variability. I hope you remember, you have heard about a word called as germplasm anta odidira, or there should be collection of germplasm. Then should be evaluation and selection of parents from this germplasm. Select agiro parents galana, it will be subjected for cross hybridization. Parents will be subjected for cross hybridization. And then there will be selection and testing of superior recombinant which comes because of cross hybridization. And these superior recombinants are tested and released as a new cultivars. Now let's look into each one of them separate steps. First one is collection of variability. ಅಂದ್ರೆ ಯಾವ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಹೈಬ್ರಿಡ್ ಮಾಡಕ್ ಹೊರಟಿದಿರೋ ಆ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ there should be germplasm collection. And what do you mean by germplasm collection? I'm sorry. The entire collection of the plants or seeds having all the alleles for all genes in a given crop is called as germplasm. Udaharanege, if you consider this as an different rice varieties, there may be some varieties which are very tall, there may be some varieties which are very dwarf. Which one do you think is better? Which triad is better for cultivation of rice? Udda irodu olleda, dwarf irodu olleda? Can I get a quick reply? Yeah, what is the reply we got? Tall. Yes, username by RM anta yaro username iroru, you are given it correct. What is suitable is being dwarf, kullukirಬೇಕು. ಟಾಲ್ ಆದ್ರೆ ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಪ್ಲಾಂಟ್ಸ್ ಗಳಲ್ಲಿ ಸ್ವಲ್ಪ ಬೆಳೆದ ನಂತರ ಆಫ್ಟರ್ ಗ್ರೋಯಿಂಗ್ ಫಾರ್ ಸರ್ಟನ್ ಸ್ಟೇಜ್ ಇಟ್ಸ್ ಸ್ವಲ್ಪ ಗಾಳಿ ಬಂದ್ರೂ ಬಿದ್ದೋಗುತ್ತೆ. So dwarfness is better. Now let us assume tall as a character of one more character that is more yield, whereas dwarf as a character that is short but less yield. Now, what should be the desirable character is, as somebody rightly mentioned, it should be tall and, it should be dwarf. It should be dwarf, plus it should be having more yield. Now, if you want to produce this kind of plant, ಈ ತರ ಕುಳ್ಳುಕಿರೋ ಗಿಡ ಮತ್ತೆ ಜಾಸ್ತಿ yield ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋ ಗಿಡ ಇರಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ನಿಮಗೆ ಮೊದಲು ಈ ಕ್ಯಾರೆಕ್ಟರ್ ಇರೋ ಗಿಡಗಳು ಬೇಕು. ಯಾವ ಕ್ಯಾರೆಕ್ಟರ್ ಇರೋದು? ಮೋರ್ yield ಮತ್ತೆ ಡ್ವಾರ್ಫ್ನೆಸ್ ಇರೋ ಗಿಡಗಳು ಬೇಕು. ಇನ್ಯಾವುದೋ ಒಂದು ಗಿಡ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಲೆಟ್ಸ್ ಸೇ ಇದರಲ್ಲಿ ವಿಟಮಿನ್ ಜಾಸ್ತಿ ಇರೋ ರೈಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಇನ್ಯಾವುದ್ರಲ್ಲಿ ಲೆಸ್ ವಾಟರ್ ಅನ್ನು ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಗಿಡ ಬೆಳೆಯುತ್ತೆ. ಇನ್ಯಾವುದೋ ಒಂದು ಇನ್ಸೆಕ್ಟ್ ಗೆ ರೆಸಿಸ್ಟೆಂಟ್ ಆಗಿರೋದಿದೆ. ಈ ಎಲ್ಲಾ ಕ್ಯಾರೆಕ್ಟರ್ ಗಳ ಕಾಂಬಿನೇಷನ್ ಇರೋ ಒಂದು ಕಲೆಕ್ಷನ್ ಅನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜರ್ಮ್ ಪ್ಲಾಸಮ್ ಕಲೆಕ್ಷನ್ ಅಂತ ಕರೀತಾರೆ, ವಿಚ್ ಇಸ್ ಒನ್ ಆಫ್ ದ ಪ್ರೈಮರಿ ಅಂಡ್ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಫಾರ್ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್. ಆಫ್ಟರ್ ದಿಸ್, among this germplasm, a germplasm ಒಳಗಡೆ you are going to evaluate and identify the plants with desirable combination of characters. ನಮಗೆ ಯಾವ ಕ್ಯಾರೆಕ್ಟರ್ ಬೇಕು? ಈಗ ಉದಾಹರಣೆಗೆ I want to make the tall plant which is having less yield, and I want to cross it with tall with more yield, but dwarf with less yield, so that my intention is to get a dwarf plant. My intention is to get a dwarf plant with more yield, which means I'm going to select this as one parent and this as another parent. That is what happens here, evaluation and selection of the parent. If wanted, this can be subjected for homozygous condition anna induce ಮಾಡಬಹುದು, breeding, self-pollination ಮಾಡಿ. And then comes the next step, which is cross-hybridization. During cross-hybridization, the plants are subjected for emasculation, bagging, dusting, ಮತ್ತೆ rebagging. ಈ 3-4 ಸ್ಟೆಪ್ ಅನ್ನ ನೀವು ಓದಿದ್ದೀರಾ ಬೇರೆ ಚಾಪ್ಟರ್ ನಲ್ಲೂ. From those steps, they are going to get, or they will try to bring about the combination of 2 different plants. So on cross-hybridization produces hybrids that are genetically combine the desired characters in one plant. ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ ನ ಒಟ್ಟಿಗೆ ತರುತ್ತೆ. But what are the problems with cross-hybridization is it is very time consuming and you may not always get the desirable characters together. After cross-hybridization, what you do is you're going to test for the superior recombinants. ನಮಗೆ ಬೇಕಾಗಿರೋ ಗಿಡಗಳು ಬಂದಿದೆ ಅಥವಾ ನಮಗೆ ಬೇಕಾಗಿರೋ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಗಳು ನೆಕ್ಸ್ಟ್ ಜನರೇಷನ್ ಅಲ್ಲಿ ಇದೆಯಾ? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ನನಗೆ ಏನ್ ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿಂದ ಡ್ವಾರ್ಫ್ನೆಸ್ ಬರಬೇಕು, ಇಲ್ಲಿಂದ ಮೋರ್ yield ಬರಬೇಕು. ದೇರ್ ಮೇ ಬಿ ಪಾಸಿಬಿಲಿಟಿ ಇದೆರಡು ಬಂದ್ಬಿಡ್ಬೋದು, ಟಾಲ್ ಮತ್ತೆ ಲೆಸ್ yield ಆಗ್ಬೋದು, ಅಥವಾ ಇಟ್ ಮೇ ಬಿಕಮ್ ಇನ್ನೊಂದೇನು ಕಾಂಬಿನೇಷನ್ ಆಗ್ಬೋದು, ಡ್ವಾರ್ಫ್ ಮತ್ತೆ ಮೋರ್ ಬರ್ಬೋದು. ನನಗೆ ಬೇಕಾಗಿರೋದು ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಡ್ವಾರ್ಫ್ನೆಸ್ ಇರಬೇಕು, ಪ್ರೊಡಕ್ಷನ್ ಜಾಸ್ತಿ ಇರಬೇಕು. ಅದನ್ನ ಯು ಕಾಲ್ ಇಟ್ ಆಸ್ ಹೈಬ್ರಿಡ್ ವಿಗರ್ ಅಂತ ಕರೀತಾರೆ. ನಮಗೆ ಬೇಕಾಗಿರೋ ಸುಪೀರಿಯರ್ ಕ್ಯಾರೆಕ್ಟರ್ ಇರೋದು. ದಟ್ ಇಟ್ ಮೇ ಬಿ ಸಬ್ಜೆಕ್ಟೆಡ್ ಫಾರ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಟು ಇಂಡ್ಯೂಸ್ ಹೋಮೋಸೈಗಾಸಿಟಿ. ಅದನ್ನ ಬೇರೆ ಬೇರೆ ಅಗ್ರೋನಾಮಿಕ್ ಟ್ರಯೇಡ್ಸ್ ಗೆ ಟೆಸ್ಟ್ ಮಾಡ್ತಾರೆ. ಗೊಬ್ಬರ ಎಷ್ಟು ಬೇಕು, ನೀರ್ ಎಷ್ಟು ಬೇಕು, ಯಾವ ತರದ ಮಣ್ಣಲ್ಲಿ ಬೆಳೆಯುತ್ತೆ, ಬೆಂಗಳೂರಲ್ಲಿ ಬೆಳೆದಾಗೇನೆ ಯಾದಗಿರಿಲಿ ಬೆಳಿಬೋದಾ, ಯಾದಗಿರಿಲಿ ಬೆಳೆದಾಗೆ ಆಂಧ್ರ ಪ್ರದೇಶದಲ್ಲಿ ಬೆಳಿಬೋದಾ, ಈ ತರ ಬೇರೆ ಬೇರೆ ಜಾಗದಲ್ಲಿ ಟೆಸ್ಟ್ ಮಾಡ್ತಾರೆ, ಅಟ್ ಲೀಸ್ಟ್ ಫಾರ್ 3 growing seasons in farmer's field. ಇದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಟೆಸ್ಟಿಂಗ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಅದಾದ್ಮೇಲೆ ಇಟ್ ವಿಲ್ ಬಿ ರಿಲೀಸ್ಡ್ ಇನ್ ಟು ಕಮರ್ಷಿಯಲ್ ಯೂಸ್. ಅದನ್ನ ಬೀಜಗಳನ್ನ ಆಮೇಲೆ ಮಾರಕ್ ಶುರು ಮಾಡ್ತಾರೆ. ಸೊ ಈ ತರದಲ್ಲಿ ಫಸ್ಟ್ ಹೈಬ್ರಿಡ್ ಪ್ಲಾಂಟ್ಸ್ ಇಂಡಿಯಾದಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದು, ಇಟ್ ವಾಸ್ ದ ಡಿಫರೆಂಟ್ ವೆರೈಟೀಸ್ ಆಫ್ ವೀಟ್. This is again important. See, ಗಮನ ಇಟ್ಟು ಕೇಳಿಸ್ಕೊಳ್ಳಿ. All 34 or what are the number of students who are present here. Rest of this chapter, it is more about you giving importance for memorizing these examples. I don't have anything to explain in these examples. Examples explain ಮಾಡಕ್ಕೆ ಏನು ಇಲ್ಲ. You just have to memorize it. ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕಷ್ಟೇ. ಇಂಡಿಯಾದಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡಿದಂತಹ ವೀಟ್ ವೆರೈಟೀಸ್ the wheat varieties that were introduced into India, which were semi-dwarf. So, Norman E. Borlaug, he was even awarded with Nobel Prize. He is considered as father of Green Revolution throughout the world. M.S. Swaminathan, India, but Norman E. Borlaug throughout the world. So, Nyapka Edkoli, he was working in International Center for Wheat and Maize Improvement in Mexico. He developed semi-dwarf wheat varieties. ಆ ಥರದಲ್ಲಿ ಇಂಡಿಯಾದಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಆಗಿದ್ದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಸೋನಾಲಿಕಾ ಅಂಡ್ ಕಲ್ಯಾಣ್ ಸೋಣ. ಇಲ್ಲಿಂದ ಮುಂದಕ್ಕೆ ಇರೋದೆಲ್ಲದನ್ನು ಇಟ್ ಇಸ್ ಅಬೌಟ್ ಮೆಮೊರೈಸಿಂಗ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್. Some semi-dwarf varieties of rice that were introduced into India. I am using the word introduced into India. ಇದನ್ನು ಇಂಡಿಯಾದಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡಿದ್ದಲ್ಲ, ಬೇರೆ ಕಡೆಯಿಂದ ತಂದು ಇಂಡಿಯಾದಲ್ಲಿ ಬೆಳೆಸಿದ್ ಯಾವ್ದು ಅಂತ ಹೇಳಿದ್ರೆ IR8, which was developed in International Rice Research Institute, which is present in Philippines, IR8. Taichung Native 1, which was the plant or rice variety which was introduced into India from Taiwan. Whereas Jaya and Ratna are the two varieties of semi-dwarf varieties of rice which were developed in India. Ratna ಮತ್ತು Jaya ಇದೆರಡನ್ನ ಇಂಡಿಯಾದಲ್ಲೇ ಡೆವಲಪ್ ಮಾಡ್ತಾರೆ. Whereas IR8 ಅನ್ನ ಇಂಟರ್ನ್ಯಾಷನಲ್ ರೈಸ್ ರಿಸರ್ಚ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಫಿಲಿಪೈನ್ಸ್ ಇಂದ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರೆ, Taichung Native 1 ಅನ್ನ ಇಟ್ ವಾಸ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ಡ್ ಫ್ರಮ್ ತೈವಾನ್. ಮತ್ತೆ ಎಕ್ಸಾಂಪಲ್ಸ್ ಯಾವ್ದು ಹೈಬ್ರಿಡ್ ಕ್ರಾಪ್ ಶುಗರ್ ಕೇನ್ ಅಲ್ಲಿ ಅಂತ ಹೇಳಿದ್ರೆ ಸ್ಯಾಕ್ರಮ್ ಬೆರ್ಬೆರಿ ಏನಿದೆಯೋ ಇದು ನಾರ್ತ್ ಇಂಡಿಯಾದಲ್ಲಿ ಬೆಳೀತಾರೆ, ಬಟ್ ಅದರಲ್ಲಿ ಶುಗರ್ ಕಂಟೆಂಟ್ ತುಂಬಾ ಕಡಿಮೆ ಇರುತ್ತೆ, ಅದರಿಂದ ಈಲ್ಡ್ ಆಲ್ಸೋ ಕಡಿಮೆ ಇರುತ್ತೆ. This Saccharum berberi was crossed with Saccharum officinarum, which is present in South India. So the resulting hybrid in India, it was able to grow in North India, but at the same time it had thick stem. North India dallu belibodagittu, thick stem ittu. Please remember the names. Saccharum beriberi, North India. Saccharum officinarum, South India. Saccharum beriberi nalli yield kadime ittu. Saccharum officinarum ನಲ್ಲಿ yield ಜಾಸ್ತಿ ಇದೆ. ಇದೆರಡನ್ನು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ they got a variety which was able to grow in North India. At the same time it had a better yield. The Saccharum berberi, North India, South India. Again, some more examples are like maize in hybrid, jowar hybrid, bajrado hybrid ಗಳನ್ನೆಲ್ಲ ಇಂಡಿಯಾದಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರು. ಇದೆಲ್ಲ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಆಗಿದ್ರಿಂದಾನೆ ನಮ್ಮಲ್ಲಿ ಗ್ರೀನ್ ರೆವಲ್ಯೂಷನ್ ಆಗಿ there was lot of improvement in the food production. Plant breeding for disease resistance ಅಂತ ಓದಿದ್ದೀರಾ? ನಮ್ಗೆ ಕಾಯಿಲೆ ಬಂದಾಗ we have to prevent that disease by spraying fungicides and bactericides. Fungus ಇಂದ ಕಾಯಿಲೆ ಬಂದ್ರೆ fungicides ಯೂಸ್ ಮಾಡ್ತಾರೆ, which are going to kill fungus. Bacteria in the kaile bandre, they are going to treat it with bactericides, which are going to kill bacteria. So resistance of the host plant is genetic ability to prevent the pathogen from disease without using the fungicides and bactericides. Again, it is all about examples. In textbook indane examples idu. On swalpa work ಮಾಡಿ, you have to memorize all this. Fungal disease ಯಾವ್ದು? Rust disease, which is caused by Puccinia triticina anta wheat ali, brown rust for example. Red rot of sugarcane, which is caused by Coleotrichum falcatrum. Late blight of potato, which is caused by Phytophthora infections. Black rot of crucifers is caused by a bacteria called as Xanthomonas campestris. Tobacco mosaic virus and turnip mosaic virus causes viral disease. How to breed the disease resistant? One conventional method. What do you mean by conventional method ಅಂತ ಹೇಳಿದ್ರೆ, ನಿಮ್ಮ germ plasm ನಲ್ಲಿ ಏನು ಕಲೆಕ್ಷನ್ ಇರುತ್ತೋ ಆ ಕಲೆಕ್ಷನ್ ಇಂದ ಪೇರೆಂಟ್ ಗಳನ್ನ ಚೂಸ್ ಮಾಡೋದು. ಯಾವ್ದಾದ್ರು ಒಂದು ಪೇರೆಂಟ್ ನಲ್ಲಿ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆಂಟ್ ವೆರೈಟಿ ಇದ್ರೆ ಅದನ್ನ ಕ್ರಾಪ್ ವೆರೈಟಿ, ಪೇರೆಂಟ್ ಅನ್ನ ಯು ಕ್ಯಾನ್ ಕಾಲ್ ಇಟ್ ಆಸ್ ವೈಲ್ಡ್ ವೆರೈಟಿ ಅಂತ. ನೀವು ಬೆಳೀತಾ ಇರೋ ಬೆಳೆ ಜೊತೆಗೆ ಕ್ರಾಸ್ ಮಾಡ್ತಾರೆ, ಅವಾಗ ಆ ಕ್ರಾಪಿಗೆ ಆ ಪೇರೆಂಟ್ ಅಲ್ಲಿ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆಂಟ್ ಕ್ಯಾರೆಕ್ಟರ್ ಇದ್ರೆ ಕ್ರಾಪಿಗೂ ಬಂದಿರುತ್ತೆ. ದಟ್ ಇಸ್ ಕನ್ವೆನ್ಷನಲ್ ಮೆಥಡ್. Again, concentrate and understand what I am telling now. Conventional method of breeding ಗೆ primary requirement ಇರೋದು ಏನು ಅಂತ ಹೇಳಿದ್ರೆ germplasm. Imagine a condition where in a germplasm disease resistant character illa ಅನ್ಕೊಳ್ಳಿ. Now you cannot go with conventional method. ಅವಾಗ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ in those conditions these crops are subjected for mutations. You know what you mean by mutations? Any sudden change in the heritable material is called as mutation, and the DNA ನಲ್ಲಿ change ಬರುತ್ತೆ. ಒಂದ್ಸರಿ DNA ನಲ್ಲಿ ಚೇಂಜ್ ಬಂತು ಅಂತ ಹೇಳಿದ್ರೆ ಅದ್ರ ಕ್ಯಾರೆಕ್ಟರ್ ಚೇಂಜ್ ಆಗುತ್ತೆ. ಆ ತರ ಕ್ಯಾರೆಕ್ಟರ್ ಚೇಂಜ್ ಆದಾಗ ಇಟ್ ಮೇ ಗೆಟ್ ಅಂಡ್ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್. ಸೊ ಜರ್ಮ್ಪ್ಲಾಸಮ್ ಕಲೆಕ್ಷನ್ ಅಲ್ಲಿ ಇಲ್ಲ, ಹಾಗಾದ್ರೆ ಕನ್ವೆನ್ಷನಲ್ ಮೆಥಡ್ ಮಾಡಕ್ ಆಗಲ್ಲ. Then you have to go for what is called as mutational breeding. So nothing to explain here again, but important. Plant breeding ನಲ್ಲಿ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಗೆ ಕ್ರಾಪ್ ಹೆಸರು wheat variety Imagiri. It is resistant against leaf and strip rust and eel bund. I'm telling you, most of the question may come from this, so be prepared. Solpa idike time kottu by part madi. Please memorize these things by heart madi. Crop Brassica varieties Pusa Savernum, also called as currant dry, which is resistant against white rust. Cauliflower Pusa Subra, Pusa Snowball K1. I can tell you what you mean by Pusa. Adonde helbodu ashte, ಮಿಕ್ಕಿದೇನು ಇಲ್ಲ ಇಲ್ಲಿ. Pusa is a name of a place in Delhi, which is a place for Indian Agriculture Research Institute. Jaga Pusa. ಅಲ್ಲಿ ಮೇನ್ ಇನ್ಸ್ಟಿಟ್ಯೂಷನ್ ಇರೋದ್ರಿಂದ ಅದ್ರ ಕಂಟ್ರೋಲ್ ಅಲ್ಲಿ ಡೆವಲಪ್ ಆಗಿದ್ದ ವೆರೈಟೀಸ್ ಗಳನ್ನ ಅದಕ್ಕೆ ಸುಮ್ನೆ ಪೂಸಾ ಅಂತ ಕರೀತಾರೆ ಅಷ್ಟೇ. ಕೌಪಿ ಹೈಬ್ರಿಡ್ ವೆರೈಟಿ ಹೆಸರು ಪೂಸಾ ಕೋಮಲ್, ರೆಸಿಸ್ಟೆಂಟ್ ಟು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಬ್ಲೈಟ್ ಡಿಸೀಸ್. ಚಿಲ್ಲಿನಲ್ಲಿ ಪೂಸಾ ಸದಾಬಹಾರ್, which is resistant to chili mosaic virus, tobacco mosaic virus, as well as mutation breeding. ಈಗ ತಾನು ಹೇಳಿದ್ದೀನಿ, conventional breeding is constrained by the availability of the limited number of disease resistant gene in the germplasm. Germplasm ಅಲ್ಲಿ ಇದಿದ್ರೆ okay, ಇಲ್ಲ ಅಂದ್ರೆ ಏನ್ ಮಾಡೋದು? Then it will be subjected for mutation breeding. In mutation breeding, genetic varieties are created through changes in the base sequence within the genes, resulting in creation of new character. ಯಾವಾಗ DNA ನಲ್ಲಿ ಚೇಂಜ್ ಆಗುತ್ತೋ ಹೊಸ ಕ್ಯಾರೆಕ್ಟರ್ ಗಳು ಬರುತ್ತೆ, ಅದರಿಂದ ಬ್ರೀಡಿಂಗ್ ಅನ್ನ ಕಂಟಿನ್ಯೂ ಮಾಡ್ತಾರೆ. ಅಗೈನ್ ಎಕ್ಸಾಂಪಲ್, ಇಟ್ ಇಸ್ ಇನ್ ಯುವರ್ ಕೋರ್ಟ್, ನೀವೇ ಮಾಡ್ಬೇಕು ಇದನ್ನ. ಎಕ್ಸಾಂಪಲ್ ಯಾವ್ದು ಅಂತ ಹೇಳಿದ್ರೆ ಮಂಕ್ ಬೀನ್, ವಿಚ್ ಇಸ್ ರೆಸಿಸ್ಟೆಂಟ್ ಅಗೇನ್ಸ್ಟ್ ಯೆಲ್ಲೋ ಮೊಸಾಯಿಕ್ ವೈರಸ್. ಇದು ಯೆಲ್ಲೋ ಮೊಸಾಯಿಕ್ ವೈರಸ್ ಇಂದ ಇನ್ಫೆಕ್ಟ್ ಆಗಿರೋದು, ಈಲ್ಡ್ ಕಡಿಮೆ ಆಗುತ್ತೆ. ಇದರಿಂದ ರೆಸಿಸ್ಟೆನ್ಸಿಗೋಸ್ಕರ ಮಂಕ್ ಬೀನ್ resistant to yellow mosaic virus as well as powdery mildew were induced by mutation. Resistance to yellow mosaic virus in Vendi was transferred from wild species and resulted in new variety of Vendi called as Parbhani Kranti. Disease resistant ಬಗ್ಗೆ ನೋಡಿದೀವಿ, yield ಬಗ್ಗೆ ನೋಡಿದೀವಿ. Next is plant breeding for development of resistant against insect pest. ಹುಳ ತಪ್ಪಿಸೋದು ಹೇಗೆ? ಹುಳದಿಂದ ನಮಗೇನಾದ್ರೂ ರೆಸಿಸ್ಟೆನ್ಸ್ ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಅದು 3 ತರ ಬರ್ಬೋದು. ಯಾವ್ದು? ಮಾರ್ಫಾಲಜಿಕಲಿ, ಬಯೋಕೆಮಿಕಲಿ, ಆಸ್ ವೆಲ್ ಆಸ್ ಫಿಸಿಯೋಲಾಜಿಕಲ್. ಏರಿ ಲೀಫ್ ಅಂತ ಏನಿದೆ, ಇಟ್ ಇಸ್ ಅ ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್. ನೀವು ನೋಡಿರ್ತೀರಾ, ಚಿತ್ರನೂ ಕಾಣಿಸ್ತಾ ಇರ್ಬೋದು. ಈ ತರ ಎಲೆ ಮೇಲೆ ಅಥವಾ ಸ್ಟೆಮ್ ಮೇಲೆ ಸಣ್ಣ ಕೂದಲಿದ್ರೆ ಈ ತರದ ಇನ್ಸೆಕ್ಟ್ಸ್, ಅಫಿಡ್ಸ್ ಅಂತ ಕರೀತಾರೆ ಇದನ್ನ. ದೀಸ್ ಇನ್ಸೆಕ್ಟ್ಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಅಫಿಡ್ಸ್ ಅಥವಾ ಜಾಸಿಡ್ಸ್. ಆ ಜಾಸಿಡ್ಸ್ ಗಳು ನಮ್ ಬಾಡಿ ಮೇಲೆ ಯಾವ ತರ ಮಸ್ಕಿಟೋ ಕುತ್ಕೊಂಡು ಬ್ಲಡ್ ಇರುತ್ತಲ್ಲ, ಜಸ್ಟ್ ಲೈಕ್ ಮಸ್ಕಿಟೋ ಸಕ್ ಬ್ಲಡ್ ಫ್ರಮ್ ಅವರ್ ಬಾಡಿ, ದೀಸ್ ಜಾಸಿಡ್ಸ್ ಅಂಡ್ ಅಫಿಡ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸಕ್ ದ ಪ್ಲಾಂಟ್ ಕಂಟೆಂಟ್ ಆರ್ ಪ್ಲಾಂಟ್ ಸ್ಯಾಪ್. ಸೊ ಏರಿ ಲೀಫ್ ಇದ್ರೆ it will give resistant against jacids in cotton as well as cereal leaves beetle in wheat. Again, examples are very, very important. I am telling you repeatedly, examples galna ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ. ಸೊ ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ನಲ್ಲಿ ಎರಡನೇದು ರೆಸಿಸ್ಟೆನ್ಸ್ ಗೆ ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ನಲ್ಲಿ ಎರಡನೇದು ಸಾಲಿಡ್ ಸ್ಟೆಮ್ ಇನ್ ವೀಟ್. It will give resistance against stem soft lie. Stem soft lie ಗೆ ರೆಸಿಸ್ಟೆನ್ಸ್ ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಗಟ್ಟಿಯಾದ ಸ್ಟೆಮ್ ಇರಬೇಕು. ದಟ್ ಇಸ್ ಒನ್ ಮೋರ್ ಕ್ಯಾರೆಕ್ಟರ್. ಈಗ ತಾನೇ ನೋಡಿದ್ದು ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್. ಒನ್ ಮೋರ್ ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ಇಸ್ ಸ್ಮೂತ್ ಲೀವ್ಡ್ ಅಂಡ್ ನೆಕ್ಟರ್ ಲೆಸ್ ಫ್ಲವರ್. ನೆಕ್ಟರ್ ಅಂದ್ರೆ ಗೊತ್ತಿರುತ್ತೆ ನಿಮ್ಗೆ, ಮಕರಂದ ಅಂತ. ಇಫ್ ದೇರ್ ಇಸ್ ನೆಕ್ಟರ್, then bollworms will get attracted to the nectar in case of cotton. So if there is any plant or a cotton which will produce nectar less flower and it will be having smooth leaf, then it will have resistant against bollworm. ಇಲ್ಲಿವರೆಗೂ ನೋಡಿದ್ದೆಲ್ಲ ಮಾರ್ಫಾಲಜಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್. ಈಗಿರೋದು ಬಯೋಕೆಮಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್. ಆಸ್ಪಾರ್ಟಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರಬೇಕು, ಹೈ ಆಸ್ಪಾರ್ಟಿಕ್ ಕಂಟೆಂಟ್, ಬಟ್ ಲೋ ನೈಟ್ರೋಜನ್ ಅಂಡ್ ಶುಗರ್ ಕಂಟೆಂಟ್. ಆಸ್ಪಾರ್ಟಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇದ್ದು ನೈಟ್ರೋಜನ್ ಮತ್ತು ಶುಗರ್ ಕಡಿಮೆ ಇದ್ರೆ then maize will get resistant against maize stem borers. ಕಾಂಡ ಕೊರಕ ಅಂತ ಹೇಳ್ತಾರೆ ಕನ್ನಡದಲ್ಲಿ. ಅವುಗಳ ವಿರುದ್ಧ ರೆಸಿಸ್ಟೆನ್ಸಿಗೋಸ್ಕರ ಏನಿರಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಹೈ ಆಸ್ಪಾರ್ಟಿಕ್ ಆಸಿಡ್ ಇರಬೇಕು, ಬಟ್ ಇಟ್ ಶುಡ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಲೋ ನೈಟ್ರೋಜನ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಶುಗರ್. Again, I will quickly go through this different crop, different varieties, and again they are resistant against what. Next part of the chapter deals about what is called as biofortification. What do you mean by biofortification? It is breeding of crops which is having higher level of nutrients. This is solution for what is called as hidden hunger. Hidden hunger ಅಂತ ಹೇಳಿದ್ರೆ ಹೊಟ್ಟೆ ತುಂಬಿರುತ್ತೆ. There will be sufficient amount of food, but the person may not have, but the person may not have the right amount of nutrition. ಸರಿಯಾದ ಮಿನರಲ್ಸ್, ಸರಿಯಾದ ವಿಟಮಿನ್ಸ್, ಸರಿಯಾದ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಕಂಟೆಂಟ್, ಲಿಪಿಡ್ ಕಂಟೆಂಟ್ ಇದೆಲ್ಲ ಕಡಿಮೆ ಇರುತ್ತೆ, ಬಟ್ ಹೊಟ್ಟೆ ತುಂಬಿರುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇನ್ ಮೆನಿ ಪಾರ್ಟ್ಸ್ ಆಫ್ ದ ವರ್ಲ್ಡ್ ದ ಸ್ಟೇಪಲ್ ಫುಡ್ ಇಸ್ ರೈಸ್. ಮೇನ್ ಆಗಿ ಬರೀ ರೈಸ್ ತಿಂತಾರೆ ಕೆಲವೊಂದ್ ಕಡೆ. ಅವಾಗ ಬರೀ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಸಿಗುತ್ತೆ ಹೊರತು ನಿಮ್ಗೆ ಪ್ರೋಟೀನ್ಸ್ಗಳು, ವಿಟಮಿನ್ಸ್ಗಳು, ಐರನ್ಗಳು ಇದೆಲ್ಲ ಕಡಿಮೆ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಆ ತರದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಹಿಡನ್ ಹಂಗರ್. ಅದಕ್ಕೆ ಬಡತನ ಇರಬೇಕು ಅಂತ ಏನಿಲ್ಲ. ಒನ್ ಆಫ್ ದ ರೀಸನ್ ಇಸ್ ಪಾವರ್ಟಿ. ದ ಪರ್ಸನ್ ಈಟ್ಸ್ ಟೂ ಮಚ್ ಆಫ್ ಜಂಕ್ ಫುಡ್. ಹೊಟ್ಟೆ ತುಂಬಿರುತ್ತೆ, but still he will be not having sufficient amount of right type of diet. So anta caseಗಳಲ್ಲಿ biofortification is a method for improving the protein content as well as quality. When you say quality of protein, a protein which is rich in essential amino acids. I am sure you people know what do you mean by essential amino acid. Essential amino acid ಗಳು ಜಾಸ್ತಿ ಇದ್ರೆ then you call it as improved quality of proteins. Protein content ಅಂತ ಹೇಳಿದ್ರೆ ಪ್ರೋಟೀನ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ. Improved oil content and quality. Saturated ಮತ್ತು ಅನ್ಸ್ಯಾಚುರೇಟೆಡ್ ಫ್ಯಾಟಿ ಆಸಿಡ್ಸ್ ಅಂತ ಓದಿದ್ದೀರಾ. ನಮ್ಮ ಬಾಡಿಗೆ ಯಾವ್ದು ಒಳ್ಳೇದು ಅಂತ ಹೇಳಿದ್ರೆ ಸ್ಯಾಚುರೇಟೆಡ್ ಒಳ್ಳೇದಲ್ಲ, ಅನ್ಸ್ಯಾಚುರೇಟೆಡ್ ಒಳ್ಳೇದು. ಆ ತರದ್ದು ಕ್ವಾಲಿಟಿ ಇರೋ ಆಯಿಲ್ ಇರಬೇಕು. ವಿಟಮಿನ್ ಕಂಟೆಂಟ್ ಜಾಸ್ತಿ ಇರಬೇಕು. And it should be having more amount of micronutrients as well as mineral content. So lysine and tryptophan, both of them are the examples of essential amino acid. ಆ ತರದ ಮೇಜ್ ಅನ್ನ ಡಬಲ್ ದ ಅಮೌಂಟ್ ಆಫ್ ಲೈಸಿನ್ ಮತ್ತು ಟ್ರಿಪ್ಟೋಫಾನ್ ಇರೋ ಮೇಜ್ ಅನ್ನ ಹೈಬ್ರಿಡ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. Wheat variety called as Atlas 66. It is often used as one of the parent as a donor for high protein content. Iron fortified ಅಂತ ಹೇಳಿದ್ರೆ ಕಬ್ಬಿಣದ ಅಂಶ ಜಾಸ್ತಿ ಇರೋದು. Iron fortified rice variety containing 5 times as much as iron as in common varieties has been developed. ಅದೇ ತರ ವಿಟಮಿನ್ ಎ enriched carrots, spinach, pumpkin. Vitamin C enriched bitter gourd, batua, mustard, tomato. Iron and calcium enriched spinach and batua. Protein enriched beans such as broad, lab, French and garden peas. Again, I'm telling you, these examples are important. After this, you have studied about something called as single cell protein. Biofortification ಆದ್ಮೇಲೆ single cell protein. So one basic understanding ಇಲ್ಲಿ ಏನಂತ ಹೇಳಿದ್ರೆ, all our body requires sufficient amount of protein for body growth. But unfortunately, protein rich diet are all costly. ನಿಮ್ಗೆ ಒಂದು ಕೆಜಿ ಅಕ್ಕಿ ನಿಮ್ಗೆ 40-50 ರೂಪಾಯಿಗೆ ಸಿಕ್ಕಿದ್ರೆ ಒಂದು ಕೆಜಿ ಬೇಳೆ 180 ರೂಪಾಯಿ ಆಗುತ್ತೆ. ಬೇಳೆ, dal, it is rich in protein, it is costly. Carbohydrate rich rice is less. So unfortunately, production of protein is very costly. For example, ಈ example ನೋಡಿದ್ದೀರಾ ನೀವು, 250 kg cow produces 20 grams of protein per day. Instead of that, you can go for production of single cell proteins, which are the products of microorganisms. So by using microorganism, if you can increase the production of protein, that is what we call it as single cell protein. For example, Methylophilus methylotrophus, which is a bacteria, produces 25 tons of protein. If you compare the rate in which the cow is going to produce the protein, it is going to produce 25 tons of protein. ಇನ್ನೊಂದು ಎಕ್ಸಾಂಪಲ್ ನಮಗೆ ಇದು ಸಿಗೋದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಮಶ್ರೂಮ್, which we can produce from straw. ಹುಲ್ಲನ್ನ ಉಪಯೋಗಿಸಿಕೊಂಡು ಅಣಬೆ ಬೆಳಿಬಹುದು, which is very cheap. The last content in this chapter that you have studied is about tissue culture. Tissue culture ನಲ್ಲಿ, it is cultivating or growing of plant in laboratory condition. ನೀವು ಓದಿರೋ ಕೆಲವು ಇಂಪಾರ್ಟೆಂಟ್ ಟರ್ಮ್ಸ್ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಟೋಟಿಪೊಟೆನ್ಸಿ. What do you mean by totipotency? Ability of a cell to regenerate into an entire plant is called as totipotency, where toti means total, potency means potential. Potential ಅಂತ ಹೇಳಿದ್ರೆ ability. ಯಾವುದ್ರ ability? ಪ್ಲಾಂಟಿನ ಯಾವುದಾದ್ರೂ ಒಂದು ಸೆಲ್ ಅಥವಾ ಪ್ಲಾಂಟಿನ ಯಾವುದಾದ್ರೂ ಒಂದು ಭಾಗದಿಂದ ಇಡೀ ಪ್ಲಾಂಟ್ ಬೆಳೆಯೋದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಟೋಟಿಪೊಟೆನ್ಸಿ ಅಂತ ಕರೀತಾರೆ. Part of the plant that you take for tissue culture, it is called as explant. Excision ಅಂತ ಕರೀತಾರೆ, ಕಟ್ ಮಾಡಿ ತೆಗೆಯೋದು. ಪ್ಲಾಂಟಿನ ಯಾವ ಭಾಗನ ಕಟ್ ಮಾಡಿ ತೆಗಿತಾರೋ ಅದನ್ನ ಎಕ್ಸಿಷನ್, ಎಕ್ಸ್ಪ್ಲಾಂಟ್ ಅಂತ ಕರೀತಾರೆ, which is used for cultivating in laboratory condition. And for cultivating this explant, what is required is nutrient medium, which will be having carbon source, inorganic salts, vitamins, amino acids, and most importantly, plant growth regulators. ಈ ತರ ಬೆಳೆಸುವಾಗ you are going to get something called as callus, wherein callus is a mass of undifferentiated cells which is present in tissue culture ಅಥವಾ tissue culture lab ಅಲ್ಲಿ ಬೆಳೆಸುವಾಗ ಇಲ್ಲಿದೆ ನೋಡಿ ಮೇಲ್ಗಡೆ ಎರಡು, ಇದು ಸೆಲ್ಸ್ ಗಳ ಒಂದು ಸಮೂಹ. ಇಟ್ಸ್ ಅ ಮಾಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಸೆಲ್ಸ್ ವಿಚ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಥ್ರೂ ಟಿಶ್ಯೂ ಕಲ್ಚರ್. ದಿಸ್ ಇಸ್ ವಾಟ್ ಯು ಕ್ಯಾನ್ ಕಾಲ್ ಇಟ್ ಆಸ್ ಕ್ಯಾಲಸ್. ಸೊ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಬಗ್ಗೆ ಓದುವಾಗ ಯು ಹ್ಯಾವ್ ಸ್ಟಡಿಡ್ ಅಬೌಟ್ ಸಮಥಿಂಗ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್, ವಿಚ್ ಇಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಥೌಸಂಡ್ಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ಸ್ ಇನ್ ಅ ವೆರಿ ಶಾರ್ಟ್ ಟೈಮ್. ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್ and it is a method of asexual reproduction. Please remember this, micropropagation is a method of asexual reproduction. Adrinda, the resulting offsprings, it will be genetically identical. Asexual reproduction andre genetically identical agirlebeku. So genetically identical irodunna you call it as clones anta karitira. Somatic cells galinda ee clones bandirodrinda ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್ ಅಥವಾ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಮುಖಾಂತರ ಪಡೆಯುವ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಗಳನ್ನ ಯು ಕಾಲ್ ಇಟ್ ಆಸ್ ಸೋಮಾ ಕ್ಲೋನ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಟೊಮ್ಯಾಟೊ, ಬನಾನಾ, ಆಪಲ್, ಆಲ್ ದೀಸ್ ಪ್ಲಾಂಟ್ಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ ಥ್ರೂ ದಿಸ್ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್. ಎರಡೆರಡು ಕಲ್ಚರ್ ಓದಿದ್ದೀರಾ, ಒಂದು ಸ್ಟೆಮ್ ಕಲ್ಚರ್ ಅಂತ, ಸಾರಿ ಮೆರಿಸ್ಟೆಮ್ ಕಲ್ಚರ್ ಅಂತ. ಐ ಆಮ್ ಶೋರ್ ಮೋಸ್ಟ್ ಆಫ್ ಯು ನೋ ವಾಟ್ ಯು ಮೀನ್ ಬೈ ಮೆರಿಸ್ಟೆಮ್. ಮೆರಿಸ್ಟೆಮ್ ಇಸ್ ದ ಪಾರ್ಟ್ ಆಫ್ ದ ಪ್ಲಾಂಟ್ ವಿಚ್ ಇಸ್ ಆಕ್ಟಿವ್ಲಿ ಡಿವೈಡಿಂಗ್. ತುಂಬಾ ರ್ಯಾಪಿಡ್ ಆಗಿ ಡಿವೈಡ್ ಆಗ್ತಾ ಇರೋದ್ರಿಂದ, ಪ್ಲೀಸ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ವಾಟ್ ಐ ಆಮ್ ಟೆಲಿಂಗ್ ಕೇರ್ಫುಲಿ. ತುಂಬಾ ರ್ಯಾಪಿಡ್ ಆಗಿ ಡಿವೈಡ್ ಆಗ್ತಾ ಇರೋದ್ರಿಂದ ಮೆರಿಸ್ಟೆಮ್ ಇಟ್ ವಿಲ್ ಬಿ ಆಲ್ವೇಸ್ ಫ್ರೀ ಆಫ್ ವೈರಸ್. ಯಾಕಂದ್ರೆ ವೈರಸ್ ಅಲ್ಲಿ ಇನ್ಫೆಕ್ಟ್ ಮಾಡೋಷ್ಟ್ರೊಳಗಡೆ ಸೆಲ್ಗಳು ಡಿವೈಡ್ ಆಗಿ ಮುಂದಕ್ಕೆ ಹೋಗ್ಬಿಟ್ಟಿರುತ್ತೆ. ಸೊ ಅದರಿಂದ ಈಗ ಯಾವ್ದಾದ್ರು ಒಂದು ಪ್ಲಾಂಟ್ ಪೂರ್ತಿ ವೈರಸ್ ಇನ್ಫೆಕ್ಷನ್ ಆಗೋಗಿದೆ, ಈ ಪ್ಲಾಂಟ್ ಸತ್ತೋಗುತ್ತೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ವೈರಸ್ ಇನ್ಫೆಕ್ಷನ್. ಬಟ್ ಈ ಪ್ಲಾಂಟ್ ಅನ್ನ ನಾವು ಕಾಪಾಡ್ಕೊಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಅದ್ರ ಮೆರಿಸ್ಟಮ್ ಅನ್ನ ತೆಗೆದು ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಅಲ್ಲಿ ಬೆಳೆಸ್ತಾರೆ. ಅದನ್ನ ಮೆರಿಸ್ಟಮ್ ಕಲ್ಚರ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಮೆರಿಸ್ಟಮ್ ನಲ್ಲಿ ವೈರಸ್ ಇರಲ್ಲ. So to obtain virus-free plants in laboratory condition, you are going to use what is called as meristem culture. Last concept that you have studied in this chapter is about something called as somatic hybridization. You cannot subject any plants to hybridization based on your will and wish. ನಿಮಗೆ ಬೇಕಾದಾಗೆ ಯಾವ ಪ್ಲಾಂಟ್ ಅನ್ನು ಬೇಕು ಆ ಪ್ಲಾಂಟ್ ಅನ್ನ ಹೈಬ್ರಿಡೈಸೇಷನ್ ಒಳಪಡಿಸಕ್ ಆಗಲ್ಲ. ಅದರಿಂದ ಆಲ್ಟರ್ನೇಟಿವ್ ಆಗಿ ಯು ಕ್ಯಾನ್ ಗೋ ಫಾರ್ ವಾಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಸೊಮ್ಯಾಟಿಕ್ ಹೈಬ್ರಿಡೈಸೇಷನ್ ಅಂತ ಕರೀತಾರೆ. ಅಂದ್ರೆ ಡೈರೆಕ್ಟ್ ಆಗಿ ಎರಡು ಪ್ಲಾಂಟ್ಸ್ ಗಳ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ತಗೊಂತಾರೆ. ಇದೆರಡು ಹೈಬ್ರಿಡೈಸೇಷನ್ ಮಾಡಕ್ ಕಷ್ಟ ಆಗುತ್ತೆ ಅಂತ ಅನ್ಕೋಣ. ಅವಾಗ ಇದರ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಅನ್ನ ಮತ್ತೆ ಇನ್ನೊಂದರ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಎರಡನ್ನು ಫ್ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಹಾಗಾದ್ರೆ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಅಂದ್ರೆ ಅರ್ಥ ಏನು? ಪ್ಲಾಂಟ್ ಸೆಲ್ ವಿಥೌಟ್ ದ ಸೆಲ್ ವಾಲ್. ಪ್ಲಾಂಟ್ ಸೆಲ್ ಇಂದ ಸೆಲ್ ವಾಲ್ ತೆಗೆದಾಕಿದ್ರೆ ಅದನ್ನ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಅಂತ ಕರೀತಾರೆ. ಆ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಗಳನ್ನ ತೆಗೆದು ಫ್ಯೂಸ್ ಮಾಡೋದನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಕಲ್ಚರ್ ಅಂತ ಹೇಳಿ, ಅದರಿಂದ ಬರೋ ಹೈಬ್ರಿಡ್ ಅನ್ನ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಸೊಮ್ಯಾಟಿಕ್ ಹೈಬ್ರಿಡೈಸೇಷನ್ ಅಂತ ಕರೀತಾರೆ. ಸೊಮ್ಯಾಟಿಕ್ ಹೈಬ್ರಿಡೈಸೇಷನ್ ಇಂದ ಐ ಹೋಪ್ ಯು ನೋ ಒನ್ ಆಫ್ ದ ಮೋಸ್ಟ್ ಸಕ್ಸಸ್ಫುಲ್ ಸ್ಟೋರಿ ವಾಸ್ ಟು ಹೈಬ್ರಿಡೈಸ್ ಅಂಡ್ ಟೊಮ್ಯಾಟೊ ಪ್ಲಾಂಟ್ ವಿತ್ ಪೊಟ್ಯಾಟೊ ಟು ಗೆಟ್, ಇದು ಪೊಟ್ಯಾಟೊ ಅಲ್ಲ, ದಿಸ್ ವಾಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಟೊಮ್ಯಾಟೊ ಅಂತ ಕರೀತಾರೆ. ಇಟ್ ವಾಸ್ ದ ಕಾಂಬಿನೇಷನ್ ಆಫ್ ಟೊಮ್ಯಾಟೊ ಅಂಡ್ ಪೊಟ್ಯಾಟೊ, ಬಟ್ ಅನ್ಫಾರ್ಚುನೇಟ್ಲಿ ಇಟ್ ಕುಡ್ ನಾಟ್ be commercially utilized. Yeah, this is about synopsis of this chapter. If you have any doubts, you can let me know at any point during this question and when we are solving the questions. First, let us start solving the questions from this chapter. Solpa fast agogana so that we can cover both plant as well as animals. Quick agi ಹೇಳ್ತಾ ಹೋಗೋಣ. Which of the following techniques plays a pivotal role in further enhancing of food production? Food production ಗೆ ಮ್ಯೂಟೇಶನಲ್ ಬ್ರೀಡಿಂಗ್ ನೋಡಿದೀವಿ, ಟಿಶ್ಯೂ ಕಲ್ಚರು ಮಾಡಬಹುದು, ರಿಕಾಂಬಿನೆಂಟ್ DNA ಟೆಕ್ನಾಲಜಿ ಮುಖಾಂತರ ಜೀನ್ಸ್ ಗಳನ್ನ ಬೇರೆ ಬೇರೆ ಕಡೆಗೆ ಟ್ರಾನ್ಸ್ಫರು ಮಾಡಬಹುದು. ಈ ಮೂರು ಮೆಥಡ್ ಗಳನ್ನ ಯೂಸ್ ಮಾಡಿ we can enhance the food production. So the answer should be all of the above. Next question. Which of the following is the route for any breeding program? Without options, ನೀವು ಹೇಳ್ಬೋದು. Genetic variability, emasculation, selection, bagging. ಯಾವುದೇ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮ್ ಮಾಡಬೇಕು ಅಂದ್ರೂ ಅದರ ಬೇಸಿಕ್ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಜರ್ಮ್ ಪ್ಲಾಸಮ್ ಕಲೆಕ್ಷನ್. ಜರ್ಮ್ ಪ್ಲಾಸಮ್ ಕಲೆಕ್ಷನ್ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಬೇರೆ ಬೇರೆ ತರದ ಜೆನೆಟಿಕ್ ವೇರಿಯಬಿಲಿಟಿ ಇರುತ್ತೆ, which may be in even wild plants ಗಳಲ್ಲೂ ಇರಬಹುದು. ಹಾಗಾದ್ರೆ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಗೆ ಬೇಕಾಗಿರೋ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ಪ್ರಾಪರ್ಟಿ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಜೆನೆಟಿಕ್ ವೇರಿಯಬಿಲಿಟಿ ಆರ್ ಕಲೆಕ್ಷನ್ ಆಫ್ ಜರ್ಮ್ ಪ್ಲಾಸಮ್. ಇದು ಕ್ವಿಕ್ ಆಗಿ ಮೂವ್ ಆಗ್ತೀನಿ. ದಿಸ್ ಆರ್ ವೆರಿ ಸಿಂಪಲ್ CET ಕ್ವಶನ್ಸ್. ಎಂಟೈರ್ ಕಲೆಕ್ಷನ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಆರ್ ಸೀಡ್ಸ್ having all diverse alleles for all genes in a given crop is called as germplasm collection. Very good. I can see at least some of you are giving the answers, and that too you are responding very quickly. This really encourages us to solve questions. Wonderful. Namig encouragement irli bidli, ನಿಮ್ಗಂತೂ ಇದು ಸ್ಪೀಡ್ ಮತ್ತು accuracy ಕಾನ್ಫಿಡೆನ್ಸ್ ಎಲ್ಲ ಜಾಸ್ತಿ ಆಗ್ಲಿಕ್ಕೆ ಕ್ವಶನ್ಸ್ ಗಳನ್ನ ಸಾಲ್ವ್ ಮಾಡೋದು ಇಂಪಾರ್ಟೆಂಟ್. Next, Sonalika and Kalyan Sona are varieties of. Again, without options, you should be able to tell it. Rice variety ನೀವು ನೋಡಿರೋದು ಎರಡು ನೋಡಿದ್ದೀರಾ. ಯಾವ್ದು? ಒಂದು ಜಯ, ಇನ್ನೊಂದು ರತ್ನ. Both of them were developed in India only. ಅದರ ಜೊತೆಗೆ IR8, which was introduced from International Rice Research Institute. ಇನ್ನೊಂದು it is called as Taichung native, which was introduced from Taiwan. ಮೇಜ್ ಇಂದ ಯಾವುದು ಎಕ್ಸಾಂಪಲ್ ಇಲ್ಲ ನಿಮ್ ಬುಕ್ ನಲ್ಲಿ. ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಇದಕ್ಕೆ ಏನಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಶುಡ್ ಬಿ ವೀಟ್. ಕೊಂತೀರಾ? ಸೊ ಶುಗರ್ ಕೇನ್ ಅಲ್ಲಿ 2 ವೆರೈಟೀಸ್ ನೋಡಿದ್ದೀರಾ? ವಿಚ್ ಆರ್ ದೋಸ್? One is Saccharum berberi, which was growing in North India. Other one is Saccharum officinarum, which grows in South India. Daya and Ratna are the varieties of rice. We have just now seen it. Saccharum berberi is a variety of sugar cane. Yelliruthe. Saccharum berberi, it is found in North India, but yield it will be less. ಸೌತ್ ಇಂಡಿಯಾದಲ್ಲಿ ಬೆಳೆಯುತ್ತೆ, ಈಲ್ಡ್ ಜಾಸ್ತಿ ಇರೋದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಸ್ಯಾಕ್ರಮ್ ಅಫಿಷನಾರಮ್. ಸೊ ಇದಕ್ಕೆ ಆನ್ಸರ್ ಏನಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಈಸ್ ದ ವೆರೈಟಿ ಆಫ್ ಶುಗರ್ ಕೇನ್. ನೆಕ್ಸ್ಟ್, ವಿಚ್ ಆಫ್ ದ ಫಾಲೋಯಿಂಗ್ ಇಸ್ ಅ ಫಂಗಲ್ ಡಿಸೀಸ್? ಬ್ರೌನ್ ರಸ್ಟ್ ಆಫ್ ವೀಟ್, ರೆಡ್ ರಾಟ್ ಆಫ್ ಶುಗರ್ ಕೇನ್, ಲೇಟ್ ಬ್ಲೈಟ್ ಆಫ್ ಪೊಟ್ಯಾಟೊ, ಆಲ್ ಆಫ್ ದಿಸ್. ಈಗ ತಾನೇ ಒಂದು ಸ್ಲೈಡ್ ಅಲ್ಲಿ ನಿಮ್ಗೆ ಫಂಗಲ್ ಡಿಸೀಸ್ ಯಾವುದು, ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಡಿಸೀಸ್ ಯಾವುದು, ಮತ್ತೆ ವೈರಲ್ ಡಿಸೀಸ್ ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ಬ್ರೌನ್ ರಸ್ಟ್ ಮತ್ತೆ ಸ್ಮಟ್ ಅಂತ ಡಿಸೀಸ್ ಕೇಳಿದ್ದೀರಾ. ಅದು ಫಸ್ಟ್ ಸೆಕೆಂಡ್ ಚಾಪ್ಟರ್ ಬಯಾಲಜಿಕಲ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಓದುವಾಗ ಫಂಗಲ್ ಕಿಂಗ್ಡಮ್ ನಲ್ಲಿ ರಸ್ಟ್ ಅಂಡ್ ಸ್ಮಟ್ ಡಿಸೀಸ್ ಆರ್ ಕಾಸ್ಡ್ ಬೈ ಫಂಗಸ್ ಅಂತ. ದಿಸ್ ಇಸ್ ಫಂಗಲ್ ಡಿಸೀಸ್. ರೆಡ್ ರಾಟ್, ನೀವು ಕಬ್ಬನ್ ತಿನ್ಬೇಕಾದ್ರೆ ನೋಡಿರ್ತೀರಾ, ರೆಡ್ ಕಲರ್ ಒಂದು ಲೈನ್ ತರ ಹೋಗಿರುತ್ತೆ. That is red rot of sugarcane, which is also caused by fungus. Late blight of potato, which is caused by Phytophthora infectious. Idelladu kuda agirodrinda answer should be fourth one. Next question. Good. Very good. In mung being resistant to yellow mosaic virus and powdery mildew were induced by, almost nim question alla answer sikkogutte. Induce ಮಾಡೋದು ನಿಮಗೆ polyploidy breeding ಇಲ್ಲ ಬಿಡಿ, ನಿಮ್ ಸಿಲೆಬಸ್ ನಲ್ಲಿ ಇಲ್ಲ. Selection is not an induction, hybridization is also not an induce ಮಾಡೋದಲ್ಲ. Induce ಮಾಡೋದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ it is by mutational breeding. ನಾನು ನಿಮ್ಗೆ ಪದೇ ಹೇಳ್ತಾ ಇದ್ದೆ ಆಗ್ಲೇ, ಪ್ಲೀಸ್ ಮೇಕ್ ಶ್ಯೂರ್ ದಟ್ ಯು ಗಿವ್ ಮೋರ್ ಇಂಪಾರ್ಟೆನ್ಸ್ ಫಾರ್ ಮೆಮರೈಸಿಂಗ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್ ಅಂತ. ಈ ರೀತಿ ಬೇರೆ ಎಕ್ಸಾಂಪಲ್ಸ್ ಗಳನ್ನ ಬೇರೆ ರೀತಿನಲ್ಲಿ ಕೇಳ್ತಾರೆ. ಸೊ ಅಗೈನ್ ಸಿಂಪಲ್ ಡೈರೆಕ್ಟ್ memory based question. Rabani Kranti is a variety of rice dalla, citrus dalla, beans dalla. It is a variety of bendi. Atlas 66 is a variety of. Norna, let's take up this. Can anyone tell me answer for this without option? Option ildale. Atlas 66 Raisa Lavanya. Chandru, wheat. Yen Vishesha adralli. Correct. Wheat is correct. What is the speciality of this Atlas 66 wheat variety? I think ninge sari board allu athwa yavadlo nodi ಜ್ಞಾಪಕ ನನಗೆ ಯಾವುದು ನಿಮ್ಮ preparatory exam allo rich in protein. Very good. So correct. So Atlas 66 is an wheat variety which will be having high variety of protein, and hence it is used to plant breeding technique for the purpose of biofortification igoskara use ಮಾಡ್ತಾರೆ ಇದನ್ನು. So answer should be first one, that is wheat variety. The most popularly used algae as single cell protein is Anabenana. Single cell protein agi use ಮಾಡಲ್ಲ, but I hope you know Anabena is an cyanobacteria. Anabena is an cyanobacteria which is meant for nitrogen fixation. Nostoc is also an cyanobacteria meant for nitrogen fixation. Spirogyra is an algae, but it is not used as single cell protein. Answer should be spirulina. Spirulina ಜೊತೆಗೆ ಇನ್ನೂ ಕೆಲವು ಎಕ್ಸಾಂಪಲ್ ನಾವು ನೋಡಿರೋದು ಕ್ಲೋರೆಲ್ಲಾ ಅಂತ ನೋಡಿದೀವಿ, ಇನ್ನೊಂದು ಮಿಥೈಲೋಫಿಲಸ್ ಮಿಥೈಲೋಟ್ರೋಫಸ್ ಅಂತ ನೋಡಿದೀವಿ. ಬರೀ ಅಂತ ಹಾಕ್ತೀನಿ, ಮಿಥೈಲೋಫಿಲಸ್ ಮಿಥೈಲೋಟ್ರೋಫಸ್. ಇನ್ನೊಂದು ನಾವು ನೋಡಿರೋ ಸಿಂಗಲ್ ಸೆಲ್ ಪ್ರೋಟೀನಿಗೆ ಮಶ್ರೂಮ್ ಅನ್ನ. Sometimes even yeast is also used as single cell protein. ಇದನ್ನ ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ, ಮಿಥೈಲೋಫಿಲಸ್ ಮಿಥೈಲೋಟ್ರೋಫಸ್ ಏನಿದೆಯೋ it can be easily grown in even some industrial waste. Potato industries ಗಳಿಂದ ಏನಾದರೂ ವೇಸ್ಟ್ ಬಂದ್ರೆ ಅದನ್ನು ಯೂಸ್ ಮಾಡಿಕೊಂಡು you can grow ಮಿಥೈಲೋಫಿಲಸ್ ಮಿಥೈಲೋಟ್ರೋಫಸ್. ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸಿಂಗಲ್ ಸೆಲ್ ಪ್ರೋಟೀನ್ಸ್ ಈಗ ತಾನೇ ನೋಡಿದ್ವಿ. ಸ್ಪೈರುಲಿನಾ ಇಸ್ ಅನ್ ಆಲ್ಗೆ, yeast is an fungus, chlorella is an algae, whereas Mithylophilus mithylotrophus is an bacteria. So answer ಏನಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ it should be fourth one. Very good. Cellular totipotency refers to, ಇದು ನೀವೇ ಆನ್ಸರ್ ಹೇಳಿ. I am not going to tell the answer. You have to give me the answer for this. One minute. So most of the answers says it is 4, but Chandru, username by the name Chandru, says it is 3. Let's see. Development of organ from a cell or a medium cannot be called as totipotency. It will be called as organogenesis ಅಂತ ಕರೀತಾರೆ. ಟೋಟಿಪೊಟೆನ್ಸಿ ಅಂತ ಹೇಳಿದ್ರೆ ಕಂಪ್ಲೀಟ್ ಪ್ಲಾಂಟ್ ಬರಬೇಕು, ಪೂರ್ತಿ ಗಿಡ ಬರಬೇಕು. Development of complete plant from a cell in a tissue culture medium, it is called as totipotency. So answer should be fourth one. Next, plant part which is used as inoculum to grow the plant or organs in vitro is called as. Plant ಇಂದ ತಗೊಂಡು ಇನ್ ವಿಟ್ರೋ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಬೆಳೆಸಕ್ಕೆ ಏನನ್ನು ಉಪಯೋಗಿಸ್ತಾರೋ ಅದನ್ನ ಏನಂತ ಕರಿತೀರಾ? We have seen all of this in one. ಇದೊಂದು ಬಿಟ್ಟು ನಿಮ್ ಸಿಲೆಬಸ್ ನಲ್ಲಿ ಇಲ್ಲ. We saw callus is a mass of undifferentiated cells ಅಂತ ನೋಡಿದ್ವಿ. A mass of undifferentiated cells ಅಂತ. Whereas tissue is found everywhere. It is plants, animals, common ಆಗಿರುತ್ತೆ. It is a group of cells which are having similar structure, origin. ಅದನ್ನ you call it as tissue ಅಂತ ಕರಿತೀರಾ. ಆನ್ಸರ್ ಆಗೋದ್ರಲ್ಲಿ ಏನಾಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ explant, which is the part of the plant which is taken for tissue culture. Resistance to yellow mosaic virus in Bindi was transferred from wild species, which resulted in knee variety. ಇದನ್ನ ಈಗಾಗಲೇ ನೋಡಿದೀವಿ. yellow mosaic virus ಗೆ ರೆಸಿಸ್ಟೆನ್ಸ್ ಆಗಿರೋ ಬೆಂಡಿ, it will be ಪ್ರಭಾನಿ ಕ್ರಾಂತಿ. Iron and calcium enriched vegetable crops are ಕಬ್ಬಿಣದಂಶ ಮತ್ತು ಸುಣ್ಣದಂಶ ಅಥವಾ ಕ್ಯಾಲ್ಸಿಯಂ ಜಾಸ್ತಿ ಇರೋದು ಯಾವುದರಲ್ಲಿ? It will be in case of spinach and batua. See, ಈ ಕ್ವಶನ್ಸ್ ಗಳು ನನಗೆ ಇಲ್ಲಿ ಆನ್ಲೈನ್ ಅಲ್ಲಿ ಸಾಲ್ವ್ ಮಾಡ್ಸಕ್ಕೆ ಬೋರಿಂಗ್, ಯಾಕಂದ್ರೆ ಇದರಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲನೇಷನ್ ಇಲ್ಲ. ಬಟ್ ಸ್ಟಿಲ್ ಆದ್ರೂ ಕೂಡ ನಾನು ಈ ಕ್ವಶನ್ಸ್ ಗಳನ್ನ ಹಾಕಿದ್ದೀನಿ ಫಾರ್ ದ ರೀಸನ್ that you should understand questions most often they will come in the form of examples. Adukoskara, I am repeatedly telling this. Please make sure that you are going to spend time on memorizing all these examples. Next question. Physical barriers used in somatic hybridization are cell wall. Somatic hybridization anta ಹೇಳಿದ್ರೆ we saw fusion of two cells ಅಂತ. ಎರಡು ಸೆಲ್ಸ್ ಗಳನ್ನ ಫ್ಯೂಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಪ್ರಾಬ್ಲಮ್ ಏನಾಗುತ್ತೆ ಅನ್ನೋದು ಫಸ್ಟ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. ವಾಟ್ ಇಸ್ ದ ಪ್ರಾಬ್ಲಮ್ ಇಸ್, plant cells are covered by something called as cell wall. And cell wall are rigid structure. ನೀವು ಇದೆರಡನ್ನು ಫ್ಯೂಸ್ ಮಾಡಕ್ ಹೋದ್ರೆ ಅದೆರಡು ಫ್ಯೂಸ್ ಆಗಲ್ಲ. They do not get fused because it will be having cell wall which will be rigid structure. ಹಾಗಾದ್ರೆ ಈ cell wall ಅನ್ನ ತೆಗೆದಾಕ್ಬೇಕು. If you want to remove the cell wall, you are going to treat these with cell wall degrading enzyme. You know what is cell wall is made up of? Cell wall is made up of cellulose, hemicellulose, cementing material ಆಗಿ ಅದರಲ್ಲಿ ಪೆಕ್ಟಿನ್ ಕೂಡ ಇರುತ್ತೆ. ಹಾಗಾದ್ರೆ ಇದನ್ನ ತೆಗೆದಾಕಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ you are going to treat it with enzymes such as cellulase. Cellulose ಅನ್ನ ಬ್ರೇಕ್ ಮಾಡ್ಲಿಕ್ಕೆ cellulase ಬೇಕು. ಅದೇ ತರ ಪೆಕ್ಟಿನ್ ಅನ್ನ ಬ್ರೇಕ್ ಮಾಡ್ಲಿಕ್ಕೆ ಪೆಕ್ಟಿನೇಸ್ ಬೇಕು. ಇಲ್ಲಿ ಅಮೈಲೇಸ್ ಬೇಕಾಗಿಲ್ಲ, ಇಟ್ ಇಸ್ ಮೆಂಟ್ ಫಾರ್ ಡೈಜೆಷನ್ ಆಫ್ ಸ್ಟಾರ್ಚ್. ಅಮೈಲೇಸ್ ಬೇಕಾಗಿಲ್ಲ, ಸೆಲ್ಯುಲೋಸು ಬೇಕಾಗಿಲ್ಲ, ಬೇಕಾಗಿರೋದು ಸೆಲ್ಯುಲೇಸ್. ಪ್ರೋಟಿಯೇಸ್ ಬೇಡ, ಯಾಕಂದ್ರೆ ಸೆಲ್ ವಾಲ್ಸ್ ಆರ್ ನಾಟ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಪ್ರೋಟೀನ್. ಅದರಿಂದ ಆನ್ಸರ್ ಶುಡ್ ಬಿ ಸೆಕೆಂಡ್ ಒನ್. Hidden hunger is due to diet rich only in some nutrients, diet lacking essential micronutrients, both 1 and 2, none. So hidden hunger anta ಹೇಳಿದ್ರೆ it is a diet. Diet anta ಹೇಳಿದ್ರೆ food that we eat. Diet lacking essential micronutrients is considered as hidden hunger anta ಕರೀತಾರೆ. Tomato ಗೊತ್ತಿದೆ ನಿಮ್ಗೆ, ಇದನ್ನ ಕ್ವಿಕ್ ಆಗಿ ಸ್ಕಿಪ್ ಮಾಡ್ತೀನಿ. Tomato was produced by hybridization between potato and tomato, which was produced by the technique called as somatic hybridization. ಈ ಕ್ವಶನ್ ನೀವೇ ಸಾಲ್ವ್ ಮಾಡಿ. Meanwhile, I'll just quickly look into your Q&A. ಅವಾಗಿಂದ Q&A section ನೋಡಿಲ್ಲ. If you have any doubts, let me see that and then proceed. Meanwhile, ಸ್ಕ್ರೀನ್ ಮೇಲೆ ಇರೋ ಕ್ವಶನ್ಸ್ ನ ಸಾಲ್ವ್ ಮಾಡಬೇಕು. ನೋ ಡೌಟ್ಸ್. ಆನ್ಸರ್ ಏನಾಗ್ಬೇಕು? ಕೂಸಾ ಗೌರವ್. Pusa Gaurav variety of brassica is going to show what is called as pest and insect resistant. Illinda ಸ್ವಲ್ಪ complicated multiple choice questions ಇದೆ. The phase referred to green revolution during which varieties are developed? Rice, wheat, maize, sugarcane ಅಂದ್ರೆ ಮೇನ್ ಆಗಿ ಎರಡು ವೆರೈಟೀಸ್ ಗಳು ಗ್ರೀನ್ ರೆವಲ್ಯೂಷನ್ ಅಲ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಜಾಸ್ತಿ ಆಗಿದ್ದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ರೈಸ್ ಮತ್ತೆ ವೀಟ್. Varieties of wheat are, this is rice variety, this is rice variety. Sonalika and Kalyan Sona are the examples of wheat varieties. So ಎಲ್ಲೆಲ್ಲಿ ಈ ತರ again examples ಗಳಿದೆಯೋ, wherever we see the examples, will go little fast. Which of the following is viral disease? Late blight of potato is a fungal disease. Brown rust of wheat is again fungal disease, whereas tobacco mosaic virus and turnip mosaic virus causes tobacco mosaic and turnip mosaic disease. So the answer should be third one. Which of the following characters do not attract bollworm in cotton? Smooth leaves irbeku, nectarless flowers irbeku, solid stem in wheat. It is going to prevent the stem soft lie. I'm repeating, stem soft lie anna wheat ಅಲ್ಲಿ ಕಡಿಮೆ ಮಾಡಲಿಕ್ಕೆ ಸಾಲಿಡ್ ಸ್ಟೆಮ್ ಇರಬೇಕು. ಇಟ್ ಇಸ್ ನಾಟ್ ರಿಕ್ವೈರ್ಡ್ ಫಾರ್ ಕಾಟನ್. ಅದರಿಂದ ಥರ್ಡ್ ಅಂಡ್ ಫೋರ್ತ್ ಎರಡು ತಪ್ಪಾಗುತ್ತೆ. ಆನ್ಸರ್ ಶುಡ್ ಬಿ ಫಸ್ಟ್ ಅಂಡ್ ಸೆಕೆಂಡ್. Which of the following leads to resistance in maize stem borer? Stem borer ಏನೇನ್ ಇರಬೇಕು? ಹೈ ಆಸ್ಪಾರ್ಟಿಕ್ ಆಸಿಡ್ ಇರಬೇಕು, ಲೋ ನೈಟ್ರೋಜನ್ ಇರಬೇಕು, ಆಸ್ ವೆಲ್ ಆಸ್ ಲೋ ಶುಗರ್ ಕಂಟೆಂಟ್ ಇರಬೇಕು. ಹೈ ನೈಟ್ರೋಜನ್ ಇದ್ರೆ ಹುಳನ ಅಟ್ರಾಕ್ಟ್ ಮಾಡುತ್ತೆ, ಜಾಸ್ತಿ ತಿನ್ನತ್ತೆ. ಸೊ ಲೋ ನೈಟ್ರೋಜನ್, ಲೋ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಆರ್ ಶುಗರ್ ಕಂಟೆಂಟ್ ಅಂಡ್ ಹೈ ಆಸ್ಪಾರ್ಟಿಕ್ ಆಸಿಡ್. ಸೊ ಮೂರು ಇದ್ರೆ ಮೇಸ್ ಸ್ಟೆಮ್ ಬೋರರ್ ಅಲ್ಲಿ ಕಡಿಮೆ ಆಗುತ್ತೆ. ಇಲ್ಲಿವರೆಗೂ ವಿ ಸಾ ಅಬೌಟ್ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್. ಇಲ್ಲಿಂದ ಮುಂದಕ್ಕೆ ಅನಿಮಲ್ ಬ್ರೀಡಿಂಗ್ ಟೆಕ್ನಿಕ್, ಅಂಡ್ ವಿ ಹ್ಯಾವ್ ಅರೌಂಡ್ 20 minutes. So in next 20 minutes, at least 15 minutes we'll spend time on these questions, and last 5 minutes we'll look into any doubts if you have any questions. When breeding is between the animals of same breed, when breeding is between the animals of the same breed, it is called as dash. While cross between the different breed, it is called as same breed. Adre you will call it as inbreeding. Different breed adre you will call it as outbreeding. So answer should be B. Next question. The easy agide, answer madi. I want the answer from you. Meanwhile, I'll just check your question and answers if you have any questions. Wonderful. See, one thing is for sure, I'm very happy the way in which you people are responding. Kadme jana idrunu, whatever the interaction is happening, I'm very happy with this. Which one of the following is a new breed of sheep developed in Punjab? Punjab aliro ur hesru Isar. Isar ali ವೆಟರ್ನರಿ ರಿಸರ್ಚ್ ಸೆಂಟರ್ ಇದೆ. ಅಲ್ಲಿ ಬಿಕನೇರ್ ಈವ್ ವಿಚ್ ಇಸ್ ಅನ್ ಫೀಮೇಲ್ ಫ್ರಮ್ ರಾಜಸ್ಥಾನ್ ಅಂಡ್ ಮೆರಿನೋ ರ್ಯಾಮ್ ವಿಚ್ ಇಸ್ ಅನ್ ಮೇಲ್ ಫ್ರಮ್ ಯುರೋಪಿಯನ್ ಕಂಟ್ರೀಸ್, ದೀಸ್ ಟು ವೇರ್ ಕ್ರಾಸ್ಡ್ ಟು ಗೆಟ್ ಇಸಾರ್ ಬ್ರೀಡ್. ವರಾಸ್ ಇನ್ ಮಿಕ್ಕಿದ್ದೇನಿದೆಯೋ ಇದೆಲ್ಲ ಚಿಕನ್ ನ ವೆರೈಟೀಸ್ಗಳು. ಆನ್ಸರ್ ಶುಡ್ ಬಿ ಎ. ವಿಚ್ ಆಫ್ ದ ಫಾಲೋಯಿಂಗ್ ಸ್ಟೇಜ್ ಇಸ್ ಟ್ರಾನ್ಸ್ಫರ್ಡ್ ಟು ಸರೋಗೇಟ್ ಮದರ್ಸ್ ಇನ್ ಲೈಫ್ ಸ್ಟಾಲ್ ಬ್ರೀಡಿಂಗ್ experiment? We saw a method called as multiple ovulation embryo transfer. What are the steps that are involved in multiple ovulation embryo transfer? ಯಾವ ಸ್ಟೆಪ್ ಇರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಫಸ್ಟ್ it will be subjected for, superior female will be subjected for super ovulation, because of which she will produce around 6 to 8 eggs. That is what you call it as super ovulation. Super ovulation can be injected or induced by injecting FSH. Super ovulation admele, it will be mated with superior male, or there may be artificial insemination, and that produces multiple embryos. Embryos anna tagondu it will be transferred into surrogate mother. These are the different steps which are involved in multiple ovulation embryo transfer. So ಈ ಎಂಬ್ರಿಯೋನ ಯಾವ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ 8 to 32 celled ಎಂಬ್ರಿಯೋನ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರೆ. So the answer should be C. Next, which one of the following is or are marine fish? So rohu is example for freshwater. Katla is example for freshwater. Also the common carp. More example anna freshwater alli nodidire. Ilsa, mackerel, sardine and pomfret. These are the 4 fishes that you have seen under marine fishes. So the answer should be both B as well as C. Now, again, you should answer this now. Which of the following statements about breeding is incorrect? It's incorrect. Answer A. Okay. By inbreeding, pure lines cannot be evolved. It can be evolved. Inbreeding indane pure line barodu. Pure line anta helidre genetically speaking it should be called as homozygous condition anta karitare. Continued inbreeding, especially close inbreeding, reduces fertility and productivity. This is correct. Statement correct. Answer sari antalla. Statement correct. This is what you call it as inbreeding depression anta karitare. Crossbreeding allows desirable qualities of two different breeds to be combined. Idu correct statement. Question irodu identifying incorrect. Inbreeding exposes harmful recessive genes that are eliminated by selection. Idannu kuda with diagram we have seen during synopsis. This is also correct. Homozygous condition barodrinda recessive genes ಗಳು homozygous condition bandaga it will be expressed and hence it will be eliminated from the breeding program. ಹಾಗಾದ್ರೆ incorrect statement ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ by inbreeding pure line can be evolved but ಇಲ್ಲಿ cannot be evolved ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. Answer should be A. Next question. Read the following statement or statements and answer the questions. Animal husbandry is the industrial practice of breeding and raising the livestock. It is a correct statement. Industrial level alle ನಡೆಯುತ್ತೆ ಕೆಲವೊಂದು ಕಡೆ. Thousands of cattle are grown by individuals in a very large scale at the industrial scale itself. Dairy farm management deals with processes which improves the quality and quantity of milk production. That is also correct. Poultry is a class of domesticated birds which are used for food as well as egg. That is also correct. It may include chicken, duck, geese, turkey, any kind of domesticated birds. So answer should be D. That is all of this. Next question. I will wait for your answer. Which of the following statement or statements is or are correct regarding inbreeding? It is mating of more closely related individuals within the same breed for 4 to 6 generations. It is a correct statement. So one illa idrali C will be wrong. B also it will be wrong because one is right statement. It decreases homozygosity, wrong. It increases homozygosity, wrong statement. Hagadre second ellido the wrong agutte. A is also wrong. Answer should be D. It exposes harmful recessive genes that are eliminated by selection. Correct? Inbreeding depression results due to continued inbreeding, especially close inbreeding may reduce the fertility as well as productivity. Correct? Answer should be D. Which of the following includes all the characteristics described given below? In this method of breeding, mating of animals occurs with the same breed. Same breed ನಡುವೆ ಕ್ರಾಸಿಂಗ್ ಆಗ್ತಾ ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಯಾವುದು ತಪ್ಪು ಅಂತ ಹೇಳಿದ್ರೆ ಕ್ರಾಸ್ ಬ್ರೀಡಿಂಗ್ ತಪ್ಪಾಗುತ್ತೆ. They have no common ancestor on either side of their pedigree for 4 to 6 generations. No common ancestor. ಹಾಗಾದ್ರೆ ಇದು ತಪ್ಪಾಗುತ್ತೆ ಯಾಕಂದ್ರೆ ಇನ್ಬ್ರೀಡಿಂಗ್ ನಲ್ಲಿ it will be cross between superior male and superior female belonging to the same breed and have common ancestor for about 4 to 6 generations. So A tappagutte. It is best for those animals that are below average in productivity. Then this method and it is going to help in overcoming the inbreeding depression. ಯಾವ್ದಿದು ಅಂತ ಹೇಳಿದ್ರೆ It will be called as outcrossing anta ಕರೀತಾರೆ. ಇಲ್ಲಿ ಕೊಟ್ಟಿರೋ 4 ಸ್ಟೇಟ್ಮೆಂಟ್ಗಳು is best suited for outcrossing. Which of the following statement or statements is or are correct? Fishery is an industry which deals with catching, processing, marketing of fishes, as well as other aquatic animals such as prawns, crabs, lobster, edible oyster, etc., that have high economic value. This is correct statement. This is not correct then. Both aquaculture and pisciculture are same. I told you during synopsis, aquaculture includes growing of any aquatic animals. It may be fish, prawns, crabs, lobster, shells, elladu irutte aquaculture nalli. Whereas pisciculture is commercial growing of only fishes. So that cannot be same. ಹಾಗಾದ್ರೆ ಸೆಕೆಂಡ್ ಆಪ್ಷನ್ ಇರೋದೆಲ್ಲ ತಪ್ಪಾಗುತ್ತೆ. The development and flourishing of fisheries industries is called silver revolution. That is correct. White revolution for milk, silver revolution for fishes, green revolution for agriculture product, brown or yellow revolution for oil production. So idu sari ide. ಹಾಗಾದ್ರೆ answer should be C. Many people have deficiency as they cannot buy fruits and vegetables, because of which their diet will lack important minerals, vitamins, as well as proteins. ಏನಂತ ಕರೀತೀರಾ ಇದನ್ನ ಅಂತ ಹೇಳಿದ್ರೆ, this is called as biofortification. A matches with 4. That option illa illi. Let's see. Crops with higher vitamins. I'm sorry. A matches with 4, that is hidden hunger. So this it will be wrong. Crop with higher vitamins, proteins, and fats. Breeding of plants or crop which is having higher vitamins, proteins, and fats, it's called as biofortification. So B is 5. Growing microbes as alternative source of protein. It is called as single cell protein. C matches with 1. So then this is also not correct. This is also not correct. Answer should be C. Capacity to generate plants from single cell or explant. It is called as totipotency. Regeneration of entire plant from an explant is called as totipotency. Production of thousands of plants through tissue culture in a short span of time is called as micropropagation. Offsprings which are produced through micropropagation, which are genetically identical, they are called as somoclones. So F it is matches with 3. Next question. I just need to check your questions if there are any questions that check ಮಾಡ್ತೀನಿ. ಅಷ್ಟೊಳಗಡೆ try to answer this. Okay. 1 minute. Yeah. Semi-dwarf varieties of wheat. Sonalika, Kalyan Sona. A matches with 1 as well as 2. Semi-dwarf varieties of rice, all of them. So answer should be D. So I will take up this as probably last question. ಅದಾದ್ಮೇಲೆ if you have any doubts, ಇದಾದ್ಮೇಲೆ ಇನ್ನೊಂದು question ಇದೆ, ಅದನ್ನು solve ಮಾಡೋಣ. After that, if you have any doubts, we'll discuss about it. So choose a correct sequence during the process of hybridization. So first and foremost requirement ನಮಗೆ ಬೇಕಾಗಿರೋದು germplasm collection, which will be having all kinds of genetic variations, which are the basics for plant breeding. ಅದಾದ್ಮೇಲೆ ಬೇಕಾಗಿರೋದು evaluation of germplasm for our desirable characters. After evaluation, what we need to do is hybridization of selected parent. Hybridization involves following steps, which I hope you remember. It is emasculation, which is removal of anther or male reproductive structure. Bagging, which is meant for preventing unwanted pollen grains from pollinating. And then dusting, that is transfer of pollen grains from desirable plants. And then rebagging. So then it will go to obtaining progeny of F1 generation, and out of that progeny you have to select for superior recombinants, and it will undergo evaluation, agronomical testing, and then there will be release of new varieties. So the answer should be first one. Last question. Correct combination. Chili, Pusa, Komal, disease resistant, no. Flat bean, Pusa, Sem2, insect and pest resistant, correct. Bindi, this is the third time we are seeing while solving the question. Rabani Kranti, disease resistant, correct. Wheat variety, Himagiri, Ilbund, correct. So second, third, fourth, Muru, correct. The answer should be third one. So as I told you, last 5 minutes, we'll keep it for discussion on questions. If you have any, I will be online for next 5 minutes. If you have any doubts, please let me know. So ಈ ಎರಡು ಚಾಪ್ಟರ್ ನಾವೇನ್ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವೋ ಇದರಲ್ಲಿ ಏನಾದರೂ ಡೌಟ್ ಇದ್ರೆ ಹೇಳಿ. Please let me know if there are any doubts from these two chapters. That is evolution as well as strategies for enhancement in food production. How to remember all varieties of crops and disease to which these are resistant? I mean, can you say some tricks? See, I'm not so good at remembering the tricks, but I can tell you some of them. For example, nimge idantu gottagirlebeku. Sonalika Kalyan Sona, I'm sure you will remember it. IR, which comes from International Rice Research Institute, so rice variety. Taichung native, I'm again sure ಇಷ್ಟೊತ್ತೆಲ್ಲ ಓದಿರೋದ್ರಿಂದ ನಿಮಗೆ ಗ್ಯಾರಂಟಿ ಜ್ಞಾಪಕ ಇರುತ್ತೆ ಅಂತ ಅನ್ಕೊಂಡಿದ್ದೀನಿ. Taichung native. Sacrum, berry, just try to sacrum ಅನ್ನೋದು it comes from the word sugar, saccharides. Barberry ಅನ್ನೋದು it is North Indiaದಲ್ಲಿ ಇರುತ್ತೆ. Officinarum ಅನ್ನೋದು ಅಫಿಷಿಯಲಿ ಎಲ್ಲಾರು ಬೆಳೀತಾರೆ ಅಂತ ಮಾಡ್ಕೊಳ್ಳಿ. ಅಫಿಷಿಯಲಿ ಎಲ್ಲಾರು ಬೆಳೆಯೋದ್ರಿಂದ it is not the reason to call it as, but ನಿಮಗೆ ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಕ್ಕೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದೀನಿ. ಅಫಿಷಿಯಲ್ ಆಗಿ ಏನಕ್ಕೆ ಬೆಳೀತಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ ಅದರಲ್ಲೇ ಜಾಸ್ತಿ ಬ್ರೀಡ್ ಅಥವಾ ಅದರಲ್ಲೇ ಜಾಸ್ತಿ ಫುಡ್ ಪ್ರೊಡಕ್ಷನ್ ಇರೋದು ಅಥವಾ ಈಲ್ಡ್ ಜಾಸ್ತಿ ಇರೋದು. ಅದ್ ಬಿಟ್ರೆ ಇದೆಲ್ಲ ಎಲ್ಲದಕ್ಕೂ ಆಗಲ್ಲ. ಈಗ ಉದಾಹರಣೆಗೆ ಕಾಲಿಫ್ಲವರ್ ನೋಡಿ, ಇಟ್ ಇಸ್ ವೈಟ್ ಇನ್ ಕಲರ್, ಅದರಿಂದ ಇಟ್ ಇಸ್ ಶುಭ್ರತೆ ಇರುತ್ತೆ. ಅಥವಾ ಸ್ನೋಬಾಲ್, ಅಗೇನ್ ವೈಟ್ ಇನ್ ಕಲರ್. ಸೊ ದಿಸ್ ವೆರೈಟಿ ಇಟ್ ಇಸ್ ಫಾರ್ ಕಾಲಿಫ್ಲವರ್. ಪೀಕಾಕ್ ಅನ್ಕೊಳ್ಳಿ, ಪೀಕಾಕ್ ಕೋಮಲವಾಗಿರುತ್ತೆ, ಸೊ ಪೂಸಾ ಕೋಮಲ್. ಚಿಲ್ಲಿಗೆ ಅದ್ ಏನಕ್ಕೆ ಸದಾಬಹಾರ್ ಅಂತ ಕೊಟ್ಟಿದಾರೋ ಗೊತ್ತಿಲ್ಲ. ಚಿಲ್ಲಿ ಯಾವತ್ತು ಸದಾಬಹಾರ್ ಆಗಿರಲ್ಲ. I mean, it is too hot. But anyway, these are some of the ways. I mean, different people will be having different methods for remembering it. So, right to remember this. ಇದಂತೂ ನಿಮ್ಗೆ ಇವತ್ತು ಜ್ಞಾಪಕ ಆಗಿರ್ಬೋದು. ಬೆಂಡಿ ಪದೇ ನೋಡಿದೀವಿ, ಪೂಸಾ, ಸವರ್ಣಿ. So these are the ways. So I think I'm not suitable to answer that last question. That I don't know anything in physics. Can I get good marks in KCET? No. You should be having something. You should be knowing at least something about physics. You bari chemistry, maths, biology melene KCET marks ತೆಗಿಯಕ್ಕಾಗಲ್ಲ. So ಕಷ್ಟ ಆಗುತ್ತೆ. You have to work and score marks in physics also. See, brain capacity ಒಂದು ಅರ್ಥ ಮಾಡ್ಕೊಬೇಕಾಗಿರೋದು, you have to just look into the sequence. Like earlier ancestors ಗಳಿಗೆ ತುಂಬಾ ಕಡಿಮೆ brain capacity ಇತ್ತು. As the evolution went on, the brain capacity went on increasing. So again, memory ಮಾಡ್ಕೊಳ್ಳೇಬೇಕು. ಬರ್ದು practice ಮಾಡಿ. Yeah, it's 8 o'clock. So we will end the session.
Hello.
Yes, sir.
Time is up, sir.
Yeah, yes.
Thank you so much. Dear student, thank you so much. As one of the students wrote physics, don't know. At least you might have attended your board classes, no? Based on the board classes, 25% of the questions would be easy and moderate. Those questions you can answer. And there is no negative marks in CET. Attempt the questions which you have little doubt. With that, I think you will be able to do good. Hope you are good with chemistry and biology. And altogether, I hope you would be getting good marks. Work hard. If you are having more interest towards chemistry and biology, work hard towards that. At least whatever the marks less you are getting in physics, that can be nullified with the 2 subjects. Any other questions? I have left my number in chat box. You can just message me. I will be available to answer you. Thank you so much. Happy learning, dear students. Thank you, one and all.